anybody there, just tell me that tell me if the mic working, tell me if everything coming through. We're starting just now in about 8 minutes.
This is your brain. This is Spurwin. This is your brain on Spurwin. Any questions?
Good afternoon, everybody. Welcome to Spurwin Live Streams, Biology Cape Unit 2. If all you really think about it, and if this is the last paper 2 that you're doing, this is the last paper 2 that you'd probably have to do in your whole life. So you have to absorb it tomorrow. Yeah. I don't know if I can remember if it's an AM or a PM exam, but take it in. No more paper 2s after this. Right, all yeah. So let me get into the order of the day here. It's 2:05. But first, let me talk to all you a little bit about, well, what you're going to do after CXC. What were you going to do after CXC? You could play games, you could watch TV, you could watch anime, and this is why, Omaira wo shinderu. You are already dead unless you sign up for Crunchyroll today. Well, this is what Crunchyroll is for. You sign up for the thing. That's what we're here for today. By using Crunchyroll, you can get free 30-day trial and then you watch all the anime that you want. You watch all the Naruto and Boruto and Bleach and any Neon Genesis, Evangelion. You watch all the anime for free. You want to watch all the latest anime ad free after CXC, after Cape. You have nothing to do. You watch anime. You have all them summer anime out there. You're watching anime. In fact, if you love anime so much, you could even become an anime yourself. Yeah, man. But first, you have to sign up for Crunchyroll. You won't regret it. Watch anime when CXC done. Laters. Back to the video. Alright, so let's get started. Hold on, I'm up about this chat here. That's not right. Right, so I'll tell you now, what we're doing today is more content review than past papers. We'll do the occasional past papers. So But if you check my channel, now I say this running from 2 p.m. to 10 p.m. It mightn't run to 10 p.m. It might run to a little before that. Given that I find all the gear away this year, it had no action potential, it had no translocation, it had no Krebs cycle, it had none of them things. I find all they kind of get through this year. So I say it's more content review. If you check my channel, what I'll do is that I'll post a list of past papers, the answers, the solutions. But if you check my channel, I have videos going through the theory and so on of probably the last 6 years or so. Anyhow, let me get started today. So the first order of the day here, let me know if music or anything. Let me know if music too loud. So I'll know to turn it down a little bit. So the six broad topics that we have, right, is I think it's 6 is photosynthesis and ATP synthesis. That is basically that's one chapter that doesn't really include respiration. So I'm not going to go through the non-broad topics here. Feel free to read up on it for yourself though, but I doubt that if it come for like a little 1 mark and thing. So all this also know the thing anyway. All they get broad topics, all they good to go. Everybody else didn't get no broad topics more than 2 years ago, all they good to go. So I think it's photosynthesis in module 1 plus biodiversity and conservation. Well, I make the damn video, so I should remember. Transpiration in module 2. And mammalian heart, well, the circulatory system. And module 3 is health and disease, which is a kind of chain-up topic in my opinion, because that could come with almost anything. And immunology, which is probably one of the longest parts of that module. So let me get my chat pop out here. So let me start. Let me start with photosynthesis. Music too loud? Let me know if music better now. Is that smooth out the smooth before we start. Alright, better? Okay. Let me go all here. Alright. So first, let me understand a thing or two about ATP. ADP and ATP, right? This is the beginning. If you know us, this is kind of going through, but let me go through it anyway. So first of all, so we have ATP and we have ADP. And of course, ATP stands for adenosine triphosphate. That's because it has 3 phosphate groups. Tri means 3. And this functions as the intracellular energy currency in all organisms. Remember, photosynthesis, also ATP synthesized here, not just in respiration. You get some ATP here too, because ATP is needed to power through some of the different things that are happening here. Right, that's now. I'm gonna get this chat to stick here. The game this is from is Valhalla. Is a visual novel. Right, so ATP, so you don't need to know the molecule ATP and thing. I'm just doing this as a preamble to when we go into photosynthesis. So this contains a nitrogenous base, which is adenine, a ribose sugar, which means that it has 5 carbons in it. Ribose is 5. Or pentose is 5, but it has a ribose sugar and 3 organic phosphate groups. So I say, remember, it's 3. So when these bonds are broken, So when these bonds are broken here, between these three, you basically get energy being released. So ADP has two inorganic phosphate groups. So if it was to break off one of these phosphate groups, basically you will get ADP, right? So that's basically the gist of it. All you should know this already. Now, the word that some people up to this level is going to kind of chain up with is this word phosphorylation. When something gets phosphorylated, it just means that you add a phosphate to it. So for example, if I have a phosphate here and a phosphate here, and then I drag a next phosphate here, a next phosphate get binded to it, that is called phosphorylation. So why I say to keep track of this word is because photosynthesis involves something called photophosphorylation. So if you think about what that means, photo just means light. So it just means phosphorylation using light. So we'll get to that just now. Let me slow down a little bit. And let me just get into the basics of what photosynthesis is, right? So photosynthesis, all you should know this from primary school, all you should know this from CSEC, all you should just know this, man, all you read books. So the photosynthesis is where producers or autotrophs take in organic, it's not coffee talk. It's not that one. Anyway, so it takes in inorganic molecules, right? And this would be like carbon dioxide and water, and produce organic compounds such as glucose, and glucose would eventually be converted to starch. All you know this. So this occurs in chloroplasts, which are in green plants, right? So this would be in the leaves, which contain chlorophyll. Of course, there are different types of chlorophyll, but you don't need to know that for your exam. There's also different types of pigments, but we won't really get into that. So chlorophyll is not the only one, but chlorophyll is the green one. So this has a light-dependent stage and a light-independent stage. So, which basically means that one stage uses light, the next one doesn't need to use light. Long time they used to call it the light stage and the dark stage. Now they kind of, call it light-dependent and light-independent. So, we know that if we look at the structure of the chloroplast here, so if you look over here, we could see we need to take it really into account, just really two parts of it. Unless they ask you to label it, but I don't think there's really bring that. That's more common unit 1, where they talk about cells and things. But there's two parts here that we really have to take note of: the thylakoid membranes and the stroma. So the thylakoid membranes is basically where you are getting the light-dependent reaction. So the light-dependent reaction is, of course, the one that, you take sunlight, and if you remember from CSEC biology, this is where water is used to split. Water is split by light energy, and this is called photolysis. And this is where the light-dependent reaction occurs, and this is in the thylakoid membranes. A stack of thylakoid membranes is called a granum, and it's kind of look like a stack of green pancakes, I like to describe it. But on the surfaces and within the membranes of that, we have different molecules. We have enzymes like ATP synthase, and that helps to make ATP. We have different antennae complexes to kind of catch the photons from the light. And we have different what we call electron carriers. Those basically bounce off electrons. So the electron is kind of like a pinball machine in there. You go bling, 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 but except of a ball, it have an electron. And it bouncing off all these little protein complex, chlorophyll protein complexes in there. So visualize like a pinball machine. And you go pling, 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 pang, pang, ping, 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 ping. That's the electron. And that electron basically going from like high energy to low energy. Kind of like a ball. And you bounce in the ball, and eventually the ball runs out of energy. So that's basically the light-dependent reaction to start that whole pling, 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 pling process. The light-independent reaction is called the Calvin cycle. So I exactly get a little thing called the Calvin cycle because Calvin was kind of selfish now. His name was Melvin Calvin, right? We get a little bacchanal here. So Melvin Calvin, now, he was working with a man named Benson. And he's like Benson, he didn't show up, it was Benson who really kind of helped him come up with it. And so Benson was like, well, yeah, put my name there. Calvin's, nah, you're fired. Get out of here. So Calvin win the Nobel Prize, Benson get nothing. And they wanted to basically call it I think it's the ribulose biphosphate pentate cycle or something like that. But I find that name wasn't sounding sexy enough. So Calvin was like, we are calling the Calvin Benson cycle. That song is stupid. We're just calling it the Calvin cycle because it's me. So, whatever. It is kind of the same thing that had happened with Krebs, but with Krebs, the situation was a little different. With Krebs, he didn't want it to be called the Krebs cycle. Hans Krebs, he didn't want it to be called the Krebs cycle. He wanted it to be called the citric acid cycle. And people was like, nah, man, Krebs, we call it the Krebs as citric acid. That's something stupid. So we just call it the Krebs cycle. And so that thing was kind of named against Hewell. So, but that's a different man. Krebs are good, man. Calvin, not so good. So anyway, for the sake of ease though, and for all the learning in school, I go call it the Calvin cycle, right? Anyway, so this occurs in the stroma. So what exactly is the stroma here? So the stroma is basically the background material in the chloroplast. So you can think of the thylakoid membranes as the foreground. The stroma is like the background. And in the background it's kind of dark, so you don't need no light for any back day. So yeah, it's kind of like, Calvin, he copies somebody labs and he won, next person get 2, man, he make meritless, all kind of so that basically what happening. But as I say, the things you gotta keep track of here: thylakoid membranes, stroma, forget everything else for now, right? Thylakoid membranes is where you have the light, because you think of it as is like brighter part, is more to the front. The stroma is like the background material, so think of that as like in the dark. So that's the light independent reaction. And Calvin, the man have a kind of dark soul, the man thief somebody else. He a thief it, but he kind of prevents somebody else from getting noticed. So Calvin, he kind of like a dark soul. So that's the light independent reaction. He lose the light in his soul. So that's the light independent reaction. Remember that reaction don't need any light. So photosynthesis now. We could skip all this. This is C-sex stuff. Right. So one thing you have to know, and this is C-sex stuff too, but it does come. We have to know about the adaptations of a die-cut leaf. So you probably do this for a lab already, where you get a slide to draw back in Form 5 or maybe even this year. A die-cut leaf is basically what you see in here. This is the basic diagram that you see in here. Right, and just to go through the different parts here. Of course, what's gonna happen here is that sunlight is supposed to where the action happening is here. This is called the palisade mesophyll layer, right? They might be like, well, what I mean? Why are we calling it palisade? Where that word come from? Well, if you ever see a picket fence, like a bunch of planks forming a fence, an individual plank is actually called a palisade. So when so it basically looks like a bunch of picket, planks together, like a fence. So that's why I call it palisade mesophyll. Don't ask me why they didn't just call it fence mesophyll or plank mesophyll. Right, but they call it palisade mesophyll. It sounds a little cooler. Anyway, so this is where the party happening, and this is where you're gonna have all the majority of your chloroplast, not all, but the majority of your chloroplast is gonna be here. So of course, this is where you're gonna get the highest rate of photosynthesis taking place. This is the party, right here, right here. There's the party right here. Inside here, everybody want to be here. That's the place that's happening. So what exactly have to be there? Well, remember, we raw materials have to make it up there. First, and energy. So first we need sunlight to make it into here. We need water to make it into here. And we need carbon dioxide to make it into there. So those are the 3 simple components that we need, right? So we need energy from the top, that's from the sun. We need water, that's coming through transpiration and thing. And we get carbon dioxide diffusing through the stomata. We'll get more into the water and thing when we're doing transpiration. So what we have to understand now is how exactly is this leaf adapted to getting all these people to the party. So first of all, we talk about the upper epidermis. So the upper epidermis is transparent. So it's a transparent layer. And of course, you could think of it like a skylight or like a window on the top there where it's like glass. The sunlight could come in, you know what I mean? So it's like a nice sunroof. So here, so the upper epidermis is actually devoid of any chloroplast. There's mainly only cytoplasm here. It might have the odd chloroplast, but mainly it's transparent. You basically just have like your protoplasm and cytosol and things like that in there. And on top here is called the waxy cuticle. The waxy cuticle is what gives it this kind of shiny appearance, the leaf, but what it does is that it limits water loss through the top of the leaf. So if water trying to leave, it can't leave, them doors closed, right? So it can't leave through the waxy cuticle. It only have one way it can leave. You know, like when you go in a room, it have one door open on one side and one door closed, and there where you to sneak off, right? So they lock that door. So the only way you could really leave is to go back out where you came from. So easy stuff. So the watertight waxy cuticle limits water loss, right? The lower epidermis is right over here. This is the lower epidermis. And this is a thin layer of cells similar to the upper, but the exception now is that we have, and I'm going to take out some of this thing that up here so it don't look a little too cluttered, right? So it has these here, which are guard cells. Again, we'll get more into guard cells when we talk about transpiration. And but basically what these do is that they can get flaccid or they can get turgid. When they get turgid, they allow water to, they allow the entry or exit of water, or basically the exit of water, I should say, the net exit of water. But they could also allow carbon dioxide to come in. So where there's a high carbon dioxide concentration outside, so there's a high CO2 concentration outside, and there's a low CO2 concentration inside, of course, that's going to cause carbon dioxide to diffuse into the leaf. So far we understand how sunlight gets in, because again through this little skylight here, right? So this skylight here called the upper epidermis, the carbon dioxide making it through the stomata, and the water is coming through the vascular bundle here. The vascular bundle consists of mainly the xylem, the phloem, and other things like the cambium. So you should remember now that the xylem is made out of lignin, and it's kind of like a dead vessel, uninterrupted, which allows the flow of water. And the phloem allows the flow of sucrose through the use of pumps and ATP and the active transport and all that stuff. So basically around the vascular bundle we have the spongy mesophyll cell. And well, why they're called spongy is because they have a lot of air spaces, kind of like a sponge, and they're loosely, irregularly packed with a lot of air spaces here. And these facilitate gaseous exchange between the palisade mesophyll here and the stomata here. And I should also say the vascular bundle, because water could also flow up to there. Easy. So those are the adaptations. And if we continue here, you also have to know adaptations of just the palisade mesophyll. And so this is where we get more into a cape level of things rather than C-sec. So palisade mesophyll cells, so we're concentrating on where we see in just this green box here. Basically, there's a large number of chloroplasts for light absorption, right? Large number of chloroplasts, we know this. But palisade mesophyll cells, sorry, if you think about them, if you think about it, just a typical animal cell, not animal, plant cell, sorry, they usually have a large central vacuole, right? And for the palisade mesophyll, these are actually unusually large. They're much larger than from other plant cells. So what this does is that any chloroplasts, it kind of pushes them to the edges. So it prevents them from grouping up in the middle. So how is that helpful? Well, this is because a lot of the chloroplasts end up getting pushed to the edges, like to the top here. And that there helps to maximize light exposure. So if all the chloroplasts was like, you know, little cowards and there's kind of just in the middle here, you know what I mean? They wouldn't really catch this, because remember that the photons from that sunlight have to be actually captured by the antennae complexes of those of that chlorophyll within there. So they have to be on the outside, you know what I mean? They can't just stay inside an expected area, right? So these here, this polycy mesophyll is actually quite close, usually quite close to the vascular bundles. And of course, as I said, these supply water, which is a raw material for photosynthesis. Lastly, the chloroplasts have very thin cell walls for rapid diffusion of gases. Easy to go. Let me know if everybody okay. Tell me if I'm going too fast or if it's just so long. We have real time, but we still have 6 topics to cover, you know what I mean? So let me know if I'm going too fast, but as I say, it's day before exam, so we had to be going at a kind of pace. Well, let me know. What's the consensus day? So I think I'll slow down at this part because this is where everybody brain, they start to get, you know, you get a little brain fat, you get confuffled by this. If you're saying confuffled, especially all who are too good at English, this word, this quite scary word. Photophosphorylation. Photophosphorylation. So I say, break down the word. Photo means use of light. Like photosynthesis means making something using light. And phosphorylation is just a big nasty word that means adding a phosphate to a compound. To phosphorylate something means to add a phosphate. So again, if I have 2 phosphates, and this would be ADP, and I add a phosphate, for it to become ATP, I am phosphorylating that ADP. So that is why it's called phosphorylation. Remember that, right? So it's just the word scary, but the concept not scary. And the term photo just means that you are adding that phosphate with the help of light energy. Photophosphorylation. So you really only encounter this word when you're talking about photosynthesis. Keep chemistry, I wish. Chemistry is my Achilles heel. I could do almost anything else, but I can't do chem. I could do biochem, but I can't do chem. I shouldn't say I can't do it. I can't do it right now. I could sit down and learn it, and I could probably learn it in a month or so, the whole thing. But that's not fun, and I just do these things for fun. So, yeah, this is fun for me. Chem is not fun. I ain't good. Anyway. So as I said, the concept is simple. Photophosphorylation is a process. Lower the lo-fi, no problem. Okay, back to this now. So say if you had to define it, photophosphorylation is a process that uses light energy to attach a phosphate group to an organic compound. That's alright. The assistance is my pleasure. So ADP has 2 phosphates, right? ADP has 2 phosphates and ATP has 3. So phosphate is added to ADP using energy from sunlight. Of course, this process is not so simple because as you could see here, this looked like a whole little story happening here, right? That's a whole book that's probably happening there. So don't worry, we are getting into that just yet. I'm just gonna go into the basics of it first, and then I'll get into it. I'll get into it with a little more energy a little after. So you just have to bear with me. So photophosphorylation, right? We could think of it as being either cyclic, and this is usually what you'll find in bacteria, or it could be non-cyclic, and this would be in like cyanobacteria or green plants, and this is where you would have like more complex, you'd have more photosystems involved. So I use a word there, photosystems. So what exactly is a photosystem? So this type of photophosphorylation that you're seeing here, this is called non-cyclic that you're seeing here. And don't worry, don't expect it to to catch everything all at once. So we have two photosystems. We call it PS2 and PS1, not the gaming systems, but the photosystems. Oops, just now, Right, so photosystem 2 and photosystem 1. So how does this work here? And I say I'll go through this when I'm comparing them again. I'm just going through what the basic thing is be like. Now, we have photons, which are light particles. If you remember, Einstein's quantum theory says that light has a wave-particle duality, which means that it could behave as both a wave and a particle. So these photons are like light particles. You could think of them as little energetic particles, and they move from high energy to low energy. And these are caught by little antennae complexes, not depicted on this diagram, but you could think of them as like if you remember glycoproteins and glycolipids and things from Unit 1, and it would look like little strings of molecules attaching from them transmembrane proteins and peripheral proteins on the phospholipid bilayer. They kind of like that. So they like little antennae, like, for a TV, a long time TV really, that is pick up signals. So they have these little complexes there. And what happens here now is that these electrons will move through a chain of carriers. These carriers have different names. You don't need to know the different names. So like one is called cytochrome, for example. But basically these carriers are like proteins or complexes that are deposited along the thylakoid membrane. This here is basically the membrane here. So what happens now is that, I'm not going through it step by step just yet, but if we imagine we have an electron here, I say the best way to really think about it is a pinball machine, and this way pling, 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 pling gonna come here. So you're gonna go like this, you're gonna go pling, 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 right? And then as that's happening, you're gonna get a bunch of hydrogen ions eventually are gonna flow through this thing called ATP synthase. I'll go through that in more detail just now, just giving the basics. So again, what's happening here is that light of certain wavelengths, you don't have to memorize the wavelengths, but it's about 680, 700 nanometers or so. And I actually can't remember the number myself, but you don't have to remember that. But the difference between the two photosystems is the wavelength of light that they absorb. But the thing here is that this electron thing here goes pling, 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 pling. And eventually, what that does is that, imagine like it deposits energy as it is moving. Just like a ball hitting things and things lighting up in the pinball machine. And it making all kind of sound. The electron don't make no sound. They're making all kind of sounds. Everything lighting up bright and all kind of thing. And that's kind of like what's happening here. But just releasing energy and activating things. And it losing energy as it kind of goes along. But you'll notice that I kind of draw it like a Z. So you could think of it like gaining energy as it going up here. Losing energy as it coming down here. Gaining. Right? And then eventually this will do something with this thing called NADPH. That is a next thing by itself. We'll talk about that just now. But what happens here is that this will drive a bunch of hydrogen ions, and these would flow through these little transmembrane proteins here. So what you're basically going to get is a bunch of hydrogen ions PS2 is 680 and PS1 is 700, thank you, right? So that's it there. So it depends on the wavelength. So you're going to get a bunch of hydrogen ions, and think of these hydrogen ions as gas. But the vroom, 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 the vroom, vroom is this thing here, the car. When you start the car, I don't know, that wasn't a car. But the hydrogen ions is the fuel that is basically going to forward through this little engine here. And this engine is called ATP synthase. Right, so this is called ATP synthase, and this is like an engine that's gonna start to produce energy. Basically, what this is going to do, and if you think about the word ATP synthase, it synthesizes ATP. So let me come back to the word here, photophosphorylation. How are you going to synthesize ATP? Well, all you have to do is have a ADP, and you have to add a phosphate to that ADP, and it becomes ATP. And that is with the help of light, because the light is actually what stimulates these electrons to move. So I know, again, that sounds like a whole thing, but I'm going to go through it slowly when I go through cyclic versus non-cyclic. But this is just a overview of what happens. You don't have to get everything right away if you get frightened. Now, just keep in mind, we have photosystems involved. In the case of non-cyclic, we have two of them, PS2 and PS1. I actually don't know if you have to remember, if you have to draw it. Yeah, I would say try to draw it, yes. But it not hard. Draw the thing on a piece of paper. Draw it like 2 times. It should be alright. But yeah, the basic thing is that it has photosystems. These photosystems and carriers allow the passage of electrons. These electrons allow energy to flow and deposit and so on. And these trigger off a series of reactions that would eventually form ATP. Now the light here is splitting this water molecule down here. Right? Just remember that. I'm going to get into that just now. Remember photolysis back in Form 5. The light, or Form 4, whatever. The light is splitting this water molecule, and that's actually what giving the electron. But I'm going to come back to that just now. But first, I say, let me start with just what has happened. And let me write something here just now. Let me start with what has happened here, right? So I got this. Right. The light. Make this a little bigger. The light come in the thing, and it break up the water, and some of the water, and part of the water is electrons now, right? So them electrons, let's go pling pling on the photosystems, and eventually ADP is phosphorylated. That's basically what happening there. That's the basics of it. So the light come in the thing, it pass through the upper epidermis, come into the palisade mesophils, gets absorbed by the chlorophyll, by the antennae complexes, gets absorbed by photosystem 2. And photosystem 1, we start with photosystem 2. And what that does is that it breaks apart water. Remember that water come from the roots and move up the xylem and come out the vascular bundle, all kind of thing, right? They break apart that water. Think about what water is. Water is hydrogen, oxygen, and then you have some electrons. The electron, there's a pling, 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 pling. Yeah, man. This is the Caribbean Examination Council, CAPE, is the Caribbean Advanced Proficiency Exam. The light coming thing, it break up the water. Part of that water is electrons. Yeah. So them electrons go pling, 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 pling along these photosystems and carriers there. And then eventually what's gonna happen is that ADP will get phosphorylated into ATP. Of course, a whole set of other things has happened. We will get into that just now. Take all your time. Get into that just now. But the next thing you have to remember here is that this does occur in the thylakoid membranes. And if you remember here, this is the light. This requires light. So this is the light dependent reaction. Okay. Now, but this does not occur, and this is basically in green plants. Now, you want to see that I had something called NADP, right? NADP stands for nicotinamide, adenine dinucleotide, phosphate. And NAD is just the same thing without phosphate. So, what exactly is that? And I say you could be reaching here today on the 13th of June, and the exam is 14th of June, 9 a.m., and you still ain't know what that is. So these are reducing agents, right? And I said the main one we're going to concentrate on here is NADP, because this is the one involved with photosynthesis. NAD is more when you're talking about glycolysis and Krebs cycle and thing. So these are reducing agents, right? So these are, you could think of them as coenzymes. They just help out enzymes. It have a type of enzyme called a dehydrogenase, right? And let me just think about that word. Let me just think about that word. A type of enzyme called a dehydrogenase, right? And this is basically a hydrogen removing enzyme. That's what dehydrogenase is. Hydrogen removing enzyme. And we need to take into account all that. But basically, you have a bunch of these that's gonna take out hydrogen, right? So you're gonna remove hydrogen from some compounds. And what's gonna happen here is that when they take out that hydrogen going like yay, yay. However you say it, all the young people. So anyway, so when you take out that hydrogen, something had to catch it. You can't just take the hydrogen and pelt it plow by somebody's house. Nah. You're pelting it like a cricket ball, and that hydrogen there, somebody had to catch that. Somebody have to catch that hydrogen. So what happens here is that NADP, we're looking more on this side, by the way, right? Oh shucks. We're looking more on this side. NADP, when it catch 2 electrons and a hydrogen, it becomes NADPH, or what we just call it reduced NADP. That is all it is. It's a thing that is catch the hydrogen. It's a thing that is catch the hydrogen and the electrons. It's a thing that is catch it, because when a dehydrogenase take out that hydrogen, it can't hold it in the hand. It can't pelt it, plow! It can't pelt it by somebody. Might break somebody's window. So instead, what it is do is that it be NADP just be like, hey, pelt that by me now, boy. Hey, pelt it by me. So then NADP does catch it, and when NADP catch the thing, right? When NADP catch the hydrogen, well, and the electrons associated, it becomes NADPH. You know, like a Dragon Ball Z when they're fusing. Well, imagine NADP is like one super Saiyan, and H is a next Saiyan, and they fuse, and they form NADPH. That's all it is. The same thing is NADPH. Next way you could say is reduce NADP. It means the same thing, right? So don't get chained up. If you see reduce NAD, it's just the same as NADPH, right? So remember, you have oxidize and reduce, right? I going through no chem with all you. I hate chemistry. I love biochem, but chemistry, all you know chemistry, man. Yeah, man. Anyway, so you could think of them as catcher's mitts for hydrogen ions and electrons, typically 2 electrons. So whenever either NAD or NADP, they catch a H ion, a hydrogen, and 2 electrons, they turn into their reduced form, which is NADH and NADPH. So NADH and NADPH release the ion, hydrogen, and 2 electrons, and they could convert back to it. But an easier way to think of it is like this. Imagine we have a ball. There's a ball, right? I know there's baseball in the Caribbean. We don't play that. But it was kind of hard to find stock photos, like cool stock photos of cricket, Brian Lara and thing now. So I use, and we don't have the catcher's mitts for cricket. So think of NADPH, right? This is like they're holding this hydrogen ball and these 2 electrons, and it grasping it. It holding it. It had the power in the hand, right? You're holding it. So when it pelt out this ball, right? So this is here where say it get reduced. Now, a next way you could think about it is like a charged battery. So NADPH, you see there, that's now NADPH, oh shucks. NADPH is like, I'll find a place where I could type this good. NADPH is like a charged battery that can release hydrogen and electrons and become NADP. NADP is like the uncharged battery, right? So NADPH is the thing that have the hydrogen and the electrons in it, and now it catch it and now it could pelt it back out. So it's like a charged battery. Think of it like that. That's the easiest way I could really say it for you without getting into the case. As say if you know what it is, you're kind of halfway going through, but that's what we're here to do, right? We're trying to not be going through in this life. So NADPH is like a charged battery, and it can release this hydrogen and associated electrons and become NADP. So think of it like a catcher's mitt, and then it could pelt out that hydrogen when it's ready. Now remember that hydrogen and electrons is energy. So it's like a charged battery, and then it could, it could release all those electrons onto something else, and it could release energy basically. That's what NADP and what NAD is. They're very similar, but except they involve in different processes. You more see NAD in glycolysis and Krebs cycle and things, for example, but you see NADP more with photosynthesis. Now, and as if you're mixing up the two, just think of the P, NADP, photosynthesis. The P don't actually stand for, the P stand for phosphate, but think of the P for photosynthesis. Anyhow, so we have two types of photophosphorylation. We have cyclic and non-cyclic, right? So we have cyclic, and the next one is non-cyclic. And then I'll put a table just to compare the two. So this is where we're going to get into more of the little nitty-gritty of what's happening in these processes here. Okay, so cyclic photophosphorylation. So I'll say this is... Oh, it's sort of a C-Sec physics. Forget everything. That's why you shouldn't cram. That's why you shouldn't come spooing. You shouldn't have to come spooing. You really should come spooing for a little brush up on the knowledge. I don't know if I equipped to make you learn everything in a few hours. Anyway, synthesis of ATP during the light dependent stage. So this occurs in bacteria, as I said. So not cyanobacteria, but just your regular prokaryotic bacteria that don't have chloroplasts and things like that. They don't have any major autotrophic capabilities. They could still make their own food, but it's not all that they depend upon. So there's only, first of all, you're going to see here that this looks a lot simpler. The schematic for it looks a lot simpler than the one we just looked at. So this is because there's only one photosystem in here. There's not two, there's just one. So there's photosystem 1 here. Now remember, in the other one that we looked at, it had two, photosystem 1 and 2. So this involves a cyclic movement. And if you think about what's a cycle, like water cycle, carbon cycle, nitrogen cycle, is something that you have a bunch of materials, and those materials could be reused. They could be recycled in a way, right? And maybe not forever, but at least for a while. So cyclic means this, that these electrons, if we start from the chlorophyll, if we start from photosystem 1 would be somewhere around here, right? Photosystem 1, we have electrons that will be stimulated just due to being caught by the antennae complexes and things like that. So these excited electrons are gonna be going like up here, pass through this electron transport chain. Remember, this is just a bunch of protein complexes and things. I like to think of it as like a little bouncy conveyor belt for electron, where it go like, and as it goes, it loses a little bit of energy. Imagine like you're bouncing a ball. It go bounce, 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 and it come back here, right? So, but cyclic because it goes back to the photosystem that it originated from. It doesn't move on to our next photosystem, so to speak. Now, as this happens here, these excited electrons are going to drive the formation of ATP. And if you remember what drives the formation of ATP, it is this sweet little enzyme here called ATP synthase. And these are found on the membranes, like on the thylakoid membranes or whatever membranes that are within the bacteria, right? Those like inner prokaryotic membranes. So that is called ATP synthase. And that there synthesizes ATP. It takes the ADP, adds a phosphate group to it, and becomes ATP. And that is cyclic. Now, there's of course more processes that happen here, but we're going to get into that now with non-cyclic. But first, the marks all here say, all they probably want to relax something after this paper too, especially how is the last paper too, you know what I mean? So, you probably lock away your phone in your drawer, but as soon as you get back your phone, all you know what you have to do. Raid Shadow Legends, one of the biggest mobile role-playing games of 2022, is totally free. Almost 20 million users, 10 million, enjoying Raid over the last 6 months, and it's one of the most detailed models and environments. Silky smooth 60 FPS animations, all the champions, they change the strategy and your buff and your debuff and your abilities, dungeon bosses, all them nice things, man. When all they done in exam, all they go play some Raid Shadow Legends. God, oh yeah, back to the live stream. There we go. So say we now describe non-cyclic photophosphorylation here. And I lose myself, yes. Sorry, we now describe cyclic. We want to go back to we just described cyclic, and I say it's like, imagine like it's an electron ball bouncing, bounce, 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 and it comes back to the original photosystem. As that happens, ATP synthase is kind of activated by all these hydrogens and electrons and things like that. All this energy, it drives the formation of ATP from ADP. Okay. So now we're gonna get a little more into non-cyclic photophosphorylation, right? And give me 1 second here. A visual I wanna find. All right. Let's know, eh? Not eh? I just wanted to find this thing. All right. Okay. So if I watch this here, okay, so here we are the chloroplast, right? More work. More work, less talk. More talk, less work. So all you can see it our photosystem 2, right, which absorbs 680 nanometers, and you can see there that we have the electron passing along the carriers there. And this here, this is like a transmembrane protein. It allows hydrogen ions to pass. You can think of it as it creates something called a proton gradient. We'll get into that just now. So let me look at what happens here. So let me look at that again. We have a water molecule, right? H2O. And we have this thing, light bunks up this thing. And what happened here? We get 2 hydrogen ions, oxygen molecule, right? So that's half O2. And we have electron, right? And typically here, you'll have more than one water molecule, so you'll have more than one electron here, of course, right? So let's just imagine one electron. Let's just imagine the passage for one electron. This is gonna pass through. Remember, this here is the first. This is photosystem 2, and right. Remember, this is photosystem 2 here, and this is 1. This is photosystem 1, so it goes from 2 to 1. Forget all these other things here. These are just like complexes that help the electron to flow. This forms part of the electron transport chain. So ferredoxin and all kind of things. Forget all of that. You need to know it at this level. Maybe if you become big plant scientists like Melvin Calvin and them, yeah, then you have to know all that. Okay. So, let me look at what's happening here. What we have here, at the end here, we have NADP. Right, so we have NADP, and then when this accepts a hydrogen and electrons, it becomes NADPH, because remember what's happening here is that the dehydrogenase is throwing out some hydrogens and electrons, and NADP is like a catcher's mitt. They're going to catch it. So this is where this NADP, which is oxidized by the way, is going to get reduced, and it becomes NADPH. Now, as this happens, now we get a buildup of hydrogen ions. Now, I think the video that you're watching here, it lacking in a little ways because it's a little out of order, but this is just for you to get the visual. I will go through it with my diagram after. But look at what's happening here now. What's going to happen here now is that we're going to get a buildup of hydrogens. These are hydrogen ions. And remember what these are going to do. These are like gas. And they're going to power the ATP synthase. And that's going to help to phosphorylate ADP and turn it into ATP. That's basically to make the energy. Right? So look at it again. These hydrogens that develop in here, remember these will come from water molecules and things. These will come from pumps. Yes, the cytochrome complex also acts as a transmembrane protein to draw some hydrogen ions as well. I don't think that's depicted in this here, but that's what happens as well. That's correct. Let's say the point here is that, okay, so I'm going to explain the proton gradient now. Let me come back here. So, coming back here now, we start taking it from the top with non-cyclic. Non-cyclic photophosphorylation occurs in green plants. Where's my pen there? Right, and I say, well, why green plants is because they have these two photosystems, photosystem 2 and photosystem 1. Remember, cyclic only had one. We have NADP being involved here, which then reduces to form NADPH. And we also have photolysis involved here. Sorry about that. We have photolysis. So the excited electron moves from PS2 to PS1, photosystem 2 to photosystem 1. That's why you're seeing pling, 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 pling over here. Now, the light that comes in, that is going to split water molecules, which is called photolysis. So remember that. So this H2O that we have here, this is going to split into hydrogen ions, right? So we're going to get hydrogen ions, we're going to get electrons, and then we're going to get oxygen molecule, right? Now, this oxygen here, this actually don't really matter in the process. This just gets released out of the stomata. You know, the plant is taking carbon dioxide and release oxygen during photosynthesis. That's this here. So this don't really account for any of the other parts that we're going to talk about. The things that we have to take note of is these hydrogens and these electrons. This here is going to create what you call a proton gradient. So let me go into what was happening here. Probably 10 p.m. with breaks. When I finish photosynthesis, we'll take a break. Electrons are accepted by NADP and turn it into NADPH. So remember, this electron here is coming from this water molecule. When the water molecule splits, I said it splits into hydrogen ions, electrons, and an oxygen molecule, and the oxygen molecule, that'll leave. That'll go on, may I want to be here again, I busting it all here. I go on, I is oxygen, I good. I going back out into the atmosphere. So, the electrons now come from these water molecules. This is what you need the water molecules for. And these electrons are released from the photolysis of these water molecules. And they go pling, 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 pling along the photosystems. And they provide energy to these complexes called, say one called cytochrome, for example. But you could think of them as proteins, and some of them act as transmembrane proteins. If you remember integral proteins and protein carriers and channels and things from our membranes in Unit 1. Well, this is basically what's happening here. And this energy is kind of like a voltage gated channel. And well, the IT people, get the hell out of here, IT people. We're doing biology, we're doing serious thing today. Biology keep Unit 2. I don't even know how to use a computer. I go do IT. Get out of here. Right, so what's going to happen here, right, is that these electrons are going to stimulate these transmembrane proteins here. And what they're going to do is that they're going to take, you're going to have hydrogen ions outside here, by the way. Right, so you're going to have some hydrogen ions outside here. And these come from other molecules, right? Don't study too much where they come from, but they're there. But they can't really pass through this bilayer here. They have to pass through this transmembrane protein. And this transmembrane protein is only activated when the electron passes into it. You know, it only wake up and it say, hey, time to do some work. When this electron passes, the electron say, hey boy, hey, cytochrome, wake up. Hey, protein, wake up. Time to open up. Now, these hydrogen ions now, they kind of accumulate here. And from the water molecules, remember, hydrogen is down here too. So hydrogen accumulates down here as well. Right? So this is going to create, well, you don't really say concentration when you're talking about these ions. But you could say that they have, you know, they have a higher positive charge up here and a lower positive charge down there. So through the process of something called, it's kind of like chemiosmosis. What happens there is that these hydrogen ions, they go plops, 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 plop, plop. And that's basically what a proton gradient is. And so let me clear up some of this thing that I have there. But basically, it opens up this channel to allow a whole set of hydrogen to come through here. So what you're going to get is a kind of proton gradient or hydrogen gradient that's going to allow a whole set of hydrogens to come here. Now remember, say what the hydrogen ions are like. There. L. Right. What this is like. There's like gasoline, but now it's expensive. And when they flow through this ATP synthase engine, right? So when they flow through ATP synthase, remember what's gonna happen now is that's gonna provide the energy. That and NADPH. Only remember NADPH is like a charged battery as well. So NADPH can also, you only remember all this NADPH and thing, all this could release hydrogen. So this is why we get so much hydrogen and thing up here. When this electron go pling, 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 pling here, and it NADP turns into NADPH. All you remember was say something had to catch that H. Right? So the NADPH holds on to it and then it could pelt it back out. So this pelt it back out and that's where it comes down here. That's basically what the proton gradient is. It's a whole set of sharpness. There's a whole set of sharpness. But the thing is here, we end up with a bunch of hydrogen ions down here and that's like gasoline. It's not as expensive as like gasoline. And then it goes through the vroom vroom ATP synthase, the vroom vroom engine, and that there photophosphorylates, so phosphorylates ADP to form ATP. Now what you'll notice here, none of these electrons here, where are they? None of these electrons here actually returning to photosystem 2. Those electrons not returning. Instead, those there coming to help with those electrons basically helping to form NADPH. They're not returning. So this is why it's called non-cyclic. So that means that some plants can actually switch from non-cyclic to cyclic if it have like high temperatures or a lack of sunlight. Some of them could, if they are to photosynthesize, like they could actually help this electron get back to photosystem 2. That's a whole other story. But it don't return. So that's why it's called non-cyclic. So once more from the top, light comes in, basic, light comes in, splits this water molecule. It forms hydrogen ions down here. It forms electrons. And it forms an oxygen molecule. Oxygen molecule, he busts it, he gone. Now, this electron goes pling, 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 pling to photosystem 1. Activates these proteins here, and this now is like a gate open, right? It's like a gate. This gate opens, the electron ends up in an NADP molecule, right? And becomes NADPH. It gets reduced. Remember, this is the electron and some hydrogens there that would do this, right? So some hydrogen ions would be here already, and that would be caught by NADPH. Now, what will happen now is that NADPH would oxidize and release a bunch of these hydrogen ions. Remember, this happens every time electrons bounce across and go pling, 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 pling. Now, because now you have a higher buildup of hydrogen up here, right? This is going to initiate a kind of chemiosmosis process, which is going to allow the hydrogen ions to flow down these transmembrane proteins. And then remember, we have hydrogens from the water too. We're going to get a whole set down there, and that's gas. Not gas when your belly hot, eh? Gas when you're powering something. What are we powering? ATP synthase. Why? To turn ADP into ATP. ATP synthesize A's. We get A's all there, A's and keep. That's it. It looks scary. I now lean back here and I'm watching it looking like a real mess. But that's what it is. Well, I gotta tell all you. Watch back the video. Right. So basically, we have thing that is catch hydrogen. If I was to write it like this, I write it in Caribbean talk folio. And then we are getting to the differences. We have thing that does catch hydrogen and electrons. And we have thing that is move them hydrogen ions down to the thylakoid, if you understand what I mean. And when plenty H plus ions got up, it is form a proton gradient. Right? So you have H plus, H plus, H plus, H plus up here. And then what is happening here? When all them H plus flow down the protein, the protein carrier thing, we get H plus gather up, which could then operate the ATP synthase now. And we go get some ADP, some nice ATP from some steel ADP. That is the thing. That is the thing. Non-cyclic. So let me get into the differences. Now, the reason why I stay on so much time with this, all this I come in years. Cyclic versus non-cyclic I come in years. And this might be the year. It might be all a year. All a special. All again this I come in like 8 years. So all is special, or they might get this. We hope not, but all they might get. COVID churn. Not churn who are COVID. Churn who survived COVID. Churn what? Anyway, so there's the differences here. So we are cyclic. Right? So we are cyclic and non-cyclic. And this is basically what a list of things. You should be able to list at least 4 or 5 differences, right? So where does it occur? Cyclic occurs in photosynthetic bacteria, non-cyclic in green plants, which of course are eukaryotic, more complex. Is ATP produced? Yes. Yes. That's the whole point of photosynthesis. Well, not the whole point. The whole point is to produce sugars, but ATP also to be produced. Now, are photosystems involved? Yes, for both, but cyclic is PS1 only. I say because they're prokaryotic, they're not as complex. For green plants, they involve the two, PS1 and PS2. Remember, it starts with PS2. The electron travels from PS2 to PS1. I actually don't know what's the reasoning for that. I think they name it after maybe the discovery, or I don't know if anybody know. Is reduced NADP. This is NADPH, right? Is reduced NADP made? And remember, this is when it catch the hydrogen and the electrons. Is this made? For cyclic, no, we don't have any coenzymes involved there, anything like that. But for cyclic, yes. No. Where's the PS1 replacement electrons from? So where in photosystem, remember, cyclic only has one photosystem. So where does the electron get replaced? Well, basically, remember, it's cyclic, so it just reuses the same electron. So from the same electron that was first emitted. Okay, that's sure they discover because they investigate. Okay. Well, I expect it was order discovery. So, non-cyclic, where does the PS1 replacement electron come from? Well, this comes from the one emitted in PS2. So remember, the one in PS2 moves to PS1. So when the one in PS1 is used up, the one in PS2 then takes over. So, as I say with this, when this electron in PS1 is used up, this one in PS, remember this is like a stream. This is not just one, it is a stream of electrons. So when this one busts out, when that one use up, this one here go come and take over. I'd be like, shift done. I'm taking over for you. So the PS2 electron replaces the PS1 when this done. Where do the PS2 electrons come from? Well, with cyclic, that's easy. It don't have a photosystem too. But with non-cyclic, where the electrons are found is when you have photolysis of water. Well, you remember, say when light is split apart, you get hydrogen, ions, oxygen, and electrons. And the electrons come from the water. Cyclic predominates in anaerobic conditions. This is why you don't usually have water involved. You don't have any oxygen being evolved and things like that. So there's no evolution of oxygen here. But you have evolution of oxygen in non-cyclic. When always learn about photosynthesis and Form 5, you always learn about non-cyclic the whole time, basically. It's just now it get like real complex and, all kind of thing all over the place. But yeah. So non-cyclic in green plants, cyclic in bacteria. Okay. So let me take a 5. We'll take a 5 and then I'll go on to limiting factors, right?
Hello, good day, sir. We are doing a survey. We want to know why you just come on YouTube and watch Spurwin.
Because after I
What's Spurwin live stream?
and I kiss my babes, it sends shivers up her spine.
Ooh, it can't get better than that.
Because it freshens your breath.
Brother, tell me why you just watch Spurwin.
Because no matter where I go, no matter what I do, I must always do my best.
Because it's refreshing.
For me, it's the only thing that can relieve my upset stomach.
I'm a smoker.
That Spurwin live stream.
It does help freshen my breath and take away all the smokiness.
It is very good. It is very useful.
It keeps my breath fresh. Full stop.
Need to keep it fresh all the time. So you're from Tobago?
Yes, I'm from Tobago. Yes, I love Spurwin. Original.
Spurwin.
God, you're real spit in there. All right, Sam.
Jesus, exam thing.
Hey,
Who's that, me?
yes you. Fix that face now. What happened?
Another red man. Not the red man I'm looking for.
The rain is way harder and the wind is...
We wish all our candidates the very best in making the final preparation for your
This is what you're reading?
Jesus, exam thing.
Just right.
In Trinidad, this going on?
That is real ginger beer.
You want to tell the secret to being successful at CSAC?
Yeah, tell me now.
Balancing at sideways.
Really? That is your big secret?
Yes. This man real good.
Real ginger beer.
I went from alright to real good.
The letter of the day is J.
J is for
Jesus, exam thing.
Plenty people dying, but we can't do nothing about that. If it's dead, they go dead.
correct. 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 B is for
And the answer is
Alright, I ain't that. Wait, you wanna help me study later?
study. Study the Guardian. See, I practice that.
Jesus, exam.
You will do well.
And you're cook yet? Aloma, what you gonna do? Don't worry, I organize.
I'm hungry like a dog, girl. I'm going to be just put food on that table for me.
You mean please?
Yeah, like your flavor with the pot. Holy girl, this is a meal fit for king.
Yes, only the best corned beef for my king.
Corned beef, what corned beef is this, girl?
Rainbow corned beef, darling. Rainbow corned beef.
Distributed by ADM.
Let me tell you something about CXC. Nah, not this cinema CXC. The Caribbean Examination Council. CXC is amazing. Oh my God, you don't need nothing else when you have CXC. First, you have your paper ones. Amazing. Multiple choice exams. You shade little dark circles and come closer. Come closer. Let me tell you a secret. When in doubt, choose C. Better yet, all you have to do is a bunch of paper ones from before, and you're guaranteed to have done at least 50 out of 60 questions by the time you write the exam. Thank God for Google Drive. Incredible. And you get questions like somebody get paid to draw this, yes? But then you have your paper twos. They call these the structured questions. You just answer the questions. Simple as that. Explain to Sunil the advantage of his testes being carried outside body. What, you stupid? Just answer the question, dummy. And then you have your paper trees, labs, SBAs. See, the trick is to not do your labs and SBAs, and then at the last minute let pressure build up and then steal somebody else's book and copy all of their labs. You're guaranteed to do real good. CXC is gangster. See, what you do is you pay them to write exams that you don't want to do, while they just do whatever they want. It have hurricane, exam, volcano, exam, pandemic, exam. Even when things go wrong and a bunch of people receive the wrong grades, like in 2020, they denied for years because they can never be anomalies. Oh my God, what a beast! CXC is the best. 10 out of 10. Would recommend for everybody.
Alright, Olia. If we be back. Feel as a little more than 5 minutes. Time ticking, time ticking. Really. Right. So it has one more part of photosynthesis here that does actually come all the time. I feel like coming for all the year, but who knows? And that is, oh shucks, I forget about this man Calvin. I thought the next part was limiting factors. There we go. Alright. So let me start with the Calvin cycle. Hey. So I figured we just do this light dependent part, right? So that now we have the light independent part. Actually, this you know the strange thing about this? This is probably like one of the most complex parts of this topic, and it has never come. So who knows? Who knows? So anyway, so Calvin cycle, or if you want to be a little more appropriate, the Calvin-Benson cycle, because Calvin kind of fired the man and take the Nobel Prize for himself. That whole thing does work, and you just do all the work, Next man come and bounce it on and copy it on and take it and he get the prize for it. You know, copying lab and all kind of thing. So anyway, so I would call it the Calvin cycle. So the Calvin cycle, now remember what is the main reason for photosynthesis? What we synthesizing? Well, so far all we really talk about was ATP. But the ATP that we really needed it for this cycle here to occur. Now, this here, this ATP here, this is really what we need this for. We need this ATP to power the cycle in a way. Not really to initiate it, but to kind of get moving. So we actually have 3 parts involved with this. And remember, the whole point of this cycle here is to make sugar, to make glucose. And if you remember, glucose is, the molecule is C6H12O6, which means there are 6 carbons, 12 hydrogens, and 6 oxygens, right? What we're really focusing on here is that it has 6 carbons. Okay, so therefore it is called a hexose sugar, which means it's a 6-carbon sugar. Now, the whole process here is catalyzed by an enzyme called rubisco. And rubisco is the most prolific enzyme on Earth, is the most prolific enzyme in the world, because every green plant, and there are plenty plants in the world, every leaf, every little chloroplast have to have rubisco in it. And rubisco stands for ribulose biphosphate carboxylase, right? But we could just call it rubisco. You know, it sounds nicer. And what rubisco does, it catalyzes the joining of carbon dioxide, which is seen here, to a 5-carbon compound called ribulose biphosphate. This is a pentose sugar, and we could just call it RUBP. And it don't sound as nice as rubisco, but you name your child rubisco now, right? So anyway, RUBP is a 5-carbon sugar. So coming back to primary school mathematics here earlier, if we have 5, and that's from RUBP, 5 carbon, and carbon dioxide have 1 carbon, 5 plus 1 is equal to 6. 6 carbons. So we have 6 carbons here. So together they will form a 6-carbon sugar, but this is not yet glucose. This is simply what you call an intermediate, right? So this is an intermediate. It have a name for it. Nobody care about the name because the thing does just break apart almost immediately. But it's a 6-carbon compound. And you'll notice here that there are 2 phosphate groups attached to either end of it, right? So I'm going to repeat what I was saying. If we start with this RUBP compound, yeah, rubisco is really sound like a biscuit. For real. So if we start with this RUBP here, right? And we add this carbon from this carbon dioxide here, rubisco allows that carbon to bind to this 5-carbon to produce a 6-carbon, right? That's what you're getting here. So this is the process of carbon fixation, and that is the first part of it. It's a three-part cycle, right? The first part is called carbon fixation. The second part is called reduction, and the third part is called regeneration. Regeneration allows the cycle to keep continuing because it's a cycle now. It's kind of like the water cycle. If water evaporates, rain had to fall for the water to evaporate again. For rain, it had real rain today. Well, in Trinidad at least. I don't know if we have a regional audience here. So that is the first part where we take in 5 plus 1 and equal to 6. Now, this 6-carbon sugar breaks down into 2-carbon compounds. So this is the 6-carbon intermediate. Or let us study view count now. This thing here for the ages. This thing here for whoever in the future. Some poor soul in the future who's struggling in biology. Bio is real thing, you know. I can feel bio, you know. Look up Bio Crash Course Mad Marathon or Bio Marathon. This is Spurwin. 10 years in the future, Spurwin, thank you. I don't know. I don't really care, honestly. If it's like 5 people, I still doing it. It's whoever want to learn. So the intermediate is a 6-carbon compound, right? Now this is unstable. So this 6-carbon compound, this sugar breaks down into 2 3-carbon compounds. So we have here, we have a 6-carbon compound, 1, 2, 3, 4, 5, 6. And eventually now this will break down into 1, 2, 3, 1, 2, 3. And this would have a phosphate attached to the end. This would have a phosphate attached to the end. And of course, these also have phosphates here when they break up. So when they're breaking it up here, this forms something called phosphoglyceric acid. Now you can actually get away with just calling this thing PGA, which is the name of a golf tournament, I think. But anyway, you could get away with calling it PGA. And that is a phosphoglycerate or phosphoglyceric acid. So this happens when this 6-carbon intermediate splits down there and it breaks down into these 2 3-carbon things here. Now this is not this is of course not glucose yet, because this is only 3 carbons. Glucose is a 6. So right now we have these PGAs. So what's gonna happen here now? The ATP, right? Now we have what we call the reduction phase. Remember we had it breaking because it's unstable. So it's not a stable molecule. It's kind of like when you have, it's not the same, but you know, in a radioactive compound, and you know, it just break down into our next one because it's unstable. Kind of the same with this molecule. It's not stable. You have plenty of molecules like that. They just exist to break apart into other molecules. So you just call it an intermediate. So anyway, so now we have this process of reduction. So what's happening now is that this ATP, remember all this ATP that we make in the non-cyclic photophosphorylation. Well, now we need to use the phosphates from that ATP. So now we need to turn that ATP. We need to use the energy from that to power through the cycle. So the ATP is going to convert to ADP. And remember all that NADPH that it build up to? When the electron and the hydrogens latch on to the NADP, and the NADP kind of grab on the H atoms. Unstable like me too, unstable like Spoo. Anyway, so all this NADPH, all of that, gonna only remember what I say. This is like a charged battery that holding a hydrogen. So they're gonna pelt out that hydrogen. Right? So it basically gonna oxidize. Right? What this is gonna happen now is the hydrogen gonna catch on to these so let me draw a thing here. Show you. So this NADPH, right? So this is gonna pelt out with hydrogen, boy, pelt out some electrons too, right? We're gonna pelt out. And so this getting oxidized, of course, when it losing those things. So the phosphoglyceric acid that we had, the PGA, that there is gonna add the hydrogen ions, right? So you're gonna get the hydrogen ions, you're gonna get the electrons and things like that. So those things gonna latch on to the end. So this gets reduced, right? So this gets reduced. And so that's why I call it the reduction phase, because that now is accepting the hydrogen and the electrons and things like that. So that gets reduced and it turns into something called triose phosphate. Triose phosphate, or G3P, which stands for glyceraldehyde 3-phosphate. This is the boy that we're looking for, because this G3P here, this is what's being used to make glucose. So that's that there that goes on to make glucose. So that there, you could think of it like as part of a glucose molecule, and then they could combine and form glucose eventually. It's not as simple as I'm making it sound, but that's really what happening there. So let me explain now what we're talking about so far. Right? So in Caribbean speak, this is what's going on here. The 6-carbon intermediate kinda shaky now, so not shiny, it's kinda shaky. So it is break up into 2 3-carbon compounds. Right? And this called PGA. Right? So we have 2 PGAs here. And them PGA them just catch some hydrogen ions and some electrons let out from ATP and NADPH. It is catch that. So of course, when it catch them hydrogen ions and thing, it is get reduced. That reduced form there, we just call that G3P, also known as glyceraldehyde 3-phosphate, but we could just call it a triose phosphate. Yeah, when it catch them thing. So we call it a triose phosphate. This stays sweet and eventually form glucose. Well, that part I come through there, but that just stays sweet and eventually form glucose. So these things here, oops, this triose phosphate, that there becomes glucose. But you have to remember now that this is a cycle, and the cycle actually have to keep cycling now. So what had to happen here is that some of these G3Ps are recycled. Okay, so what? PGA and G3P is not the same thing. G3P does form after the thing get reduced. But so you could say triose phosphate. If it takes in there, you can look up, it have a TED Ed video that's actually sum it up really nice. I think the name of it is Nature's Small Factories or something like that. It have a video that sum it up really nicely, and you could probably look up that TED Ed video, and they have a nice animation with some kind of Dr. Seuss looking things. But yeah, textbook, skip a step there. But anyway, it actually have a lot more to it than this, because it is actually perform a little simultaneous equations and a lot arithmetic. The system. Hey, sometimes I just wonder if all of this was by chance, and it is seem weird, because you know in science it's kind of taught that everything not really designed, everything come from evolution, but I just have a doubt sometimes when they go deeper and deeper and deeper into these things, it does seem like a little too much to be random, a little too much. But my brain's small, so I probably missing a few things. Anyhow, so not all of these G3Ps are used, right? Actually, what happens is that this cycle has to cycle about 6 times. 1, 2, 3, 4, 5, 6. You don't need to learn that, by the way. But it actually has to cycle 6 times to form molecules of glucose and to sort of reform an RUBP molecule. So this is where we'll have regeneration. So regeneration now is where we're gonna have the excess G3P molecules. So at this level, we don't need to know about how much they split apart to form 5 and how much is split apart to form 6, because it is split to form 3, which is then to form 7, and then it is form 10, and then it is split into 5. All kind of stripping is this happening. All you need to know that at this level. All you just need to know that the excess G3P molecules are recycled and they will be reused to regenerate RUBP. So of course we can't use, it's kind of like this. You imagine you're working somewhere, right? And they're giving you $10,000, right? And they give you $10,000. What are you going to do? You're going to spend that $10,000, the whole thing on sugar. You're going to spend that whole thing on KFC. You're going to spend that whole thing on food and rum. Nah. You need to spend some of it, of course. You need some of it to make food, but you also need some of it to kind of carry about the rest of your day. You need like it's not just about buying all these sweetie and all these bobby and things, spending whole $10,000 on sugar and bobby and thing. Nah, you need to conserve some of it. You need to save some of it. The excess. I say 10,000 because 10,000 is still a little bit, but that's still a little bit. So some people pay $3,000 and to do more work, but but you need to use the excess to carry on the cycle. You need to get through the month. So the excess triose phosphates, the excess G3P molecules, those are recycled and they will be used to regenerate RUBP. And that, of course, that uses ATP as well. So we need an ATP molecule to do this as well. So of course, this is what, remember where this ATP come from? This come from non-cyclic photophosphorylation, right? So basically, in a nutshell, Rubisco affixes CO2, right, to RUBP to form a 6-carbon intermediate. This six carbon intermediate breaks down into 2 phosphoglyceric acids, 2 PGAs, right? That's he and he, or she and she, whichever one. So then what happens here now is that PGA gets reduced because the phosphates, the hydrogens from the ATP and from the NADPH, remember the NADPH pelting out them hydrogen and electrons, and it's going to reduce PGA to form G3P. G3P is triose phosphate, right? Triose phosphate, the tri means 3, phosphate means the thing have a phosphate to it, right? So it have a phosphate group. So G3P makes glucose, and any excess G3P regenerates the RUBP. That is it. That is it. Mr. Melvin Calvin, that is it. That's the Calvin-Benson cycle. If all you understand, watch back the video. What you gonna do? Right, limiting factors. This thing of photosynthesis, eh? All this near 2 hours, 2 hours of madness that I just went through. Sometimes none of that is coming. Like none of that is come, and they just end up bringing this instead, limiting factors. Now the thing is, I was looking through the past, like the last 7 years of past papers. They tend to bring limiting factors every 2 years, whether it's a graph or whether it's just to describe what is a limiting factor or some kind of associated problem involving limiting factors, where one of them is absent and what would happen to the plant, or if the rate would drop and why would it drop, things like that. Typically that's via photosynthesis questions, but you never know this time. I say we want this to be the last P2 that you write. I want to write no P2 next year. We want this to be the last one. We want this to be the last. We want the last P2 in your whole life. No more P2s. That is it. We fed up. No more P2s. Anyway, so what is our limiting factor? So this is what the graph for limiting factor looks like. The y-axis, right? The y-axis has the rate of photosynthesis. Is the y-axis, and the x-axis would have some factor like light intensity or temperature or carbon dioxide concentration. To double, you could get a KFC pepper with $10,000. I notice I'm saying $10,000, and that's probably like an extremely little bit amount for Guyanese. So I talk about TT dollars, right? Don't worry, that's not $10,000 could be like a little bit fooey. $10,000 could buy a little sweetie from the parlor. You could buy a little taxi ride to Shogunas. That's in Trinidad. Anyway, so here we have the rate of photosynthesis. Now, there's really two parts of the graph that we have to take into account, where light is a limiting factor and where another factor has become limited. Okay, so firstly we understand what is a limiting factor. So we have, remember the things that photosynthesis requires. It requires energy from sunlight. Remember, that's to split apart the water molecules. Carbon dioxide and water. Carbon dioxide has to affix the RUBP to make the 6-carbon intermediate via Rubisco. And water, of course, as I said, needs to split into hydrogen, electrons, and oxygen, right? It needs a reasonable temperature. Why does it need a reasonable temperature? Because a reasonable temperature allows Rubisco to work. Rubisco is an enzyme, and an enzyme can denature if the temperature is too high, or the enzyme wouldn't have enough kinetic energy if the temperature is too low. So you need a reasonable temperature for this to work. And lastly, you need chlorophyll, which is the site of all these light-dependent and independent reactions in the thylakoid membranes and the stroma. So of course you need all these things. Now the thing is, if one of these things was, let me say one of these things was to get totally removed, let me say that we was to take out energy from sunlight, it is highly likely the plant might have a backup mechanism. Some plants do, but let me just say in general, it is highly likely that no photosynthesis is going to occur. If we remove chlorophyll, photosynthesis is not going to occur at those parts because they don't have chlorophyll or the photosystems or anything like that. If we remove carbon dioxide, we don't have any carbon to affix to the RUBP for it to eventually become triose phosphate and then glucose. So if you remove water, there's no way the electrons can be replaced in photosystem 2 to help to eventually power those protein channels, the protein carriers, and to form NADPH, and eventually to power ATP synthase. So eventually now we need all of these things, right? But if one of these things was to get reduced, if one of these factors becomes reduced, so will the rate of photosynthesis. Light is the best example here to use. So let me say, for example, that we only have this amount of light intensity, right? Well, this would be the rate of photosynthesis here. It will be rather low. But if I was to increase the light intensity, let me say to here, let me say to increase it to here, this would be the rate of photosynthesis here. To answer the question, Anonymous, yes, some plants, not all, but some can switch to cyclic. The same way how we could switch to anaerobic respiration is like a good analogy. Some can switch to cyclic. In case there's a different deficiencies, they could switch, but only on a temporary basis, and it doesn't really produce much ATP. It's like a backup, emergency. So we could say here that light, at this point here in the graph, light is a limiting factor, because if we was to increase or decrease the light at this point, right? Limiting factor is when increase or decrease, it actually influences the rate of a reaction at this point. So light, if I decrease the light intensity, like here, the rate of photosynthesis is lower. If I increase it a little bit here, the rate of photosynthesis is higher. So that's what limiting factor. The limiting factor, it means that the rate of photosynthesis can actively be limited by that factor. So going the opposite way now, where light is not a limiting factor, which will be here, right? Where it is a plateau. This is where light would not be a limiting factor, right here. So why is that? So this is kind of like this. If I was to keep increasing light intensity past this point, this would just keep going like this. This would just keep at a plateau. If I increase the light intensity, the rate of photosynthesis doesn't change. Why? Because there's really only so much electrons that can be stimulated in those photosystems. There's only a limited amount of photosystems. There's only so much carbon dioxide that could be affixed. There's only so much water molecules that can be broken apart. So if I keep adding photons, if I keep increasing the light intensity, it doesn't make a difference past a certain point. It's kind of like enzymes. It's like if we keep adding substrate and we only have a certain amount of enzymes, the rate will remain the same. I'll remember enzymes from unit 1. Well, it's kind of like this. If I have 5 toilets, right, and I invite 50 people to a party and all of them have to pee, and then I add 100 more people to the party and all of them have to pee. Well, the only way I can actually increase the flow of those people having to pee is to just add more toilets. So in the case here, the number of toilets is a limiting factor, because that is actually limiting the rate at which people could go in the toilet and pee. That's a confusing example, but yeah. But in the case here, if I have a light bulb on the plant and it producing a certain amount of sugar, and then I shine a big floodlight on the plant, it's not going to increase the amount of sugar past a certain point. In fact, the plant might dead from the heat, but yeah. The limiting factor is past a certain point. Another factor has become limited. If you keep increasing this light intensity in this one here, it doesn't change. The rate of photosynthesis doesn't change. So there are different limiting factors to observe. Carbon dioxide. So for example, if we have a low level of carbon dioxide, we can see that the rate of photosynthesis, the max rate is kind of low. And if we have a higher level, the max rate is a little high. And well, I explained this already. Because if carbon dioxide concentration is limited, the Calvin cycle process is slowed down and photosynthesis decreases. There's less carbon that Rubisco could act upon. There's less substrate, basically. At high levels, it reaches a saturation point, where all of the Rubisco enzymes are currently occupied, kind of like all the toilets are currently occupied. And since Rubisco is an enzyme, the efficiency is affected by kinetic energy or heat. You remember, enzyme is a tertiary protein. Unit 1 thing again is a tertiary protein structure, which means that it has these hydrogen bonds, hydrophobic interactions, disulfide bridges, ionic bonds, all these different things that giving it a very specific shape. There's a very specific sequence of amino acids. And heat can break those hydrogen bonds and break all those bonds, and it can turn this specific enzyme shape into a big globular mess, just a bunch of stringy alpha helices and beta pleated sheets. So too much heat can denature an enzyme, causing the photosynthesis to drop to 0. And of course, the rate at which photosynthesis would be the highest is called the optimum efficiency. That differs by plant, but it is usually around what the temperature is outside. It might be around between 30 and 40 degrees Celsius. Actually, no, it's a little higher than what the temperature is outside. But yeah. And lastly, what they will ask you to do is how to apply your knowledge of limiting factors. This is the final part of photosynthesis in the syllabus. So there can be, you would know how to operate greenhouses, because greenhouses are where you would have artificial light, and dependent on the type of plant, you would know how much, how much light intensity or luminous intensity, or how much, light intensity, how much candelas or how much lumen or whatever those lights should be to optimize photosynthesis in those plants. Because eventually those plants is probably crops, and you want those crops to bear fruit, or to so you could sell food to make a profit. So you need the knowledge of limiting factors to see, well, what is the maximum light level so that your electric bill don't be too high. Because next thing you have the thing on too high, And you just kind of waste because the plant won't photosynthesize past a certain point, past a certain light intensity. The other thing here is growth chambers, where you have seedlings, tissue cultures, things like that, and these are highly dependent on temperature and carbon dioxide levels, and you would need to regulate them. An example of a very sensitive crop is groundnuts, and you would need to regulate those levels as the plants grow so that you can maximize their rate of photosynthesis. And that there is Module 1, Chapter 1, Photosynthesis and ATP synthesis. So we're not doing Krebs and thing, of course. Tell me if I'm feeling a little more confident with this. Fortunately, the second part of this is a lot shorter and a lot easier, which is biodiversity and conservation and things like that. That should take us a lot less time to go through. But I say sometimes you just had to power through the thing. Learning is a skill, you know. Learning is a skill, and you want to build up your skills as much as possible. So of course, this is why. Speaking of skills, let me take a few minutes to talk to you about a website called Skillshare. Have you ever wanted to try to learn something out of the ordinary, something interesting? Well, you can. Just take a few classes online at Skillshare.com. It's better than lessons.
You want me to show you tough? I'll show you tough.
Where you're taught by instructors and have their own class rosters. Hell, I learned how to juggle and make a peanut butter and jelly sandwich the right way using Skillshare. What are you waiting for?
I never lose.
It had dozens of online courses that you could take, ranging from physics all the way to finger painting, all the way to starting your own YouTube lesson channel. Come now, man. Skillshare.
As the adage says, you give a poor man a fish and you feed him for a day. You teach him to fish, you give him no. Cue.
All right, Ollie. Back to the live stream. When we finish biodiversity, we'll take our next break. All right, there we go. Okay, so this next section is called, somebody can remind me, this section is biodiversity, conservation, and something systems, energy something. I forget. The name of the thing. I mean, I know the work, but I can't remember the name of the actual chapter. But this is the second part of this is the second broad topic that we have here. That take me 2 hours, to go through for 2 hours, to go through photosynthesis. But photosynthesis is a big one. Anyhow, so in this topic here, thank you, Anonymous. The ecological systems, biodiversity, and conservations. All right. So, of course, I expect you to have the syllabus. All this is to pack that syllabus in all your head. That was the break. That was the break just now. That was the break. Okay. So biotic factors, the C-sex stuff. You could take this as the break, because this real easy. It might come for like 15 marks something, but it's real easy. Anyway, so biotic factors. Biotic factors are basically living factors that involve living populations and interactions. And remember, it have abiotic factors as well. So the abiotic factors is like rainfall, climate, temperature, sunlight, light intensity, pH, turbidity, which is like how muddy the water is, topography, or like the layout of the land. Those are abiotic factors, which means that they are non-living. And biotic factors and abiotic factors come together in order to form an ecosystem. So this module here, this chapter here more focuses on abiotic factors. Just making sure here. Yeah, because ecological pyramids and thing was in the last one. Right. Energy flow and thing, yeah. Anyhow. Yeah, well, 15 marks for cellulose and sucrose. Now it was cellulose and starch. Which actually kind of worse. But yeah. So biotic factors consist of members of living population and their interactions with each other. Typically, when a question like this comes, they will ask you for 6 types of relationships. And you might hold your head and ball and you might say, I only learned 3. But if you really think about it, of course, it's more than 3, right? So we're going to get into the 6 of them here. What you should be able to know are the 6 relationships, how to describe them, and at least one example. And this is come for like a 12 marks and things sometimes. Sometimes it might just come for 6 to just name them and describe them, but you never know. The first one that we have here is predation. So predation is basically where one organism hunts and eats another organism, all in this. Predator eats a prey. And this helps to control several populations in the food web. If you remember anything about predator and prey, you'll know that predator populations are usually smaller than prey populations, because prey populations is like the food. It's kind of like if you invite 20 people to a party and you only have food for 10 people, well, people are not going to stay in the party, right? So predators going to bust out if you don't have enough food. They're either going to go to our next ecosystem or they'll just stay there and dead and compete with each other, and let natural selection take care of it. So sometimes they get predator population graphs in this. I mightn't really go through that here, but you should know how to define it. Predation, one organism hunts and eats another. This is different from parasitism, which is a type of symbiotic relationship. Of course, the predator benefits from getting the food here and the prey dead, but parasitism, trying to the parasite doesn't want the host to die, right? The parasite doesn't want the host to die, because if the host dies, the food source gone. So imagine like you as a parasite copying labs from your partner. Well, you don't want your partner to dead, because then you won't get any more labs. That's a parasite. So we'll get into that just now. The next one we have is competition. Competition is where multiple organisms, they tend to occupy similar niches and they must vie for limited resources. So if we look at this example here, it look like this leopard and this lion there again on they're probably fighting over a gazelle, or they're fighting over territory or something like that. They're not fighting over a woman, because they wouldn't really it's not reproductive mates, but they're probably fighting over territory, and they say, hey, I'm supposed to be in this seat, boy, bust out of here now, right? So, yeah, right. But anyway, you have 2 types of competition. You have intra-species competition, and you have interspecies competition. So one of those is basically between members of the same species, and one of those is between members of different species. So you could have different lions, for example, competing with each other. Or you could have lions and cheetahs and hyenas and all kind of things, because all of them kind of fighting over the same kind of food. So that's actually a interaction, which is competition. It's not a symbiotic relationship, it's an interaction. It's a biotic interaction. So these are the 4 others here. The next one here is parasitism. So parasitism is like ticks on dogs. People copy in a lab. Don't use that as an example. But when one organism benefits while harming the other organism. So for example, ticks on dogs. The parasite tends to live on the organism or within the organism, like humans and tapeworms, for example, lice and humans. You have fleas and cats, ticks and dogs, and so on. So these are parasites. You also have plant parasites. You have one called Cassita filiformis, which is an orange vine. This called the love vine because what it does is that it doesn't have chlorophyll, it's orange. And it cannot photosynthesize. So what it does is kind of wrap around other trees and sap the nutrients from them. And it's kind of like it loving it up, the love vine. Actually, that's not what it's called, the love vine. It's called because people used to boil it and think it's an aphrodisiac. But anyway, so that's what a parasite is. It benefits while harming the other one. So one is harmed, one benefits. Commensalism is where one organism benefits while the other is unaffected. The example is a remora fish and a shark. So the remora fish or suckerfish, it attaches to a larger organism. This is not a shark here, by the way, but it attaches to a larger aquatic organism, and that aquatic organism moves around, and the fish it gets a free ride basically. So that's commensalism. Yes, CX is like a parasite. It harms you, but they benefit. Don't put that there. Mutualism, both organisms benefit. Example would be leguminous plants like pea plants and rhizobium bacteria, which helps with nitrogen fixation. All you want to know these examples already. So parasite is like somebody, you copy in somebody's labs and then them get catch and you get through. Commensalism is kind of like you copy in somebody's labs and nothing really happened to them, but you benefit. Mutualism is like all your copy lab, all your work on the labs together, I guess, and all they get through, right? That's mutualism. Altruism is usually common among insects like ants and bees. This is where organisms would cooperate. It's not seen in all organisms. Some organisms like to fight. But this is like everybody in the class come together and try to do the labs and try to help out each other. Well, you know what I mean? Everybody try to help out each other. So ants now, they would form a colony. And everybody, they would kind of have a division of labor. Some of them would try to get food. Some of them would try to scoop out territory. Some of them would help to protect the area. But that is called altruism, where organisms cooperate towards a common goal, and that helps with survival. So that is the 6 biotic factors there, and 6 examples. So we have predation, we have competition, parasitism, commensalism, mutualism, and altruism. So these are some abiotic factors, and typically you don't need to memorize too much of these. You should know about 4 or 5 of them. But typically when they ask about abiotic factors, it more relates in contrast to biotic factors. I don't think I need to really go through all that, so I'm just going to read them really fast here. So first we have light intensity. And this, of course, impacts rate of photosynthesis and animal routines if the animal is nocturnal or diurnal, sleeping patterns, things like that. Everybody's sleeping kind of late nowadays. Temperature impacts water supply, type of vegetation, metabolism, even rate of photosynthesis if it affects enzyme, if it affects enzyme activity and things like that. Gas concentrations, this is like nitrogen, oxygen, carbon dioxide, this impacts rate of respiration, photosynthesis, nitrogen fixation, things like that. pH impacts habitat survivability, so some organisms can only survive in alkaline or neutral pHs. Some plants cannot survive in acidic soil, things like that. Also, how much water is available, drinkable water. Type of soil, this includes the aeration of the soil for some subterranean organisms like earthworms. pH, particle size. Particle size helps with retention of water, mineral concentrations for plants, right? Then we have water itself. This includes the availability of the water, the type of habitat, the turbidity, that's how muddy the water is, the salinity, that's how salty the water is, whether it's brackish water, fresh water, salt water. Remember, like freshwater fish can survive in salt water, and pH. Wave action, this is more for coastal ecosystems, where if you have a lot of wave action, it can result in erosion. It could help with but it could also help with the movement of nutrients from the coast to land or from the water to the land. Humidity, humidity and wind speed we'll get into when we talk about transpiration. And topography is the layout of habitats like the land. So for example, some animals may want to live at high altitudes, some would live at low altitudes, and so on. Okay, so this is where the bulk of your marks might come in, because there's there's some 9-mark questions and things that involve biodiversity. And we have to know biodiversity as an amalgamation of 3 factors, which is species diversity, genetic diversity, and ecosystem diversity. So biodiversity is said to be linked to ecosystem stability, right? And basically what that means is that what is ecosystem stability? So ecosystem stability is like this. I want to imagine that wherever island that you're living on, right, that it get hit by a bad hurricane. We hope that will happen. They get hit by a bad hurricane or some kind of natural disaster. Of course, what's going to happen is that a lot of the, not just urban destruction or residential destruction and things like that, but you're going to get destruction of the ecosystem. You're going to get habitat loss. You're going to get, the topography is going to change because you have trees that topple, soil going to get uplifted, you're going to get flooding. You're going to get, some animals will die, some plants will die, and so on. You're going to get water mixing everywhere. You're going to get diseases, all kind of different things. So ecosystem stability relates to the ecosystem's resilience and the ability to return to a normal state. So you could think of it as a restorative state, so where it could be restored to what it once was after being negatively impacted, of course, without the help of man. So how long it would take to actually return to normal. So sometimes things has happened and it's never returned to normal, and that usually means that area had a low biodiversity. So if the area, they said the theory is that if there's a high biodiversity, the ecosystem would be more stable. And we have to be able to explain how are those two things linked. Because biodiversity is species diversity, genetic diversity, and ecosystem diversity. How do those 3 things actually help to restore an ecosystem if it was to be partially destroyed? And that's typically how your questions come. So first we have to understand what is meant by those 3 things: species, genetic, and ecosystem diversity. So first, let's look at species diversity. So species diversity here refers to the variety, it refers to the variety of different species found within a biome. So basically the sheer number of species populations that you're going to find. So if you look at this picture here we have some elephants, we have some zebras, we have some lions, we have organisms that would be in this little pond here, we would have insects in the grass, we have the grass itself, we would have many different types of grasses. Trinidad is actually, or the Caribbean I should say, not just Trinidad, is actually a highly diverse ecosystem. A lot of the islands are very diverse, because if you look at any old bush and you look and say, I'd be like, hey, them living in the bush, that's bush. But it actually have a bunch of different types of grasses. So if one type of grass was not to survive, at least the next types could. You have paspalum grasses, you have Rhizospora curvula grasses, you have what is called elephant grasses, You have what is called fever grass. You know, you have all them different things. You have a whole set of different, when you look anywhere, you will see at least 10 different species of plants in any bush. Whether it's like in the cover below, whether it's like growing above it, you would have a whole set. So the species diversity refers to the variety of different species, like how many different species can I count? And this would not just be those large organisms like the elephant and the lion and thing. It would include the plants. It would include prokaryotic organisms. It would include algae. It would include insects. It would include arachnids. It would include birds. All these different things here. So what it doesn't include though is the abundance or the size of the populations. So that could be one of the little limitations of using species diversity. Sometimes the place could just have one lion, and you don't really, you still have to account for that species being there. But examples would be a savanna may have many different species of grasses, shrubs, trees, birds, insects, large mammals. Tropical regions tend to have high species diversity because we have a high rainfall, so there's a lot of water available. And there's a lot of nutrients, that could be deposited all over the place. And we also have high light intensity here. We don't have like long periodic moments of darkness and thing, We kind of have a steady enough climate and steady enough temperatures and things like that. So that could be good and that could be bad, but it seems that a lot of organisms have thrived in these tropical environments here. And you can see it. You can just go outside and you can look. So this is species diversity. Now, genetic diversity refers to the variation of genetic information and phenotypes within the populations of a species, right? So the DNA, the phenotypes, all the different variations of organism. So you know that only remember natural selection in Unit 1, where you had Darwin's finches, the Galapagos finches, and all of them originated from a common ground finch. And what was different between them was their beak size. And because the beak size was different, these birds didn't necessarily have to compete with each other. One of them could have fed on fruits. The one with the thick beaks could have fed on fruits. They could have cracked nuts with it. The ones with the longer beaks could have feed on insects and worms. Some of them could have feed on seeds. So they didn't have to compete. It reduced competition, this genetic diversity. So this meant that, let me just say that all of a sudden, all the worms was to disappear from an area. Well, this fella might dead, but the rest of them might still survive. And these here could still reproduce and speciate and all kind of thing and overcome selective pressures. So high genetic diversity means that if the environment was to suddenly change or suddenly be altered, where the temperature was to increase, or whether the water level was to rise, or whether a certain type of food was to stop being available, at least some of the organisms might still survive. So this one might dead, and let me just say that all the insects suddenly move out of the area. Well, he might dead, but the other members could still persist. So this is how genetic diversity actually contributes to ecosystem stability. So having selective pressures and sorry about that. Having selective pressures and high rates of outbreeding. Outbreeding means like sexual reproduction, basically. Also reproducing outside your family, not incest, basically. Increase the size of the gene pool and the number of favorable alleles and traits. So again, that's like Unit 1 stuff. So members of the species are more likely to resist disease and environmental changes. And this raises chances of survival. So when you have some kind of pressure that comes in, whether it's natural or whether it's artificial, whether it's man-made. I'll give you an example. I'll give you a Caribbean example. I don't know if you all know, about 100 years ago, the Caribbean had seals. We had Caribbean monk seals. We had seals over here. I never see them. They're dead. So this is because well, it had poaching first of all. There was around Jamaica, so the Jamaicans and them killed them. And they also had like over the years it had temperature rises in the oceans now. Eventually they couldn't survive. And the seahorses, which wasn't found in the Caribbean, but they tend to produce about 1,000 and 2,000 babies at a time because maybe only 5% or 2% of that survive. But because they produce so much of them, because there's an overproduction of offspring, that increases survivability because they have a lot of genetic variation. Leatherback turtles too, they overproduce offspring so that many, could survive. So anyway, so that genetic diversity would help with repopulation. The last thing that we have here is ecosystem diversity. So ecosystem diversity refers to variation in ecosystem types. So in Trinidad, we have what we call the ASSR, not ASMR, ASSR, which is the Aripo Savannah Scientific Reserve. And it usually like normies and things can get in there. You need to be a scientist, so you need to have permission for some kind of research to get in there. But all Trinis is still go and break in there and go and squat and cut down trees, all kind of thing. But basically, this scientific reserve, this has an open savanna, a palm marsh, a marsh forest, plenty rivers, plenty different types of soil, hundreds of types of flora and fauna, many different types. And scientists actually come from all over the world to study these things. In fact, guppies, when they come to study guppies is like one of the keystone species, and those Trinidadian guppies specifically is like one of the keystone species of evolution, where they observe how their body moves, how their eyes move, how their size changes according to environment. A lot of these are actually found in that reserve as well. Selective pressures are think of them as barriers that organisms have to overcome to survive. So like a selective pressure, I'll give you an example, is like imagine a bear is chasing you and your friend. And so selective pressure would be the bear in this case. But to survive the bear, you don't have to be faster than the bear. You just have to be faster than your friend. So whoever is best adapted to the environment can overcome a selective pressure and thus survive, and then they can find a reproductive mate and then they could reproduce. So selective pressures could be anything, but it's basically something to overcome. Right now, all this selective pressure is keep chem, keep bio, keep physics, keep pure maths, all kind of thing. If all they survive, all they dead. Become vagrants and dead. So anyway. So the Aripo Reserve has many different ecosystems actually right adjacent to each other. So all of this here encourages, thanks for the comment, Ajax, or Ajax, whichever one. All of this encourages intermingling of species and provides many choices of habitats that encourage survival if selective pressures are right. So let me give you an example. Yeah, that's CXE, CSEC. Let me imagine that we have two ponds that are close to each other. The two ponds are slightly different. And we have frogs that live in, let me say we have frogs living in this pond, right? But have a next pond or next river or next water source or something that near to this. I believe we say that we have a crocodile, or not crocodile, without those. We have a caiman that come in here, right? We have a caiman that come into this river, and this caiman plop down and start living there. The caiman might eat the frog and them. But if we have a next river right next to that, or we have a next pond right next to that, right, that the caiman can't really get into, well, we frog here could just hop to the next one, and he could be safe because it have many different ecosystems right next to each other. So even if a man was to come and dump all his KFC in here, right, He was to dump all the rubbish and all the KFC and all the garbage and all the battery acid, and then he was to pull on his pants and squat and do his business right there, right? Now that might kill some organisms there. That might kill some frogs there, who knows? But if it have a next pond like this right next to it, then it have ecosystem diversity. And that frog now has a choice. I don't need to stay here. I don't need to stay here in this polluted environment. I could go in this next place that look nicer. It's kind of like if you go into a school, right? And somebody beating you up in school. Some bully come in the school and beating you up and taking your money, you know what I mean? If you're not the bully yourself. I'd be like, I don't want to stay here. I get a transfer to our next school, you know what I mean? So it's kind of like that ecosystem diversity. This place I'm working out, let me go to this place. If you have two houses and somebody come and mash up your house, right? Well, at least you have a second. Well, not much people have two houses, you know what I mean? But at least you'll be like, my house mash up, you know. But my next house, we're going that one. That's ecosystem diversity. So how does this relate to ecosystem stability? So ecosystem stability, as I said, is stable, and this is where the bulk of this topic is here. Oh shucks, just now. Oop, my bad. So a stable ecosystem is one that has the ability, as I said, to return to a normal state after disturbance. It is resilient, is able to be restored easily. So high species and high genetic diversity means that less species are prone to migration or extinction after disturbance. It means that, okay, well, somebody come and shake up the place, a hurricane come, some kind of urbanization happen, you have a bunch of noise pollution, an airport bill, who knows, something happen. But what this means, if it has a high enough species diversity, it means that enough of those species are able to survive, right, and have biotic interactions. They are actually still able to continue altruism, they're able to still continue predation, they're able to still continue competition and mutualism and all these different things. High genetic diversity, of course, ensures that you have a high gene pool, you have all these advantageous traits, you can overcome selective pressures, blah, 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 blah. All that could still happen. So you'd have all these favorable traits to adapt to the changes. The point is that you have to adapt. Things can't stay the same forever. You have to adapt. Things could be changing for only in a few months. All you have to adapt. So high ecosystem diversity makes it less likely that a species will die out or go extinct or have to migrate, as they can simply adapt to another biome. As I said, if you have a frog and an invasive species was to come like a caiman, the frogs can't be like, oh God, I have to hide from this caiman forever. Nah. What the frog could do is that the frog could simply go to our next adjacent ecosystem. All of these things here contribute to ecosystem stability. And I said this could run here for a good 8-9 marks to explain how species diversity, genetic diversity, and ecosystem diversity all contribute to ecosystem stability. All right. Let me take a 5 again. And then I'll go through into conservation.
What is this whole spewing thing about? What is it about?
What that question mean?
Tell me what this whole spurwing thing is about.
What you mean?
Mr. Montano, tell me, how come all these people watching spurwing? How come you just come on YouTube and watch spurwing?
Let me explain something to you. I live in with a higher understanding of life. And I will tell you something about life. Right? I love every being, every human being. It doesn't mean I have to be intimate with them.
Correct.
It doesn't mean I have to be intimate with them. But I love them the same. Remember I tell you, if you pay attention to life, life will pay attention to you.
Hey!
Alright, y'all. I feel we got to start back. So let me finish conversations. I mean conservations. Let me finish that. Let me finish that now. Oh, everybody feeling all they tired? You know, if you're feeling tired, sometimes it's good to stay home, to work from home, and to build a website and to try to, you know, do stuff. You could sell stuff online. And, well, you know, this is why. Squarespace is the absolute easiest way to make a website. I use them for a few different sites myself. I basically buy that domain to be sure nobody else could. I didn't really have the time or need to create a fancy website. I just take about 15 minutes to throw together a landing page. It was incredibly easy with the Squarespace template, and in my opinion, at least, it looked great. Squarespace. And we're back. There we go. So this is the next part here that is kind of like the big part of this topic, which is maintenance of biodiversity and ways in which conservation is being done. Raj, but I will actually give you a list of that after the break. I'll give you a list of the paper questions. I actually have the questions done already, so I will link you that too. So I have the paper twos done. I have the answers actually on the paper, so like typed up already. So you just have to read it, because I know the exam is an AM exam, so they have to get all the, you know, they have to get all the rest. I'm aiming to go until the time I say was 10 p.m. But I feel, you know, people need their beauty sleep, so I might aim to go to 9 p.m. I'm trying to cover. I don't know if I'll cover everything here, but trying as best to cover as much as we could. Because I know like module 3, health and disease real short, and immunology is what will take up the bulk there, but like some things will be transpiration real short too. But module 1 have 2 kind of long ones here, so that's why we're taking a little while. Anyway, so I say we want to be as in depth as we could. Alright, so importance of maintaining diversity. We have to think about this in 3 categories: intrinsic, direct, and indirect. So what do those 3 categories mean? Let's start with intrinsic. So intrinsic means anything that deals with just for the value of existence. So I'll give all the example. Me doing this live stream for all you, this is totally intrinsic. I mean, the ads here, but the ads for free too, you know what I mean? But I'm not really gaining anything from doing this, if you know what I mean. I don't give lessons. I'm not offering a business or anything like this. Literally come out from the dead just to do this, and then I'll go back to sleep when CX is done. I don't gain anything from doing this. I don't care about subscribers. I don't care about views on the videos. I don't care about how much people hear. It's totally intrinsic to me. I really just doing it to help all you out. I have no ulterior motive for doing this. I really just want to help out people. Past the subject, past things like, well, you know, what I could help with. So these are basically just totally ethical. There's no profit in doing it, right? It's just for just the value of existence. It's called existence value. Some things you don't have to do everything for money. Some things is just do it. Some things is make you feel good. Some things it is just for the benefit of society. You just do it. And so for intrinsic values, for intrinsic reasons, right, of maintaining diversity is one. Of course, ecosystem stability. Just for the existence of the thing, just for you want to see the ecosystem continue. You want to see ecological recovery after it have a disaster. You mightn't benefit from it in any way, but you just want to see that happen. You just want that there to be ecological recovery. You want certain species to not go extinct. You probably sometimes see species that was extinct and you wish they were still around. And sometimes you'd be like, hey, it might be cool if dodo birds were still around, or if the orc was still around, or Caribbean monk seals were still around. So that's an intrinsic thing. Protecting endemic species from extinction. Same thing. We have the Pawi in Trinidad type of bird. We also have the golden tree frog. Endemic means they only found here. Right? So we have the golden tree frog, which is only found along this range that actually, well, not just in Trinidad, but there's a few that found in Venezuela because we kind of break off from Venezuela. Like part of Trinidad underwater, and there's actually connected Venezuela. So it's a lot some there, but there's a lot of species here that are only found here, and you want to prevent that from extinction. There's really no benefit to prevent it from extinction. You just want to do it. Aesthetic. So aesthetic, I surely heard that word already, but nature itself is a source of relaxation, beauty for visitors. Not so much like tourists and for profit and things like that, just the fact that it exists. Just you could take your Instagram pictures and get 1,000 likes on it, make it TikTok, make it strippiness on the on mountain trails, do all kind of strippiness. So that's aesthetic. Bequest value means that you don't want it just for you, but you want to preserve this environment so that future generations can experience it. You want this to exist for your children, your grandchildren, your niece, your nephew, your whatever. You want certain things to still be around. So that's why you call a bequest value. So all these things are intrinsic. So now we have direct, which are known economic uses. And these here is to make money, right? To make dollars. Ka-ching, 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 right? So we have two main ones here. One is tourism. Right? And tourism is like, well, you can make money if you're living in a place with plenty sites. You can have nature tours, you can have trips, into Tobago, go to Tobago, people hanging it down to go to the Nylon Pool, to go to the Boko Reef, to go this reef, to go this beach, to go Argyle Waterfall, to go all kind of different things. You have all kind of trails and you have groups dedicated to doing this. So they can earn revenue for themselves and for the country. Then also you have raw materials. So this, if ecosystem stability, if there's a high ecosystem stability, there's a high chance that there's going to be a high biodiversity, as I said, and that's going to result in many different types of trees. We have mora, we have cedar, we have serret, we have all kind of different things, and this is going to provide timber, and then you have many different types of minerals in the soil that can be extracted from mining, and ores that can be responsible. There should be responsibly, not responsibility, that can be responsibly harnessed. And so those things you can make money from. I'll give you an example. It have this Pacific island called Nauru. And it's a very small island. Much smaller than a lot of Caribbean islands. And they made real money. They spent out all the money. But they made real money because they had potassium. I think they had potassium or phosphorus, one of those. They had plenty deposits on their island, and they made riches. And then they're diversifying us. So once all done, they go on through. It's a corruption. But raw materials, of course, all in Guyana, it's a real crude oil and thing. Raw materials is money once it can be exported. The other category here is indirect values, which are those that have unknown or non-immediate economic use. So these here are not necessarily to make money, but they could probably prevent you from losing money, or they have the potential to make money, if you know what I mean. So first one here is flood prevention. You have trees, you have grasses, you have many types of vegetation. Deforestation and removal of vegetation would leave the soils exposed, which means that when rain falls, the force or the impact of those raindrops dislodges a lot of soil and causes erosion. Soils become waterlogged, and the problem with this is that, well, you wouldn't necessarily lose money because it have some erosion, but if that reach, they were saying in Trinidad, that reach people's house, and that reach Port of Spain, and people can't go to work, and that reach inside your house, and it ruin your couch, and you have to buy a new couch, and people house and thing mash up. Well, you have a problem. So this can decimate habitats and surrounding residential areas, and guess what? It costs money to repair all those things. It costs money to repair the roads. It costs money to clean up the areas. So even though this is a non-direct or indirect or non-immediate economic use, the flood prevention prevents money from being lost. Lastly, we have scientific value. So this is where you can have a potential gain of money or some kind of other interest. So researchers obviously use ecosystems and organisms as models for medicine and technology. There's a really good book called Biomimicry, which shows how a lot of the things that we use today, like Velcro, for example, is actually modeled after observing how organisms behave or observing the physical characteristics of organisms. So Velcro, which is using straps like for your shoes or for your book bag or your laptop bag or something. This was modeled after birds, which is like a type of seed. So they stick into clothing, which they have hooks that stick to animal fur. Yeah, Japanese bullet trains were modeled after kingfisher birds. That's true, because when those they model after the beaks of the birds because they make very little noise when going into and coming out of tunnels. That's correct. And then you have genes from daffodils were spliced into rice to make golden rice. So I remember that from Unit 1 with genetic engineering. You have plenty different things here. You have observation of how honeycomb structures could be used to lay down bricks, for example. That we have fully implemented that yet. We have genetic engineering of plants to make caffeine, so that they could, they could use it as a pesticide. So scientific value is there. So you want to focus on intrinsic values. Direct means that you can make money from it as an economic use, and indirect, which means it doesn't have an immediate economic use, but basically you could save money. If you don't do it, it could cost you money, or you can actually have the potential to gain money from it, but it doesn't directly give you money. Right. So now we come into conservation. So what is conservation now? We was more talking about ecosystem stability. So conservation is the discipline of maintaining diversity, right, and protecting species and their habitats, basically preventing them from going extinct or preventing their populations from dropping to endangered levels. You want them to survive, you want them to reproduce, you want them to speciate, you want them to proliferate, all these different things. You want them to stick around, because if one organism disappears, that impacts the entire food web. That impacts the entire, it kind of causes a chain reaction in the biotic factors, with all these different levels of competition and predation and symbiosis and all kind of thing. One organism gets removed, it could cause a major impact. So in situ or on-site conservation aims to so in situ in Latin means on-site. So when we say on-site, it means that, well, the conservation that you're being done is on the site itself, of course, right? But it means that you're not taking the organisms out of their natural habitat. You are just doing your best to preserve the natural habitat that they are living in. So on-site conservation or in-situ conservation aims to preserve natural habitats and maintain or replenish population numbers. So either to get the populations up or to maintain what it have already. So examples of this would be like national parks, which are protected. Example would be the Yosemite Park or the Sequoia National Park, which is all these large trees here that are hundreds of years old. We also have the Serengeti National Park and so on. So we also have wildlife reserves. For example, in the Caribbean is the Aripo Scientific Reserve that I just said, where basically hunting is banned there. If you cannot go into there without the normal permission and things like that. And that's just there so that, okay, you don't want population numbers to be affected by human activity. And that is in situ conservation. So this is where it is done on site. This is where you don't remove the organisms, you are going to the organisms to help them. Ex situ conservation or off site, right? So ex situ aims to preserve target species outside of their natural habitats. So I'll give you an example. So panda bears, one reason or another, they have difficulty reproducing in the wild. And Carony swamp could be an example, yeah. I don't know if it counts as a reserve, but if you ask for, use the Aripo Reserve as a, because that's actually a legit reserve. Panda bears don't really the bad at reproducing in the natural habitats. So typically you have to kind of force them to reproduce in captivity. So they have to be kept in a man-made environment, kind of put together where, you know, reproduction could happen, right? The courtship and things like that. So you're actually removing them from their habitats. Right, you're actually moving them from their habitats and putting them in an artificial area, and so that you can get the populations up. So another example, some turkeys cannot, for some reason, they cannot really reproduce properly in the wild either. If there was the ones in the wild, although they're mostly domesticated. So you actually have a job, somebody who will stimulate the male turkey to ejaculate. So that's actually a job, like a turkey masturbator, that's a job. And they would collect the semen, and then they would deposit the semen in the female, like in vitro fertilization. So of course, again, that is excessive conservation to get population, because you have human intervention in there. I'll do predictions after the break. And examples of this would be zoos, botanic gardens, like the Royal Botanic Gardens in Trinidad, the Emperor Valley Zoo in Trinidad. You have seed banks and pollen storages. So for like plants that endangered or going extinct, you can actually just store their embryo sacs or their ovules and their pollen grains in, you can freeze them and store them, so that if they was you could grow them in germination chambers and things like that in an artificial environment. So you can have in vitro storage for sperm cells and embryos, which is animals that going extinct. Well, somebody could collect the sperm or collect the egg cells, the oocytes, and store them. And then you have captive breeding programs like the one here with the panda bear. So all of these, the botanic gardens example will be this. Example of so I'll say all of these here, you're taking them out of their natural environment and attempting to conserve the species in that way. So yeah. So here we have some comparisons between in situ and ex situ, right? So of course, in situ involves natural habitats, and in situ, involves natural habitats, so it is cheaper and allows for the process of adaptation, evolution, speciation, overcoming selective pressures, all these things, natural selection. Ex situ is usually when the natural habitats have been destroyed. So like in Indonesia, what they call, I can't remember, they just call it the Indonesian basin. It have a name, the Indonesian basin. It's like one of the main, like how it's the Amazon, and have the Congo basin, have the Indonesia one. And in there, there's the orangutan and some of the elephants. They have been going extinct because of deforestation. And they have, natural habitats have been destroyed because they actually cutting down a lot of the trees in Indonesia to grow this type of palm that produces palm oil. palm oil, you just find it in Pringles, you just find it in basically any processed food that you buy is a good preservative. But a lot of the forests have been cut down for these palm trees here to grow palm oil. And that deforestation, combustion and things actually contribute a lot to climate change. But the habitat loss from it, they had to take out some of those orangutan and elephants and kind of raise them elsewhere. So that's ex situ. So where the species have been in rapid decline or where the habitat has been destroyed, of course they can't keep them there. If the house burning down, we can't say, hey, we could take care of you. No, we have to take you out of that house and, put you in a safe way, a safe one. So in situ usually does not have any issues with breeding or reproduction, though inbreeding may be possible with plants. So typically you release these organisms in the wild and, they have sex, they reproduce, they're good. They're good to go. They know what to do. Nobody had to teach them what to do. They know what to do already. But with ex situ, some organisms are placed in a man-controlled environment, and typically they have to change the mood a little bit now. They have to change, they have to create a new lighting for them, that they have to put on some music and thing for them. They have to get them in the mood for them to initiate the courtship behavior, not the music, but it's a different factors that they have to control. Because sometimes in the natural habitat, it's kind of like, well, we can't mate, it has too much noise, it has too much madness going on around. We need to be in a quiet place. Human, please put me in a quiet place. Please put me in a nice, warm, quiet place. So I could court my mate here. You know what I mean? Yeah. So it's a man-controlled environment. And of course, when we need to change these abiotic factors for seasonal organisms and things like that. So yeah. In situ, there's no technology being utilized, no medical aid for organism health. It basically just be like, hey, I keeping all your place safe, all you have to do. Ex situ, on the other hand, you need to have medical aid, you need to have security for the organisms, you have to make sure that they're not being poached, you have to make sure that they're being fed all the time, you have to make sure that they have a proper captive breeding program, so that without incest and thing, of course, you have to make sure that you have no inbreeding between them. Next thing is that in situ requires large areas of space. As I said, is usually a whole national park that usually be preserved. The animals need a lot of space. Ex situ is done in more confined, smaller areas, like I say, zoos and botanic gardens. Though these smaller areas may affect organism behavior, basically they get kind of lazy now, or they get a little confined. But in situ is like wide out in the open, ex situ confined to just, I say like a zoo or botanic garden, or if it's like preserving embryos, they'll be like in storages. And that is module 1. That take us 3 hours to cover module 1, but it was 2 long topics. So we're going into I think we could hop into module 2 one time, and I'll try to finish work until 5:40 p.m. That's basically 40 minutes from now. And I have to eat dinner, or you know what I mean? So I'll try to finish as much as transpiration as I could, and then we have to go on to circulatory system. So we'll finish transpiration before the break, and then come back, do circulatory system, health and disease, and immunology. By then it should be like 9 o'clock or something by the time we're done. All I feel all you have the stamina still to keep going, or I feel all you're gonna make till 9:30 and thing. But we're gonna have a break between 5:40 and 6:40. Ecology is not that bad. It's a lot of writing, but it's a lot of common sense. Real stamina? Nice. That's what we like to hear. Okay. Because we have them AM exams is real killer, Okay. So, let me get started one time. We're on module 2. Module 2, transpiration. All right, so the thing about this topic here, a lot of this topic you would have done in Form 5. There's some aspects of it that are new, that would have been new here for you. And those are the parts here that I might spend a little more time on. Okay, so first of all, let's define what transpiration is. So transpiration is the movement of water through a plant and evaporation through its above ground plants, such as mainly the leaves, because the leaves have the stomata that have the hole, they open up and let some water out, right? So this is what happens here. So the water enters through the roots. So water would enter through here, and the reason for that is because typically there would be a high water potential, or where you'd say, remember for osmosis, it is go from less negative to, less negative, that is always chain map, less negative water potential to more negative. You need to talk about osmosis and things like that, just the fact that it exists. But you can say higher to lower. So when water enters the plant root due to osmosis from a higher to lower water potential up the xylem vessels, so it goes here, it goes zoop, come out the leaf, boo, right? So it comes out there, and those water molecules are like in a long chain. And this is because, if you remember, Unit 1, Objective 1, Module 1, Chapter 1, water molecules are cohesive, which means that, the polar and they have a dipole structure, the hydrogen and oxygens, which means that they could link easily into like a chain, and they could stick together. So we say that they're cohesive, right? They could undergo cohesion. So they kind of link together into this long chain up the xylem, pass through the veins, the net venation of the plants, the vascular bundles, and eventually they could leave through the stomata. So what would happen here is that sometimes they would break off kind of one by one through the stomata here. But so these would be like water vapor molecules. And it's like a straw, it's like a drinking straw. But the thing about this is the external environment actually affects how rapid this rate would be, right? We'll get into that now. So water is delivered to the cells, excess water is diffused out to the leaves. There are different factors that affect the opening and closing of the stomata, which we'll get into just now. Right. So firstly, something new here, which is apoplast and symplast pathways. So apoplast and symplast refers to ways in which this water could actually travel through the cells in the root hairs and the roots. And I like to think of it like this, right? And your teacher probably said this for you too. Apoplast, you can think of it as going across, right? Across from cell to cell, and symplast, you could think of it as going straight through, right? So A for across and symplast as in straight through. So you can see what I mean by across, right? So on the diagram here, symplast, let me start with symplast. Symplast is the green arrows. So you see what I mean by straight through. So water enters the root cells by crossing the plasma membrane. Remember, the plasma membrane is partially permeable. And here you would have a, I'll write it here. Here you would have a high, right? So here you would have a high water potential. That really looked like nothing right there, no? But they get it. Here you have a high water potential. And inside the cells you have a lower water potential. Of course, that will take water into the cells. Now, what's going to happen here is that the cell walls, which are here, the cell walls have little gaps in them. And these gaps are called plasmodesmata. The singular is plasmodesma. And these little gaps here are kind of like, think of them as little breaks in the fences. So the water could actually pass straight through these gaps here. and they can actually go straight to the xylem as a result. So that's why you call the simplest pathway. Is actually pretty simple, where it passes through the cytosol, the cytoplasm and things like that, and it can actually be deposited into the vacuole or come out of the vacuoles and travel through like that. But it basically refers to passing straight through those little gaps there. Now, that is the simplest pathway. And note here, sometimes they ask you to draw all the cells, so you kind of have to come back to Unit 1, Module 2. Plenty Unit 1 thing you have to come back to it. But remember, here we have the epidermis, the cortex, which has some air spaces, the endodermis, the pericycle, and then we would have the vascular bundle that contains the xylem, right? I don't know if somebody could come up with a little acronym for that. We have easy, no, you see epic, keep, ends, I don't know, something with P. epic keep, ends, but passes, I don't know. Or they could come out. The more stupid it is, the easier it is to remember it. Anyway, so epidermis, cortex, and endodermis, pericycle. Now, the only one really here we have to look at, we have to take note of, is the endodermis. So the endodermis is a little different from those others, and we'll explain why now. Now, apoplast or across, right, means that the water does not enter the cell cytoplasm. So there's the purple. So the water instead stays in the cell. Yeah, epic keep ends pressure. That is true. That is true. Epic keep, at the end of it is it ends all the pressure. Yeah, but pressure done. Once keep done. Who knows? Epic keep examination pains. That's a good one. That's a good one. So the more stupid it is, the easier you remember it. Anyway, so water doesn't enter the cytoplasm. Now you have to remember now that the cell wall, the cellular cell wall, is freely permeable, right? And what freely permeable means is that passage of water is uninhibited. It can simply just flow. And so the water flows along here, but look what's happening here. We old friend, I don't know if you remember, we old friend from Unit 1. We old friend Caspar and he Casparian strips. And if you forget what's Casparian strips are found in the endodermis and they are impermeable to water. They're made up of a compound called suberin. And suberin is the same compound that is used in wine corks. And it's similar to what very thick tree bark would feel like. So those Casparian strips are made up of suberin, very, very thick material. Those block water from passing through. So it say water, you shall not pass water. So what's happening now is that the apoplast pathway cannot continue like that. So it be like water, nah man. So you see, usually you would expect it to go through here and to end up with that happening. This man, there's the bodyguard. Be like, fella, you're trying to cut in line. Fella, get in line. So the apoplast pathway have to now switch to a simplast pathway to get to the xylem. So it is happening normal until it reaches the endodermis. That's what I say is only the endodermis really you have to take note of. Fella, you come here, normal. Nah man. Casparian strip, Casper, let me through now boy. Casper, nah, water, get in line. Alright, I want to get to the xylem. So that's what happening there. So, impermeable Casparian strips in the endodermis, right? Impermeable Casparian strips in the endodermis forces water, what's going on here boy? Forces water to switch to the simplast path to reach the xylem. So that's the difference between apoplast and simplast. If you have to draw the diagram, you want to draw something like this. But you could even draw more cells, like other layers of cells to this as well. So I will link all this on past papers before the break so you can see. So that's the difference between apoplast and symplast. Right, so what kind of questions could come for that? They could ask you to define both, and they could, you see this diagram here? They like this diagram. They love it. They love it. They love it. They just give you a diagram like this, and they just ask you, draw the apoplastic route and draw the symplastic route. And they might tell you, they might ask you about the endodermis, what is happening there, you know what I mean? So if you look here now, so all you gotta pay a little attention to this because this here is getting a little confusing, right? So the endodermis here now, right? The endodermis, which is region 4 in the diagram, as the water flows from 4 to 5. Right? So the endodermis there. This contains protein carriers for active transport as well. And of course, you know, active transport is the movement of substances from molecules from a low to a high concentration using ATP. And these typically employ protein carriers. So there are protein carriers on the endodermis. That's we boy here around 4. And these here would actually pump ions into the xylem. These ions could be potassium ions, some sodium ions, different minerals. So they pump ions here. So let me just imagine, I just put in I for ions, right? So we have a bunch of ions here now. Well, you know what they're doing? Well, that is lowering the water potential over here. That is dropping the water potential in the xylem. So it is making it more hypertonic. Not hypo, right? Hyper, like you have plenty of sugar. So it is making it more hypertonic. So what do you think that is doing? So if the water potential becomes lower here, that actually increases the osmotic rate. So it allows even more water to come in. So this is called root pressure. Yeah? So that's called root pressure. Let me explain that one more time, right? So it could be a little confusing. So I'm going to start from the top one more time. So this area here, the endodermis, this has protein carriers. These protein carriers are used for the active transport of mineral ions, not water, or water's osmosis. I say this is usually like potassium and things like that, and that would be in the soil. So this would allow any potassium and things like that to get pumped into the xylem. So I'm drawing this as I for ions. So these ions get actively transported into the xylem here. Remember what this is doing now is that this is lowering the water potential, that's a symbol for water potential, PSI, psi. So that there's lowering the water potential here. It's making it more hypertonic. And that there is going to increase osmotic rates because there's going to be a bigger difference between the water potential here and here. It's gonna basically widen the gap. So this is gonna allow water to flow even faster. So it's gonna provide a kind of pressure to allow water to flow even faster through the roots and into the xylem. So this is called root pressure. I've seen a few questions come on that. So that is called root pressure. So if you ever hear the term root pressure, that's basically what it is. It's not really, I don't know, root pressure is like a bad word for it. I don't know why I would have endodermis. I don't know why I would have called it endodermic pressure, something. But it's basically due to the water potential changes because all these ions getting pumped here. Alright. Hopefully that wasn't too confuzzling. Anyway, so let me look at xylem function and structure. So xylem vessels are used for the upward, right? So xylem vessels are gonna get water flowing upward here. Remember, it's like a drinking straw. And how this is gonna work is like you're gonna have the molecules, the water molecules gonna be connected to each other like this. Imagine each circle here represents a water molecule, and they're gonna experience two things. These two things are called cohesion and adhesion. Cohesion means that they have the ability to stick to each other. Adhesion means they have the ability to stick to other things easily. So in the case here, they can easily stick to the xylem vessel walls easily, and they can move upward like that. So that encourages what we call capillarity. So because they're also very narrow. I'll give you like a example of this, right? In India and some other places in the world, not to disparage any one type of faith, not saying this, but just sometimes the statues that they have, they could offer milk to them, and there's a tiny hole like where the mouth does be, and when they put the milk near to the mouth, right, it appears like the statue is drinking the milk. There's videos of it, and a lot of people, well, you know, if you don't know about capillarity and thing, you would tend to think, well, that's the spirit of the deity drinking the milk, right? I mean, it's an offering, so the sentiment is there, of course, but that is due to capillarity, because what's happening now is that because the orifice for the mouth is so small and it's kind of like a tube, what happens is that the pressure of the room, of the atmospheric pressure, pushes down on the fluid, and it actually goes up into the little mouth area, and it appears like it's drinking. So it's like a very, very tiny straw, and that there, because the molecules stick to each other and they stick to the surface, and the pressure causes it to go up. That is actually what makes it look like it's drinking. That's called capillarity. You can do the same thing with a very, very, very thin straw with some water, for example. So anyway, so these are connected because when you have a water molecule, if you remember now, water molecules have two hydrogens connected to oxygen at a certain kind of angle, and these hydrogens have a slightly positive charge and the oxygens have a slightly negative charge. So of course the hydrogen of another one could be bonded to this oxygen here and vice versa. Capillary tube in ink is our next good example. So xylem vessels are used for the upward unidirectional, and that means it goes in one direction, transport of water and dissolve inorganic ions from roots to leaves. So this would transport the water and any dissolved ions from the roots that we just saw, from the cortex, the epic cape examination pain, from the epidermis, the cortex, the endodermis, the pericycle, the xylem, and this will transport all the way up to the leaves. So these are made up of a dead material called lignin, and lignin is used for structural support. It's kind of like cartilage, but cartilage is alive. Lignin is a very dense, rigid, dead material that keeps the xylem upright, keeps it from collapsing. Now you'll notice here that the xylem has these little pits, right? And I'll circle some of them. So you'll see that there's these little pits. These little pits in the wall allow flow of water to and from adjacent cells. So some water, for example, could flow here. Some water could flow here, and so on. Otherwise, the xylem is mostly uninterrupted. So xylem vessels are hollow, they are continuous, and they don't have any end walls. I said they're uninterrupted. It's not like the phloem that has sieve plates and has sieve pores and things like that. This is like one tube, one drinking straw thing going right up. So you should know, what you should know about the xylem function is, and the structure is, that you should know at least four different structure to function things, right? So you should know that it's made out of lignin. The polymer made out of dead polymer called lignin has pits in the walls. It is hollow, and it is continuous, or it is uninterrupted. That's four things right there. Forget what I was gonna say. Right. This is common multiple choice, but there's different types of lignin. There's reticulated, there's spiral, and there's annular. The one that is mainly common is annular. Annular just means ring-like. So annular is like the ones that look like little rings inside there. So, right? Annular lignin. It's kind of like, again, it's kind of like the trachea. It's kind of like cartilage. All right. So a scent of water in the xylem. Basically the same things I was just talking about. So water molecules easily bond with each other. And I said this is called cohesion, but they also bond to the narrow xylem walls. This is called adhesion. And this facilitates capillary action, right? Or capillarity. So this is where, say, these molecules here that they can link to each other. And this is cohesion, right? Where they could link to each other. But they could also easily link to the wall. This would keep them from falling back down the xylem. They would have to flow against the force of gravity. Yeah? So transpirational pull, what is that? So this occurs when the water molecules exit the stomata. So here we have the leaf, and here we have the stoma. So there's the stoma here. Right? So you see where this pointing here, there's the stoma. So what's happening here now is that imagine like somebody's pulling on a chain of water molecules over here. So when they pull on this chain of water, so imagine like there's a force whenever one of these bonds break. There's a slight force that tugs the water out. Thank you, Bridget. I'll take a look at it tonight. So you're going to see every time you give this a little tug, the water molecules get pulled up this chain. Again, it's kind of like you're sucking on a straw or you're pulling on a little chain. So every time you pull on a little chain link here, a little bit of it comes up. So that's called transpirational pull. So this occurs when the force, due to the force of the water molecules exit the stomata, this pulls the transpiration stream higher and higher. This is the transpiration stream here. And we talk about root pressure builds up in the endodermis as inorganic ions are actively transported into the xylem. This increases osmotic potential and allows more water to flow more rapidly into the xylem from the endodermis. Alright, so the next part here, sorry about that. The next part here is measuring transpiration rates. And this is actually the last part of transpiration. So it's a very short topic. Measuring transpiration rates. So you use something called a potometer. This is a potometer here. Now, I don't think you all are getting a potometer question for your paper 2. The reason being is that the paper 3, which is the alternative to the SBA, they have transpiration there too, water uptake. Typically, when it comes in paper 3 is a potometer, so who knows? I don't know. All is still solid. So this is what happening here. We're going to have a plant or a shoot, and we're going to cut the shoot off of the roots here, right? Like off right where the roots are. And so that the xylem is exposed. So you're going to have a cut shoot with the exposed xylem securely fitted. And we'll have like a little reservoir of water, and then we'll have a capillary tube, a very, very thin tube. As Rajiv said, they're similar to chromatography. You dip it, it's going to take water up periodically due to the pressure in the room. It's going to push that water into this tube like that there, right? And what usually you'll have, and it's usually a big mess, I don't know if all you actually did the experiment in school, but sometimes the experiment does be a kind of mess because what you have to do is that somehow you have to make sure and get an air bubble in there. Sometimes they use a little bit of a dye, like hydrophobic dye in there. But that's the easiest thing is to use an air bubble. So that air bubble there will be located along this scale. The scale kind of looks like a ruler. For paper 3, they might ask you to draw the setup. Paper 2, you usually just have to label it. Or sometimes they ask you where the plant does be. Of course, the plant is on the end here where it have this rubber ball. So an air bubble is introduced, right here, and this will be somewhere. This will be it don't have to be on either end of the volume. You just have to record the position where it is. So for example, if this is 3 centimeters, pride one for water. The reservoir is there to reset the bubble, typically. So like you let you let a little bit of water flow out, and you could allow the bubble to flow to the end if you want. It's kind of like to adjust the position of the bubble. I don't know, y'all actually did the experiment in school? Let me make sure it don't have any other reason. But I know it's like to reset. Yeah. The function of the reservoir is to allow the air bubble to travel back to the start of the measure. So it's just there to when if you was to open the tap, some water would flow here, and you could actually get the air bubble to start here. That doing that practically, there's be a huge mess. Because sometimes the bubble does just go on, and you can't get back. So typically you could just start with wherever position it's in, and then measure it from there. Yeah, you could use a syringe to introduce it. It depends on how it is introduced. Let me see if I could find how it is. But I know you could use a syringe. But let me see if it have a next if I have a next way to do it. I'll Google and all here. Admitting it. But typically is use a syringe, and you could put it by the reservoir, and you could pass stopper and pour again, so you get a bubble. It is actually really difficult to get. But don't worry, all you don't actually perform the experiment for the exam, but don't ask that. So an air bubble is introduced, and the starting position was 3 centimeters, right? That you couldn't get all the way to the end. As transpiration, yeah, I know when it was being done, we tried dye worked better, but the bubble was kind of tough. Better to watch a video on it and done, yes. As transpiration occurs in the chute, of course the air bubble is going to move towards the xylem, because it's drinking water, it's sucking up water into the xylem, it's absorbing it through osmosis. So after a fixed amount of time, the final position of the bubble is marked and a distance is measured. So let me say that the bubble reached here, for example, and let me just say that on this scale this is 6 centimeters. I just making up a thing, I just saying. So of course where the bubble would have moved a total distance of 6 minus 3, which is 3 centimeters. So the rate of transpiration can be determined by measuring. You can think of it as the speed. We have a little physics in here. The speed will be the change in distance, or the displacement, over the change in time that would have occurred. And that would be the speed of the bubble, and you could use that as a way to measure the rate of transpiration. And you would do this for multiple measurements, which is hell, but you can use it for multiple measurements over for about five trials, and then you could plot the graph, and you could find an average, and that will give you the average rate or the average speed in centimeters per second or millimeters per second, depends on the kind of scale that you're using. So nothing terribly difficult, but let me show you here a question with it. So let me open up a question with it so you could see. And this would be, I think, June 2015. Let me just see how this is showing. Yeah. So this is 2015, and I'll go send all of these files after. Right. So there's like an example of how this come. Figure 2 is a diagram showing of a simple photometer which is used to investigate the rate of transpiration of leafy shoots. They say on figure 2, use an arrow labeled X to indicate the position which the leafy shoot is placed. This will of course be on the end here, to the left of the reservoir. Here you have the capillary tube, the ruler, the air bubble, I guess the next reservoir for where the water will be taken up from. And they say outline the key steps for determining rate of transpiration using the photometer shown. So this is 3 marks, so you don't need it. So usually they'll say details of setting up the apparatus are not required. So it's just really how, after you set this up, how you get your readings. So it'd be like measure the distance traveled by the bubble in a given time interval. Well, I should have here record the starting position and then measure the distance traveled, but same thing, it's 3 marks. Take readings several times for the duration of the experiment. So you could do it several trials, but you could also do it over a period of time. So for example, if the bubble is here, and every 2 seconds or every 5 seconds or every 3 seconds you measure the position. So you say, okay, well here it at 1 centimeter. 5 seconds later it at 2.5 centimeters. 5 seconds later it at 5.1 centimeters. 5 seconds later it at 7.6 and so on, right? So you could do it like that. So so instead of repeating the experiment multiple times, you can just simply do it once, but take like 5 measurements. Remember the minimum is usually like 5, right? So you can actually specify take readings 5 times during the duration, but sometimes it mightn't be 5 because this experiment can go pretty quickly. Calculate the average reading. The rate of transpiration will be obtained from the average. Oh, go the butts reach. Where the moderators on them. Alright, that's not it. Yeah. I tell you all this, so every stream I had 4 streams so far, all kind of different, so I have bots for all. And they show up at like a certain point. It's like somebody's be like, what's Spurwin streaming? Spurwin streaming, Lewis sends he students looking for a cheap one, a cheap time. I don't know what is going on. Could you move the light source like 3 centimeters each time? I don't know what you mean by the light source. But in this case here, because the experiment kind of tedious, you could just measure the change in distance and the change in time and things like that. So the next question was this, or this one just dealing with the xylem with reference to 4 structures. I'll send all that after. It had a next photometer question, you know. And let me just show you that one and then we could take a break. I can't find that question, huh? But I'm going to send all the papers and all they go see it. So, yeah, so that wraps up the first part of module 2. Transpiration. Oh, understand the light. So, no, you don't need to manipulate the light source. Just the presence of absence. Oh wait, it's that one part I forget to go over. We don't take the break. No break yet, eh? Just now. That one part I forget to go over here. Things that influence transpiration. So this is what you need to know as well. Things that influence rate of water ascent. I don't think I had that here. No, I didn't. So let me type it here. So factors that affect transpiration rates. I will link it in the chat. This is, well, one. Hey, what are we on here? Light intensity, which, because it causes stomata to open, it causes stomatal aperture to increase. Basically, stomata open more. We have temperature. Same thing, it can influence the open of stomata. 3 would be wind speed, which increases diffusion of water vapor out of the leaf. And then we have humidity, which decreases rates of transpiration. So the reason for that is, and I'm going to just give a quick explanation here. The reason for that is, imagine if we had a leaf and the type of plant would be one too. If it has sunken stomata, if they have pits, well, that's the same as sunken stomata. If there are little hairs and things, yeah, that could count too. The size of the broadness of the leaf could count as well. So imagine here, there's a leaf. I don't have any reason to make a Discord because this is not what others do. Anyway, so let me imagine now that the air is saturated out here, right? So we have all these water molecules here. There's water vapor, right? All these water molecules are already outside. So this air is saturated. So it has a high humidity. So you have to remember now that this would limit how much water vapor would come out, because here would have a high concentration of it would lower the rate of diffusion of the water vapor. If wind speed becomes too strong, maybe in an extreme it could, but typically if there's wind speed now, we have this water vapor here, right? And let me say that this is wind speed. There are wind now, and the wind woof, the wind blow away all our water vapor. Right? The wind would blow away all of this. Whoa, water vapor go on. Water vapor go away. So then more water vapor could come out. More water vapor could come out. And when this come out, the wind come again, blow that away, and then more could come out. So, another difference here, I don't think you have to know this. Somebody could confirm for me, but the stomata, when the stomata, what influences the stomata to open? When the stomata open, right? They open because water flow in, they get more turgid. And this is because sunlight, the guard cells get turgid. And this is because this is the this is the stomatal aperture, basically the stomatal opening, and this is the guard cell, and this is turgid. And basically what happens here is that light intensity influences potassium carriers to open. So when those potassium carriers open, right, what happens here now is that we get an influx of potassium in here. potassium ions. Right, so you get influx here. And remember what this would do is that this would make the cells, the K plus ions, make the cells more hypertonic. And as a result now, what's going to happen is that water is going to flow in to these cells, because remember it flows from a high water potential to a lower one. Right, high water potential to a lower, and the water flows in, and this becomes more turgid. And as a result now, the water vapor is able to, stream out of here. Transpiration could increase. I don't know if I'll get to learn that. Yeah, sure. But yeah. But light intensity influences potassium carriers to open. It basically, they kind of voltage activated. They're activated by the energy, the solar energy. So potassium ions make the cells more hypertonic. This allows water to flow in. I know for that. Anyhow. So let me link for you the past papers, so you can take a look at it while we take a break. And I'll put in the questions here that you should look at. Just now, eh? Lord. So they could screenshot this if you want. And me and the others go fit in. I'm trying to see if I can make the others fit. I don't know how to use a computer. Alright, Oli, see you all now. going on, everybody? Lower this volume. We back. I just typing out some predictions here for all you. No, it's 3 questions. 3 questions, each one worth 30 marks. But you could think of each question as like 2 questions, because one of them does just be like a real long kind of 12 to 15 mark question. The possible drawings could be the apoplast and symplast pathways, like to draw the draw like 6 cells or draw a group of cells and show that. It could also be the histology of the artery tissue, which I'll go through here today, if it have an electron micrograph, but otherwise it don't really have any other drawings, because the other drawing, I guess, is like the cross section of the leaf, which is easy. That's Form 5 stuff. But the only possible drawings is those: the cross section of the leaf, the arterial tissue, like a section through it. I don't think they'll ask you to draw the heart. Of course, they could have graphs, something like oxygen dissociation and hemoglobin saturation. The phagocytosis, that could be one, or blood cells. But those are easy. And then again, you have the apoplast and plasting, which they kind of like to bring, but I don't know. Right. So all I could screenshot this. These are my predictions. I don't know if they're worth anything, but this is, I'll tell you like how I came up with this. This is from observing 8-9 years of past papers, just trying to predict a pattern in which it would come. But you know, the papers kind of changed recently. Monoclonal antibodies came last year, so I have a feeling like they won't. They might, if it come, it'll come small. But it come kind of big last year. The graph I think that might come might be the disease one, or it might relate to transpiration. That might be a grafting. If not that, then something with oxygen dissociation and partial pressure and things like that. I think your long questions, I think your big 12 mark things going to be ecosystem stability, or in situ versus ex situ. I feel like that's going to be a big one. That's for mod 1. I feel for mod 2, I feel like your big question going to be on cardiac cycle, about sinoatrial node and depolarization and ventricular systole, and I think that is probably what will be a big question for mod 2, like your 8 mark 10 mark thing. Module 3, your big question might be the disease one. Like giving categories of disease and things, but it's different. No, not differences in module, the differences in the papers, because before I believe before 2019, I could be wrong, but before 2019, they had 6 questions, now they have 3, so it's a little more difficult to predict. So yeah, but anyway, let me get back started. Take a screenshots of this. Let me save the picture somewhere. So that's all, yeah. If you lose it, I can just show it back. Yeah, differences in cyclic versus non-cyclic could be a big one too, because I say that's a whole book by itself. Alright, take a screenshot, so counting down 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. There we go. Alright. So far we finished 3 topics. We have 3 more to go. One of them is incredibly short, which is health and disease. That is kinda, you have to use your common sense in observing trends and patterns and things like that, but yeah. So let me jump into this, a kind of big one and a kind of tough one, in my opinion. I don't know about all you, but a sort of tough one, which is circulation, the circulatory system. And this mainly we focus here on the mammalian heart. It's kind of telling that they put transpiration and circulatory system in one. You never know if they'll try to combine the two and ask you to compare plant circulation and humans. I don't think so, but because I don't think that any syllabus, but it's CXC, you never know. So anyway, let me get into some basics here. Alright. So first we have the circulatory system. Basics. The human circulatory system comprises of the following. We have the heart, which is a pump, blood vessels, which are arteries, veins, capillaries, arterioles, and venules, right? So arteries carry blood away from the heart, veins carry blood towards the heart or into the heart. And how you remember it is A far away, arteries carry blood away from the heart, and veins carry blood into the heart or towards the heart. Think of vein in. Veins have valves, of course. This is because they have a lower blood pressure than arteries. So capillaries connect arteries to veins, and basically they connect oxygenated blood flow to deoxygenated blood flow. And capillaries, the word capillare is Latin for hair, because capillaries are very thin. In fact, they're thinner than hair. So when they form a network, kind of like a mesh, and they typically surround body cells, which allows rapid diffusion because they have a very thin layer of tissue surrounding them, very thin endothelium that allows rapid diffusion of things like oxygen, carbon dioxide, and they're typically lined with carriers for things like glucose. Now, arterioles are basically small arteries and venules are small veins. You all will know this from Form 5 already. The main transport medium is of course blood, which the main fluid in that is called blood plasma. Blood has a lot of soluble material in it, such as materials such as fibrinogen for blood clotting. It has platelets are insoluble, but it has glucose, amino acids, urea, and then you have your larger molecules like leukocytes, erythrocytes, there's red blood cells, thrombocytes, which are platelets, and different types of leukocytes like lymphocytes and phagocytes and things like that, right? So all of these are transported by the blood, and all of these have their own function. So of course, only complex organisms with a low surface area to volume ratio would require such a circulatory system, like mammals, for example. Protozoa and prokaryotes would just use simple diffusion. Blood flows in a unidirectional manner through the heart and the body, which means it just flows one way, not backwards, and valves prevent the backflow and synchronize blood flow. So here we have the heart, the human heart, and easy way if you tend to forget it, you just have to remember Lord, right? And this stands for left oxygenated, right deoxygenated. So this is the left side of the heart, and this is the right. What's Chopin is talking about, boy? There's the right side of the heart, and there's the left side of the heart. You have to remember again, the heart is facing you. So the right side is deoxygenated, the left side is oxygenated. Easy to remember. And remember, any blood that is flowing into the heart is brought in by veins, because vein in. And any blood that is carried out of the heart is transported out by arteries. So remember, A far away, arteries away. Easy. Well, you should know that already. So this is a vein here. There's a vein. There's a vein. There's a vein. There's an artery. There's an artery. Arteries carry away. Veins carry in. All you know this, man. So these here are valves. We have 4 valves here. 1, 2, 3, 4. Right, so we have 4 valves. These valves have names, and I'll put up a diagram with the names here. But if you remember your stuff body heart, you have to remember that this blood vessel here, that this one is the pulmonary artery. So this is called the pulmonary valve or the pulmonic valve, whichever one. It could be called either one. And this one here, remember, this is the aorta, which is the main artery. So this is called the aortic valve. Yeah. So these here, typically, they just call them semilunar valves. And remember, these prevent backflow of blood. They basically open and close when systole and diastole occurs. These ones here, which are valves 1 and valve 4, this valve 1 here, this is called the tricuspid valve, and this separates these 2 chambers here. So this chamber on top here, this is called an atrium. So this is called the right atrium. So let's put in RA here. So this is the right atrium. The word atrium, when a building has an atrium, the atrium is kind of like a wide open area that's typically near the top of a building, and they just call it an atrium. So in this case here, they call it an atrium. And this here is called the right ventricle. So the tricuspid valve separates the right atrium from the right ventricle. So these are actually called atrioventricular valves. So valves 1 and 4 are called atrioventricular valves, so you could simply call them AV valves. Valves 2 and 3, as I said, are called semilunar valves, right? The pulmonary valve or the pulmonic valve and the aortic valve, same one. So 1 is the tricuspid and 4 is the bicuspid valve. So this is the left atrium, remember the atrium on top, and this is the left ventricle down here. The left ventricle is actually quite notable for having very, very thick cardiac tissue, very dense cardiac tissue. And if that gets worn out, what happens here is that if this is not strong enough, you have to remember that this muscle here has to be strong to pump with a adequate blood pressure and stroke volume to send this blood to the aorta so it can travel to the body cells. So that's why, you know, your heartbeat, people just say your heart is on your left side, right? So say that your heart is on your left side, but really your heart is in the middle and is in a sac called a pericardial sac, right? And the bicuspid valve is also called the mitral valve. Yeah, you can call it either one. Hey, if we go over 10, we go over 10. We're gonna see where we finish. So remember, again, the ones labeled 1 and 4 are called AV valves, atrioventricular valves, and 2 and 3 are called semilunar valves. Yeah, so I was saying if the muscle wears away here in the left ventricle, the person would have a harder time getting blood going to the body and going to the extremities. So they'll feel numbness in some parts of the body, they'll feel a little fatigue. The heart would have to beat faster, in order to get more oxygenated blood around. So let me take a closer look at the heart now. So again, this is the same things I was going through. So once again, we have the vena cava. So we have the superior vena cava and the inferior vena cava. The superior vena cava brings blood in from the top part of the body, from your head, your neck, your shoulders, and so on. The inferior vena cava brings it back from your lower part of your body, your abdomen, your legs, and so on. So these are veins. So they bring blood into the heart from a vein in, and these carry the oxygenated blood from the body cells into the heart. Next, we have the aorta, which is the main artery in the body, carries oxygenated blood from the heart to the body cells. We know that already. Then we have the pulmonary artery, or pulmonary arteries. These carry the oxygenated blood away from the heart to the lungs so that they can get oxygen again. All this you should be familiar with. The pulmonary veins, which is going to bring blood back in from the lungs, right, into the heart. Then we have valves, and again we have the tricuspid valve, the mitral valve or the bicuspid. These are atrioventricular valves or AV valves. Then we have the semilunar valves, these two here. So this is called the pulmonic valve or the pulmonary valve and the aortic. I don't think there's really asking that. Next, Rama said that this is the right side, and the right side is the oxygenated load, left oxygenated, right deoxygenated. And this is separated by a layer of tissue with very dense collagen. This is called a septum. The word septum just means like separating tissue. So like, you know, the thing that is separated two nostrils, that's also called a septum. And I said the left ventricle is the muscle dense region of the heart, where people say that your heart is on your left side is actually in the center, but because the left ventricle has so much muscle in it, the heartbeat actually feels like it's on the left. Well, the heartbeat is on the left, but it feels like your heart is on the left. This is because the left ventricle is so densely packed with cardiac muscle. So what is systole and what is diastole? So whenever your heart beats, there's what we call lub and dub. Sometimes it's labeled as that, the lub dub, right? And so it has two thumps. And this is basically due to systole and diastole. So what is systole and what is diastole? So systole is this. I like to think of it like, I used to remember it like this. I used to remember systole was like squeeze, right? So it's like squeezing the ventricles, right? I remember systole as like, imagine you're holding the heart and you're squeezing it. And diastole, I don't know, I just remember diastole is just the opposite. So diastole is like, think of it like relaxation of the chambers. So let me start here with systole, and we'll get a little more in depth with it just now. So systole is the contraction of the atria or ventricles, and this causes atrioventricular blood flow. So, or causes blood to flow out of the heart and into the arteries. So think of systole as stress or squeezing blood out. Think as D in the diastole as de-stressing. So imagine like you're giving down here a good squeeze. You're giving it good squeeze. D for downtime, D for dilate, all are real good. Or like you come up with all your own thing. So what happens here is that during systole, blood is squeezed out of the heart, right? And it's sort of pushed out of the heart. So these parts here, this contracts. And this here allows blood to go out of the heart. It's like a squeeze, and then it's like blood flows out. So causes blood to flow out of the heart. And so again, the heart is under like some kind of muscular stress because it's contracting. Diastole, on the other hand, is where these atria and ventricles would allow blood to flow into the heart to refill the chambers. So now one thing you have to take note of here is that when systole occurs, well, ventricular systole, I should say. When ventricular systole occurs, you have to remember that these valves here would close. These atrioventricular valves are going to close. And these semilunar valves are going to open. Because if down here gets a good squeeze, you don't want the blood to end up flowing back up here. Because remember, blood flow has to be unidirectional. So when one set of valves closes, all right, the next set has to open. So these valves here would close, and these valves here would open. So all can be open at the same time. Well, I mean, they could if you have a disease, but typically for your healthy human, all cannot be open at the same time. So this would allow blood to flow out like this. So same way, if blood is refilling the heart, right? And let me say down here is getting a good squeeze. This is during when the heart is being refilled with blood. You don't want here to be squeezed and blood from down here gets pushed back up here. Now, so diastole is when blood is allowed to flow back into the heart to refill the chambers. Again, stressing and de-stressing. So, hold on, eh? Oh, I thought I had a little more about it here. I see that physics. If you thought physics paper is hard, you real going through it. I watch that paper. That paper, I eat that paper. Okay. So the sinoatrial node and the atrioventricular node. I thought I had the next thing with this here. Anyway, that's okay. That's not all. I'm just looking for something. Okay. I have it here. That's later down the road. So we'll come back to systole and diastole so that we could separate it into atrial systole, ventricular systole, and diastole, right? And then come to like different associated blood pressures with it. So we have the sinoatrial node, and this is called the SAN, and the atrioventricular node, which is called the AVN. So this here, this is called the sinoatrial node. And this one in the middle here is called the atrioventricular node. So guess why it is called the atrioventricular node? Because it's between the atria and the ventricles. See how easy it is? So the sinoatrial node, I don't know what they call it. I actually don't know why they call it sino. See if anybody know. I know sino is usually related to China. Like Sinopharm. Or the Sino-Japanese wars. I don't know, like a Chinese discovery. I have no idea. See what the prefix Sino stand for. Anyhow, so the sinoatrial node, which is typically also called the pacemaker of the heart. So this is basically a group of cells that are here, right? So remember, this is the sinoatrial node, and this is in the, if you remember which chamber this is in the right atrium. Right, so this is in the right atrium. So this is located in the right atrium, and this is the site of the initiation of the cardiac cycle. The cardiac cycle is basically, think of it as a rounds of systole and diastole, right? Which is allowing blood to pump out and being refilled. Lub dub, yeah. Sino means right? Okay. So what mean left then? I feel like I should have known that one. I know Sino like typically relates like Chinese things. So I actually don't know. I never look up the etymology of it. So the sinoatrial node's muscle contracts. So remember, there's the muscle at the right atrium, right? And what this would do, so you know people have artificial pacemakers, and what the pacemaker of the heart does is that it regulates the heartbeat, but it initiates the cardiac cycle. But it also helps to regulate the pace of the heartbeat. And if you know what, when somebody has an irregular heartbeat, you say that they have an arrhythmia, right? So it makes sure that the person doesn't have an arrhythmia. And when the person's heart beats fast, it's called a tachycardia. right? Yeah, I don't think you need to know that for this, but that's just like terms you might hear like later on if you choose to further in bio. So anyway, so the sinoatrial nodes muscle contract. So there's the muscles of the right atrium here, right, on top here. And this is going to produce an electrical impulse. Sino means ringing of a bell. Well, I find that cool. So you can like, hey, a bell ringing here earlier. A bell ringing, it's time for the heartbeat. So what this does now is that this is going to initiate an electrical impulse. So it's going to start a whole depolarization process. If I start with this whole explaining that, we got done 11 o'clock earlier, so I go bother with that. But if all just basically remember what a depolarization is, you remember that a neuron has a resting potential of about negative 70 millivolts. And when there's a stimulus, like a touch or something, like some kind of response to something, the membrane gets depolarized, hyperpolarized, and well, depolarized, repolarized, hyperpolarized, and so on, right? And basically what that does is that it creates an imbalance in the amount of charges inside and out of the neuron. That there then propagates an action potential. None of this you have to explain for this, by the way, but all of that propagates an action potential, which then sends an electrical signal through nerves or muscles to receptors and things like that, right? So you don't need to explain none of that when you talk about cardiac cycle, because that's part of a different thing. Sino can be begin songs like that's that song like the thing. So the sinoatrial, sino could relate to sinus too, you know. Who knows? I'll look it up eventually. I exactly like to know where these words come from, because it has actually helped. Well, now I know, but back then it actually used to help me remember a little better, because sometimes the words in biology just seem real random. But like the words of like the bones and things, they seem real random until you look up what they actually mean. But sometimes it's help you remember. Anyway, so this muscle here contracts and it sends a electrical impulse which propagates through the atria muscles, causing them to contract. So these muscles here now, right, these here are gonna contract, and this is gonna pump blood, right? So remember, these are gonna contract and these are gonna pump blood down from the atria to the ventricles. But this impulse now, right, and let me see if I could, let me draw this. Let me switch to my MS Paint there earlier. Right, so let me switch to MS Paint. Oh shucks, yes now. So you could get some different colors here. So the signal gonna start here, and I'm drawing it in yellow. Right, so the signal gonna start there, and this is kind of gonna propagate through all these muscles here in a way, right? So you're gonna cause these muscles here to contract. So when they contract now, this is gonna pump blood down here. Right, so you're gonna pump blood down here, and while that is happening, the signal is traveling to this node here. So this node is called the atrioventricular node, and again, it only call that because it's between the atria and the ventricles, right? It's not hard to remember, AVN. Now, what this would do now is that this would relay the, remember the first one, the sand in the beach. No, not the sand in the beach, the sinoatrial node. This here causes the atria to contract, eh? Shucks, yes now. This here causes the atria to contract. So this here propagate through all these muscles, send a depolarization through all these muscles here. So now I join it in orange now. Not orange, I'm gonna draw it in green. So I join it in green now. When it hit this point here, it's kind of like a relay point. What it's going to do now is that this is gonna send the electrical signal, gonna help depolarize all these points here in the ventricles. So all this ventricular muscle. CXC spy boy, you take care of some butts from the other day. Hope everything going good. So when this electrical impulse gets sent down here, this causes the ventricles to contract. So this will cause this to contract, and it will cause this to contract. Now you have to remember now that this has a little lag point in it. So electrical impulses travel really quickly. So it is totally possible that the electrical impulse could actually start here at the sinoatrial node, cause these atria to contract, skip to the AVN, get relayed, depolarizations come down here, ventricles contract too. And you're going to end up with all four contracting. That's not what you want. So it have a little lag time between the sinoatrial node and the atrioventricular node. Right? Now coming back to this. So once again, just to go through it one more time. To go through this one more time. And then what I'll do is I'll relate this to ventricular systole and things like that, right? So one more time. We have the sinoatrial node, the SAN. This here, think of it like a bundle of cells that's going to initiate a nervous impulse. It's going to initiate the cardiac cycle. This is going to send signals all through muscles in the atria. All here. And this is going to cause it to contract. This will cause blood to flow from the atria to the ventricles. Yeah? Now, at the same time now, what is happening now is that the signal, the depolarization, the action potential is being sent to the atrioventricular node. That's we boy here. So the atrioventricular node now is going to relay those depolarizations, that series of depolarizations, to the ventricular muscles. It's going to cause those to contract. And that there would result in ventricular systole. And this process would repeat. And thus you have a lub-dub, lub-dub, lub-dub sound, right? Don't worry if you don't fully get it right now. When I get into the three main steps of the cardiac cycle, it will make a little more sense. But that's what the sinoatrial node and what the atrioventricular node do. One of them initiate, one of them is a relay point that continues the depolarizations along the muscles. Now, let me come here. Right, again into the bundle of his and thing now, right? The bundle of his. That does sound like, I don't know. Yeah, the sound is the valves. So again into that now. Don't worry. So again, that was just explaining what the sinoatria and the AVN is. Now again into the actual cycle. So the cardiac cycle refers to the sequence of events that comprises one heartbeat, right? So this first part here, this is atrial systole. So atrial systole is where the atria contract. Blood flows from the atria to the ventricles. This is where we have the sinoatrial node propagating the signals that is causing the atria to contract, right? Next, we have ventricular systole. That's this. This is where the ventricles are going to contract. This is where the atrioventricular valves are going to close. right? And blood is pushed from the ventricles to the aorta and pulmonary artery. Imagine you're squeezing the bottom of the heart and blood is flowing out the pulmonary artery and the aorta to go out of the heart, right? To go to the lungs and to go to the body cells. So this is where now the atrioventricular node, which is located here, this is going to propagate the polarizations and action potentials, or whatever you want to call it, to the ventricular tissue, ventricular cardiac tissue, and that's going to cause that to contract. Ventricular diastole is where both the atria, remember this de-stressing, where both the atria and ventricles relax. Blood flows into the heart from the veins, the semilunar valves would shut to prevent the outflow of blood. This is where de-stressing, this is where it has pumped out blood and now it's ready to accept new blood. Now, coming to these other parts with the fibers and the bundles and things like that, right? What exactly is happening here? So, you'll see that I have 5 parts labeled here. I have 1, 2, 3, 4, and 5. So we're going to take this step by step, what happens during the cardiac cycle. So remember, first, we have the sinoatrial node. That's here. So there's the sinoatrial node right here. And this is going to send out an electrical wave of excitation across the nerves in the atria. So this would be this atrium here, and remember this is the right atrium. So that's what's going on in 1. Sinoatrial node is here. Now, what's going to happen after that? The atria are going to contract. So this is going to contract here, this is going to contract here, and this is where blood is going to flow down here. Yeah. Now, as this is happening now, the atrioventricular node picks up the signal, that is where 3 is, and sends another wave to the ventricles. That's what you're seeing going on here. So all that we don't know. Now, there's a short delay so that in time, so that all the chambers don't contract at once. I guess now, just give me 1 second here. I'm just making sure something. Right. So the parts that actually receive the signal from the atrioventricular node, just making sure, these fibers here. These are basically groups of neurons that make up the electrical conduction system of the heart, right? And these are called bundles of His. And the other one, this be a kind of iffy kind of thing, because sometimes you might see it as Purkinje fibers, sometimes you might see it as like Purkinje fibers. I actually don't know, I don't know if it's nobody, but I actually not too sure what's the pronunciation of it. I just call it Purkinje fibers. But, or sometimes Purkinje fibers, depends on how you want to pronounce it. But basically, that's what's going on in 4. So these fibers here in 4, along with bundles of His, these are electrically excitable cells. So these are going to be depolarized, and these are going to conduct the signal from the atrioventricular node, remember, which was in 3. These would move the signal into branches in the septum. So these branches are called bundles of His. And these now kind of branch out to the right ventricle and left ventricle, and these are going to cause the ventricles to contract. So as I said, there's a slight delay with this, and I don't know if anybody could illuminate if there's actually any mechanism specific to cause this delay. But there's a slight delay between where the sinoatrial node propagates the depolarizations to the cause the atria to contract, and the Purkinje fibers and the bundles that propagate the signal from the atrioventricular node to the ventricles. I don't know if anybody, if all you learned it, if all you read it up, if there's any specific mechanism that actually causes the delay, because of course it cannot happen at the same time. Because to me it must have like some kind of tissue in there that's, or maybe the protein fibers, something about them, they unmyelinated or something like that. You know how myelinated axons could undergo saltatory conduction. They unmyelinated or something. I don't know if anybody know. Maybe you need to look it up, because I don't think you had to specify, but just out of pure curiosity. But it had to have something to cause the delay between the atrial systole and the ventricular systole, because as I say, the blood, all of them cannot contract at once, and because depolarizations happen so quickly, it had to have something. Myogenic. Well, the no, I know it delays its electricals, but as one, if it had a specific mechanism now, like if it had a chemical or a carrier or some kind of thing that is, I don't know. Because to me the thing don't say, hey, we had to wait, you know. We had to wait for the thing to done contract, you know. All these things just have like a little ligand or some kind of thing. I don't know. Let me don't stress all out. But that's pretty much the cardiac cycle, right? And we're getting to the graph just now. Do I have the graph here? The diameter. Okay, the diameter might make sense. Okay. That's a guess, or you're saying that? Or you read that? Preload and after that could be it too. Anyway, let me don't stress all out. I was just wondering if anybody did learn it in school. If I did learn it, I forget. I was thinking one was myelinated and one was unmyelinated or something. Anyway, let me don't stress all out with nervous system, nah? Alright, but coming to stress all out with cardiac cycle graph. You read it? Okay. Well, that makes sense then. Smaller diameter. Thank you very much. The rest all those stress, it was more for me to know. Anyway, so during ventricular systole, pressure has a steep increase. Okay, so this graph here, watch now. Let me hope this graph here come now. Might have to sacrifice something. Make sure the graph here come. So or they could sacrifice something, or they could pray. Yeah, we're doing parasympathetic nerves and thing. We're doing after. I wanna finish cardiac cycle first. When we're doing well, when we're doing responses to carbon dioxide, and we'll get into that. Anyway, so let me look at this graph here. So, as somebody pointed out there, it is the sound of the valves opening and closing that actually gives that lub-dub kind of sound. So, where the atrioventricular valves close, right? That is going to give it the lub sound. And where the semilunar valves close, that's going to give you the dub sound. Okay? Now, let me start here, and I have it nicely kind of segregated here so that you could see where's atrial systole, where's ventricular systole, And where is ventricular diastole? Nah, don't sacrifice the goat. Sacrifice a little sleep to learn it, yes? Alright, Ollie. So we're gonna take a little time with this one because the thing about this is barely common. It's more common in multiple choice, but you end up feeling wicked this year. Let me start here with atrial systole. Now remember what's atrial? Remember systole is like stress, is like squeeze. So this is where blood is being squeezed or being pushed from the atria to the ventricles. Well, the graph look like a mountain, but the knowledge to climb it is like a mountain too, but we're gonna do that now. So the atrial systole is this. You'll notice that we have three different curves here, right? You have atrial pressure, ventricular pressure, right? Atrial pressure, ventricular pressure, and aortic pressure. You'll notice that aortic pressure is always a little high because it's an artery, and arteries have high blood pressure, right? Because they have to take blood to the body cells, so blood is typically flowing very fast through them. So you notice that aortic pressure is usually kind of high. There's a big jump here, small dip here where the valves close and come in like that. Don't worry, I don't think you'll have to draw this graph from memory, but you'll have to know where. Talk low.
As the adage says, you give a poor man a fish and you feed him for a day. You teach him to fish. You give him no.
Alright, better now? I do that so I can fix my mic. Mic better? Let me go back to the graph. That's a little laughs. You finish the stupid graph. Alright. So I say aortic pressure. I was saying earlier. I saying it loud now. I saying it loud. Aortic pressure is typically high because arteries have a high blood pressure. Eh? So during ventricular systole, you'll notice that everything does take a kind of jump because this is where the heart being squeezed, right? It being stressed, and blood is flowing out of the ventricles and going to the body, right? Now, you notice that ventricular pressure, of course, takes a big jump here. That take a big jump. Aortic pressure is take a big jump because during these parts here, you have to remember here, this is where the blood getting yeet out of the heart and to the body cells. Yay! So, and remember here now, this is where the blood flowing down from the atria to the ventricles. It is going to line down there. It's just going down the road. right? So atrial systole, this is blood going down the road. Ventricular systole, this blood ready to bust out of here, right? So this is where going out of the heart into the body cells. That's why we have this big lob lunge dub dive here. Yeah, that's a good. What about lob lift? Hey, lob lift dub dive. That's sounding good. Now ventricular diastole. Now remember, this is where the heart taking a nice short vacation, a nice short holiday. Well, kind of holiday, not really. A nice short rest day. It not totally at rest, you know what I mean? And de-stressing. So this is where you're seeing most of the pressures drop here. So going down there, you see aortic pressure is dropped. You see this is where ventricular, you see during this part here, this is where ventricular pressure will drop. And this is where this other thing, atrial pressure will drop. So this is where it's nice and the distress and everything kind of going down. I have no idea if any of this helping because this graph, I'll tell you, this graph is kind of pressure, but you really, what the most it is really ask about this is to identify these three things: atrial systole, ventricular systole, ventricular diastole. I don't really find that hard. Okay, what is the pressure? What is the reason, sorry, for the rise in atrial pressure after the AV valve shut? Oh, this little thing here. This could be just due to this could be just due to what's you know when the valves close, it might have like a little rise in pressure, especially remember that whole chamber kind of getting pressure pushed against it. So even the atrium getting a little pressure against it. It's not much, but it's negligible. The atrial systole have to do with the mitral valve or the aortic valve. Atrial systole, the mitral valve would open and the aortic valve would close. Well, the atrioventricular valves would open. At the end of atrial systole, the atrioventricular valves would close. Let me type it out. Well, let me because I know this is a little. I know it could be kind of thing. But let me go through it one more time, right? Atrial systole. You have to remember here that the atrioventricular valves open. That's the tricuspid and the bicuspid or the mitral, right? These valves open. So this is causing blood to flow from the atria to the ventricles. Yeah? They're going down the road. They're going far. They're just going down the road. From the atrium to the ventricles. They're going down right there. They're going right there. So the pressure is not very high. They're just going down right from an atrium to the ventricle, right? That's atrial systole. It's kind of like you're squeezing the top of the heart. You're squeezing the atria and it's blood flowing down. So ventricular systole is like the big event. There's the big part. There's the earthquake. You know, there's the big pressure. There's the big pressure event. There's the Cape Unit 2 paper 2 exam. This basically is squeezing the bottom of the heart. Right? Remember, the ventricles will contract. The atrioventricular node sends signals to the ventricles, causes that to contract. You get huge rise in ventricular pressure, big jump in aortic pressure, smaller jump in atrial pressure because some blood kind of pushing against it. Nothing to talk about. And then it will slowly increase as it comes here. Now, as ventricular systole, as the blood kind of empties from the ventricles, the pressure takes a sharp drop. Yeah? And then semilunar valves close. Aortic pressure takes a drop. Everything takes a drop because it's time to take a nice little de-stress. the atrioventricular valves open again so that blood could flow from the atria to the ventricles. And during this point, the heart taking a little short rest, a little holiday, this diastole. This is just to allow blood to come back into the heart. So, what I can tell you, I talk enough about it, or you could read it, look it up, use a textbook, watch a video. I show it hard thing, but it don't really come. There's mostly just ask you like to label points like which one is the systole, which one is the diastole, which one is the valves opening, which part the valves closing. That's basically what is asked. So let me jump from cardiac cycle, but learn cardiac cycle. That could be a big one tomorrow. Let me jump from cardiac cycle to blood vessels. Let me ease up the pressure a little bit. Let me ease up the pressure a little bit. And come to something a little easier. So blood vessels, there are three main types. We have arteries, veins, and capillaries. So arteries, we have to be able to look at a micrograph and be able to tell what is an artery and what is a vein. So looking at this this micrograph here, we could easily tell the difference. The artery is a thick-walled vessel with small lumens. There's a lumen here, I'm marking a L. There's a lumen. The lumen is basically where the blood passes through, and this transport blood away from the heart. There are many layers of muscle cells that are called tunica layers. There's tunica media, tunica interna, and tunica externa. We'll get into that just now. And these consist of muscular tissue and collagen. Of course, collagen is a protein that helps with structural support and things like that. So it has multiple layers of muscular tissue and elastic tissue right here in the artery. So if you remember physics, pressure is force over area. And because the lumen has a smaller area, smaller diameter, cross-sectional area, the pressure is going to be higher inside the artery, right? The veins have thinner wall vessels with large lumens. This here is the vein. And you might be like, what, they're looking like a disease. But nah, there's a vein. Veins, they have larger lumens. There's a lumen here. And thinner wall vessels. So they're, oh, sorry, I said tunica interna. It's tunica intima, my bad. So they have the same three layers, but they are much thinner. You'll notice that the tunica media is where it has the main difference here. This basically just means the middle layer. Not too sure what tunica is Latin for. I don't know if it has something to do with a tunic or like a suit or something like that. But this part here, there's the tunica media. And this is where you have the main difference, that and the tunica externa. So where you would have a lot more muscular tissue and a lot more elastic tissue like collagen in the artery than the vein. This is why the vein does look like wonk, wonk, wonk. It look kind of floppy now. It ain't look too good. It ain't looking too good, but it does work. If you have no veins, you get dead. So let me do this. But the veins don't really have that kind of fixed round, like a kind of elliptical kind of shape. Instead, it looks like it is easily collapsible. That's because the blood pressure in veins, remember again, pressure is force over area, so a larger cross-sectional area means to have less pressure. Capillaries, I said, very narrow blood vessels that surround tissues and organs. It literally means coat, okay. And they have a single layer of endothelial cells. The butts don't come during the break, you know. Them butts just reach when I I don't know if it's biology and they feel people watching blood vessels and all kind of thing and they feeling that way. All your exam tomorrow, you can study that. Alright, so endothelial cells, they have a single layer of that facilitate rapid diffusion. Sometimes transport is done through vesicles via exo and endocytosis between the capillaries and the body cells. So these are just some characteristics, differences between arteries and veins. This actually didn't come in a while, differences between arteries and veins and to draw the tissue and thing. But nothing here is actually too different from what you studied in CSEC. Arteries carry blood away from the heart, veins carry towards the heart or into the heart. I remember artery A far away, vein iron in. Arteries are generally thicker. The tunica media is thicker than the tunica externa. And for veins, they are generally thinner. The tunica externa is thicker than the tunica media. Remember, the tunica media is like main muscle tissue and things like that. So the lumen diameter is smaller, and for veins, they are larger. Blood pressure thus for artery is higher, so you can actually feel a pulse in the artery. Veins, you cannot feel a pulse because the blood pressure is so low. And semilunar valves, arteries have no valves, so because the blood flow is so high, and veins have valves present because the blood flow is low, there's always liability or backflow, so that's why you have some valves there. Arteries are typically oxygenated. Blood carry oxygenated blood except the pulmonary artery, which is deoxygenated. Veins typically carry deoxygenated blood except pulmonary vein, which is oxygenated. Nothing too different. So this is something that could potentially come, which is the histology of arterial tissue. It has come already. And this is the only main, what we could say, in Unit 1, you had all these plan diagrams you had to do. Well, out of all the topics that all they get here, this is mainly the only plan diagram that you'd probably have to do, if you get. So this is a section, a transverse section of a sector of an artery, a carotid artery here. So the cells or the nuclei on the innermost layer or the so these are basically things that you want to show when you are drawing it, right? So if they say draw a section of arterial tissue, they'll probably give you a micrograph of it, and they will ask you to draw a section of it. So I'm actually going to show you a past paper with it here. Let us see which one it was by. Right, so this was June 2015. June 2015 looking like it has some things in it, but yeah, it harder to predict for keep. Alright, so I'm going to show you here. And this is the same diagram from this. It's not the same diagram from this, but so I don't think they'll ask you to draw the heart. See, second thing they'll ask you to draw heart. Sometimes, but not for Cape. Cape does more ask you about drawing micrograph to see one thing, because they assume you already have all your drawing skills from CSEC to draw organs and systems and all kind of thing. Now they're more focusing on your ability to replicate micrograph drawings and things like that. Anyway, so coming to this, this is the diagram that they gave for Cape. There's the diagram. And they had box A. And they said, Figure 3 is a student's plan drawing of a cross-section of a major mammalian artery showing the distribution of the tissue layers. And they had box A here. What they wanted you to do was to say, in the region highlighted by the box label A on figure 3, draw the cellular details of the tissue layers. And this was for 4 marks. And basically, you're supposed to draw it inside here. You're supposed to draw it, but I put it here. What the things you want to show, right? You want to show the endothelium, and the endothelium is actually nucleated. So this is because this is tissue here that has receptors and things like that. It has chemoreceptors to detect carbon dioxide. It has different receptors on it. So this endothelium here is nucleated. There is the different tunica layers. There's the tunica intima, media, and externa. So you have the elastic layer here, the elastic band that separates the endothelium from the tunica media. And this is the smooth muscle. Remember, smooth muscle is involuntary. You can't actually willfully contract it. And then you have the tunica externa and the adventitia, which is just the connective tissue. There's like the collagen and things like that. It's not hard really, but let me come back to this document here, and just to provide a checklist of where you should remember to draw in case it comes. Cells are nuclei in the innermost layer of the endothelium, right? So that is basically here. And this here is like, as is nucleated. So that is actually checked to see if you're drawing nuclei here. So even if you had to put a little dot, put that. Because they expect you to know that the endothelium or the lumen, the artery, has nuclei in it. The layers of elastic fibers beneath the endothelium. So where you have the endothelium, you will have these thick wavy lines, and these represent the elastic fibers. That's these here. These are the elastic membranes. The tunica media, right, beneath the elastic membrane. So this is the tunica media here. This includes the smooth muscle. This is also nucleated. Means it has nuclei. And it also has dense fibers and smooth muscle. The outer layer, which is the tunica externa, that's here. These have loose fibers, collagen tissue, and so on. Hopefully it will come, but you know, if you look at your textbook, you should see a better diagram of it. All right, so let me go on to blood cells now. I won't stick too long on blood cells because we have immunology, and immunology might get a little more into it. So red blood cells, or erythrocytes, or you know this already, the function is to bind oxygen to hemoglobin for transport and delivery to the cells. Oxygen combines with hemoglobin to form oxyhemoglobin, and then you know you have a biconcave structure. It doesn't have any major cell organelles, and yeah, so it has a biconcave shape, no major organelles, which means there more room for hemoglobin, small, flexible, things like that. Lymphocytes are a type of leukocyte or white blood cell, and these make antibodies. These B lymphocytes make antibodies. We'll get more into that immunology. It sends signals. These are called T helper cells or T helper lymphocytes, or they kill infected cells. These are called T killer cells or cytotoxic T killer cells. Again, we'll get more into that later. But how they look, how you can tell is that they have large round nuclei, and they typically have receptors on the surfaces to bind antigens. Again, we'll get more into that in immunology. Neutrophils have what you call a multi-lobe nucleus. So it's not a large round nucleus like the lymphocyte. So neutrophils engulf pathogens. They're able to present antigens to lymphocytes. We'll get into that in immunology. Macrophages are similar to neutrophils, but they're not as powerful. And they're typically more used to remove like dead material. There's like kind of like garbage men of the blood. They have a large irregular nucleus that's slightly folded, slightly granulated, granular site basically. Thrombocytes or platelets help form blood clots. They are not actually cells. They are actually smaller broken off or remnants of cells. And what these do is that they activate enzymes and clotting factors. And these trigger off a cascade of reactions that involves things like prothrombin, thrombin, thrombokinase. We don't need to know all that. But at least I don't think so. These things here would then convert fibrinogen to fibrin, and fibrin is insoluble and actually forms a mesh that will trap red blood cells to form a blood clot. And that's called hemostasis. I don't think I know that either. I know that from cells at work. If only what cells at work, you probably don't have to study no cells in biology. At least with immunology and things. Blood plasma, which is not a cell, this is the fluid. Transport soluble proteins, these are materials, nitrogenous waste, nutrients, etc. So there's a pale yellow fluid. So this is after it passes through a centrifuge. This is the plasma. This here is what is called the buffy coat. This is the leukocytes, and down here is your red blood cells. Okay, so you went through cardiac cycle. I never saw as only see inflammatory response come for paper one. We'll still go through it here because it's under immunology, but I don't think so. Nice. So, what is cardiac output? This is a topic that has come and has catch everybody off guard because when they're doing circulatory system, they just study heart, arteries, veins, capillaries, cardiac cycle, SAN, AVN, all kind of thing, and just forget all about cardiac output and so on. And then it has come, and people be like, "Oh, I never, I never really studied this." Anyway, so what is cardiac output? So cardiac output refers to the following, right? If you had to define it, it refers to the volume of blood, right? The volume of blood pumped out of the heart per minute. Okay? So that's your cardiac output. So the volume of the blood pumped out of the heart per minute. Basically, how much blood it could pump every minute. So what actually comprises this figure? Well, this is a combination of two things. Your heart rate, of course, you know your heart rate is how much cycles, how much beats it have per minute. The average is about 70 or so when when you're at rest. And the stroke volume. So the stroke volume is the amount of blood pumped per cycle, right? So the volume of blood pumped per cycle. So of course, if we multiply again, it's all in maths and biology come together. It will be too nice, but if we multiply the volume of blood pumped per cycle, right? And per cycle is like per cardiac cycle, of course, the atrial and ventricular systole and diastole. And you multiply that by the heart rate, you would get your cardiac output. It's as simple as that. Bring all the calculator. I don't know if all you allowed calculator. Who knows? Yeah, yes, you need calculator in case you have to do magnification and say, hey, if all you get a drawing, all you remember all you have to label that drawing and all you know that you're supposed to label the drawing, title the drawing, put magnification if it applicable, right? Remember the drawing itself, this be a little 2 marks and then putting title as a mark, putting magnification as a mark. People think the whole drawing is 4 marks or 5 marks. But the title and things sometimes carry half of the title and magnification and label lines and things sometimes carry half of the marks. Anyway, so the main factors that affect cardiac output is this. Adrenaline secretion and oxygen and CO2 concentrations. Right? Basically gaseous concentration in the blood. All of these here affect how the heart rate and affect the stroke volume. Now, there are other factors as well, but these are the two that we're going to focus on. Because the other factors is kind of focus on them in social and preventative medicine, which, are not common. This man know how long his finger is, yes? Man measure with his finger by inch and then converted with long division or something. Well, bring a calculator and you will not to do that. Men had vector diagrams in CSEC physics today and they forget the protractor. They're probably putting the two fingers together like to see what's 30 degrees. Anyway, so cardiac output. So this will then segue into oxygen dissociation and thing, right? But this we focusing on how does this affect cardiac output. Once again, you have to keep in mind what cardiac output is. So cardiac output is when you combine heart rate times stroke volume. Okay? How much times the heart beats per minute times what volume of blood is pumped out per cycle. I need to multiply. I don't know. I actually don't know what is the normal stroke volume. But if you find out what's the typical stroke volume, you multiply that by 70, which is your average heart rate, and you would get your cardiac output, which would tell you, how much volume of blood is pumped out per minute. So oxygen deficiency in cells or high CO2 concentrations in the blood are detected by chemoreceptors in the arteries. I shall say in the arteries, right? And 5 liters, well, 5 liters is 5 meters cubed, right? So 5 meters cubed by 70, that would be the cardiac output, but the average, I should say. So when there's a high CO2 concentration in the blood, this results in the release of nitric oxide, right? So what does that do? So nitric oxide is secreted. Let me make sure I get the name of this right. So nitric oxide secreted by, I believe it's just the endothelial cells. Yeah. So it's just the endothelial cells. So nitric oxide is secreted by the endothelial cells in the arteries. Yeah, I want to know 5 liters. I was like, there's a giant or 70 milliliters. Okay, so basically 70 ml by 70. And that's a cardiac output. What's that? For 4900? 4900 ml or something like that. That's basically what it is. So anyway, so what nitric oxide does, right? Nitric oxide dilates the arterioles, and it gets this is also released. I believe, I could be wrong. This is also released when you're sick. So when you're sick, nitric oxide is released, and I think as part of the inflammatory response, as part of one of the responses. And this allows the blood vessels to dilate. If you know, dilate means to get bigger. And this allows increased blood flow, and as a result, increased cardiac output. So when your oxygen levels low, or when your CO2 levels too high, this basically allows, it doesn't increase your heart rate, but it increases your stroke volume, because a higher volume of blood is able to pump. Nah, it's not mathematician, nah? It are people in this chat who much better in math, probably. So this dilates arterioles, right? And this increases stroke volume. Now, adrenaline now is released during scenarios of stress. All your people to keep bio tomorrow, against adrenaline, maybe chem, maybe pure maths, one of those, or intense physical stimulation, right? So basically, when your body is under some kind of stress for the past 3 years or yeah. So what this does is that the hormone adrenaline stimulates adrenoreceptors. That's basically receptors that respond to adrenaline, to stimulate, this is in the heart by the way, to stimulate the sinoatrial node and the atrioventricular node to increase the heart rate. So of course this would thus increase cardiac output. So let me recap what we get so far, right? So we have adrenaline. Adrenaline stimulates, adrenoreceptors stimulate SAN and AVN, and this AVGN and this ups the heart rate. The nitric oxide, and this is from low CO2 concentrations, chemoreceptors in arterial tissue, arterial endothelial tissue, dilates arteries, increasing stroke volume. So these two things combined, right? Just one of them alone would increase cardiac output, but imagine both of them combined. Both of them combined would be like a stacked effect, because you could either increase heart rate or you increase stroke volume. Increase both of them, heart rate, your cardiac output going to increase a lot. So these are the two that you mainly look at carbon dioxide concentrations, and adrenaline. Right. This has come as well. A lot of people get catch off guard with this. Blood pressure. So you have to know how to define blood pressure, how to define that, how to define pulse. A lot of people will be like, well, I know what's a pulse, but me know how to define it. And it's just like a little 1 mark thing. But blood pressure, to define it, this is the force of the circulating blood, and you have to say on the arteries or on the walls of the arteries, right? Not just the force of the circulating blood, but you have to say against or on the walls of the arteries to get a mark for it. Pulse is a force that can be felt against the arterial walls for heartbeat. So pulse is what you can actually feel. That's the singular force that you could feel, whether it's in your wrist or your neck or your heart or something. That is what you could feel. So that's the force that can be felt against these arterial walls per heartbeat. Of course, you check to see if somebody dead, if they have a pulse. If they don't have a pulse, most likely the arterial, at least the arterial blood not flowing, which probably the dead. You could use that too, cardiac output, systemic, but you could relate it like that too. Yeah, either one is fine. So, what could result in a higher blood pressure? Well, typically, if lumens have a reduced area, so for example, if you end up with atherosclerotic plaques developing here, right? And these would typically be yellow, of course. And these would be due to a buildup of cholesterol and things like that. Now, sometimes this could reduce the actual passage, the area, the passageway for the blood to flow. So this would result in a higher blood pressure unless the passage becomes really, really blocked, then it would lower it. But this is called atherosclerosis. Not going to linger on that too long because that's more social and preventative medicine. But eventually what will happen here is that if the entire passageway gets blocked, this is called a thrombus or coronary thrombosis if it happens in the heart, which then results in a myocardial infarction, which leads to a heart attack, right? So if a person has persistently high blood pressure, which means that the force of that of the circulating blood on the walls of the arteries and of course on the ventricles of the heart, if that force is too high, it would cause damage to the tissues and damage to the ventricle tissues and things like that. Eventually, the heart itself actually begins to dilate. If you Google dilated heart, you would actually see what it looks like when the ventricular tissue wears away. I don't know anything about IT. I never even do IT. You're like the 10th person who come in here asking for IT. I don't even know anything about IT. I don't even know how to use a computer. I never even do IT. I don't know the first thing about the syllabus. All I know is how to play game on the computer. Me, I know nothing else. Alright, so next thing here is blood pressure factors. And this is easy, I'll just read it. Intense exercise. And this, of course, stimulates the release of nitric oxide due to low oxygen concentrations. If you remember what that does, is it dilates arteries to allow increased blood flow, increased stroke volume. Cigarette smoking. Tobacco contains nicotine. Nicotine is a stimulant, and which it also constricts blood vessels, makes lumens smaller, also increases heart rate. Adrenaline, we explained that already, stimulates adrenal receptors to stimulate the sinoatrial node and AVN to increase heart rate. Atherosclerosis does literally what I just explained. Okay, so coming to our next important part here, hemoglobin. So hemoglobin is a protein in red blood cells with a quaternary structure. So if you remember, was a quaternary structure or a quaternary protein structure is basically when a protein structure has multiple tertiary structures mixed with secondary structures and prosthetic groups and things like that. The prosthetic group in this case in the hemoglobin being the heme group, which contains iron. Yeah. So each of the 4 subunits have heme groups. And I like to think of the hemoglobin. Imagine you have a hemoglobin car. Eh? You have a hemoglobin car. And the hemoglobin car, there's the hemoglobin car. And the hemoglobin car have 4, well, you only see the side of the car here, but it have 4 oxygen tires. Right? So it have 4 oxygen tires. And it can't have more than 4 oxygen tires, right? So I'll come back to the car just now. But each hemoglobin molecule can bind 4 oxygen molecules. So when this happens, the hemoglobin is now called oxyhemoglobin. So oxyhemoglobin, you'll see it when I write it. So I'll write hemoglobin as HB and oxyhemoglobin as HbO2. That just means that the hemoglobin has been saturated with oxygen. That means the hemoglobin car is driving with 4 oxygen tires. Yeah. So carbon dioxide could also bind to hemoglobin, but not as readily. And when carbon dioxide binds to it, you call it carb-aminohemoglobin or carb-aminohemoglobin, whichever one you want to say. But typically it's carb-aminohemoglobin. We're not going to focus on that too much, but oxygen binds to it more readily. So keep that in mind there. Now, we're gonna get accustomed with 2 terms here. The first term is dissociated, and the second term is affinity. Yeah, taxi for cooperative binding for real. So dissociated means the gas is released from the hemoglobin, such as when oxygen is released from the hemoglobin into the body cells. Affinity describes how readily the gas binds to the hemoglobin. Since oxygen binds more readily to carbon dioxide, it has a higher affinity. And carbon monoxide has an even higher affinity, and so can potentially fatally bind to hemoglobin. Alright, Ollie, so I need to take a little 5 minutes break now. I just have to take care of something here. So I'll be back in 5 minutes. Alright, y'all. Sorry about that. Did we start back? Oh yes, refreshed. Okay. Let me come back here. All I could believe all is sitting down here for like, well, all the other hour break, but like 6 hours and something. This thing going on, yes? All the real stamina. Okay. So before I get into this next part here with hemoglobin, right? Remember I said hemoglobin is a quaternary protein. Right. So remember hemoglobin is a quaternary protein. And think of it like a car with 4 oxygen wheels, right? And when it had them 4 oxygen wheels hooked onto it, of course it cannot attach more than 4 wheels, right? But it could lose wheels, but it can't attach more than 4. Probably our hemoglobin car is not the best example. But the only thing that could happen if you have 4 wheels is that somebody could come and thief your wheels. You know what I mean? So when somebody come and thief your wheels, you have 4 wheels, and somebody come and or the wheels fall off, that is when it gets dissociated. I keep saying disassociated. So dissociated. So that's called oxygen dissociation. So dissociated means gas is released from hemoglobin. Yeah. So such as when oxygen is released from the hemoglobin into the body cells. So if you have a red blood cell here, for example, and this is a body cell with a nucleus, and this red blood cell lets out some oxygen. It unbinds some oxygen from its hemoglobin basically to go into the body cell. You see that oxygen has been dissociated. Yeah? So it's kind of like it gets released. It's just a fancy word for release, but it gets dissociated. Now, affinity means, well, if we're just talking about people, imagine like you pull real good with your partner, right? You have 2 partners, and you pull real good with one, but the next one good too, but you don't pull as good with him. So that first partner, you have a high affinity to he. The second partner, not so much. So, but you still have affinity. But affinity, in terms of this, means how readily you typically one compound would bind to our next compound. In the case of hemoglobin, it means that how readily the oxygen, in this case the gas, binds to the hemoglobin. Oxygen has a fairly high affinity. It has a higher affinity than carbon dioxide. So carbon dioxide is the kind of partner that he good. And oxygen is kind of like, that's my best friend, boy. I have a high affinity for oxygen. Now, carbon monoxide has an even higher affinity than oxygen. Carbon monoxide is like a toxic friend. It's literally toxic. They rope you into do all kind of stupidness and then they kill you. So carbon monoxide why it's very dangerous is because it has extremely high affinity to hemoglobin. So oxygen might be your best friend, but carbon monoxide is that real toxic boyfriend or girlfriend that going to pull you away and make you do all kind of stupidness you wouldn't usually do and might eventually kill you in the end. Right? So that's carbon monoxide, but has extremely high affinity and can potentially, what will happen is that it will potentially bind to the hemoglobin car, but them tires on them not good. Them tires on them, as soon as they go on the highway, they're going to basically steer that car off the road and you're going to crash and you're going to die. Now, so they could bind to the oxygen and they could bind to the hemoglobin and leave no room for oxygen. Some people commit suicide by running I given nobody no ideas. Don't do it. There's run up a hose from the muffler of the car to inside the car and roll up the windows, and carbon monoxide is coming, and that's really, sometimes you just go to sleep and don't wake up. It had this movie called Midsommar, on Netflix. Don't watch it, all too young. Or let's say it's 18. But it's extremely disturbing, especially the beginning, which involves something with carbon monoxide. So yeah, so that's what affinity means. Affinity describes how readily a compound binds to the next compound. In case of hemoglobin, how readily the gas binds to the hemoglobin. Right? So from now on, you might see that I'm using HB as hemoglobin. It just is an HbO2 as oxyhemoglobin. It just means oxygen and hemoglobin bound together. Foreign use. Don't watch that show, Midsommar. Don't watch that, That's like if the devil direct a show. Don't watch it. So anyway, positive cooperativity. So positive cooperativity is this. So imagine that you have a hemoglobin molecule. There's a hemoglobin molecule here. And this hemoglobin molecule there's a hemoglobin car have no tires on it, right? And this here, well, if you're watching it, though, if the BioCape exam come hard, don't watch it, Don't watch it. If it come good and you're in a good mood, you could probably watch it. If you're in a bad mood, don't watch it, Anyway, so when an oxygen molecule binds to hemoglobin, the hemoglobin protein experiences conformational changes. And this alters the quaternary structure slightly. You would have learned this in doing proteins in Unit 1. So basically, when an oxygen molecule binds, like where I have circled here, right? When an oxygen molecule binds, the hemoglobin protein slightly changes shape. It folds a little bit to allow a next oxygen molecule to bind. And then a next one to bind. And each time this binds, each time this happens, it becomes more and more rapid. So it's kind of like this is not what is happening, but imagine like it learn how to bind one. You know how sometimes to do one thing it might take a little while, but then to do the second it take less time, to do the third thing take less time, the fourth to take even less time. It's kind of like that. That's called positive cooperativity. So it takes less and less time for each oxygen to bind until it becomes fully saturated. So this is a fully saturated oxygen molecule. So where it has 4 mounted, this is we have wheels and we're ready to go. Now, what is an oxygen dissociation curve? So this is where, so far we understand what dissociation means. It just means like release from. It will break away from, it will separate from, it will unbind from the hemoglobin to go into body cells, for example. Well, maybe me not telling it to watch it is because I want you to watch it. People will do things they're banned from doing. Reverse psychology. Yes. So every time let me come back here. Every time an oxygen binds to it, the affinity to it increases, which allows it to go faster, for it to go faster and faster. The conformational changes, it folds a little bit, allows faster and faster and faster binding. So in this case now, we have 4 oxygen molecules. So which means we potentially could release these 4 oxygen molecules to go to body cells. Now, this is where this will come in a little difficult now. So oxygen will only bind to hemoglobin, right? It will only bind to hemoglobin if the oxygen concentration is high enough. Now you're gonna see, I'm gonna refer to this as a partial pressure. So you might ask yourself now, what is a partial pressure? Why am I calling it that? So I can't say air pressure. The reason being is because air is 78% nitrogen, 21% oxygen, and the rest is carbon dioxide, water vapor, and halogens and noble gases and things like that. All I really care about is the oxygen, so this is why we refer to it as a partial pressure, basically 21% of the whole thing, a partial thing. So when I say partial pressure, I really just mean oxygen concentration, right? So in high partial pressures, and remember this means in high oxygen concentrations, So for example, in the alveoli of the lungs, this is the air sacs, more hemoglobin is oxygen saturated. And that makes sense? If you have high part, if you have plenty to go around, everybody take in. Hemoglobin, look at our oxygen there. Take some, take more. So in high partial pressures, more hemoglobin is oxygen saturated and it's more quickly saturated because there's a higher amount of oxygen ready for the grabs. So this would be like oxygen from the lungs, and binding would occur quite quickly because this would be a high partial pressure here, whereas label 1. In low partial pressures, which basically means in low oxygen concentrations, right, where oxygen is not readily available to bind. So when we say the oxygen would still be in the hemoglobin, but we mean in other tissues. So in low partial pressure, so not the lungs, for example, in muscle tissue, example, less hemoglobin is oxygen saturated. So what does this mean? So imagine like the hemoglobin pass by a little table that have like little two thing of food. Well, the hemoglobin mightn't take it. It be like, well, that's so little bit I might as well take none. Whereas if you pass by a whole buffet, that's the lungs is the buffet, and you'd be like, hey, look at real oxygen here, boy. Real food here, real oxygen. It might take some and load up the hemoglobin car with it. But in places where you don't really have much, it wouldn't really readily take any. So hemoglobin oxygen levels are lower around body tissues due to oxygen dissociation. So what's going to happen here now is that when partial pressures are low, right? Remember here we have a low partial pressure and here we have a high partial pressure. When there's a high partial pressure, oxygen would more readily bind to hemoglobin. When there's a low partial pressure, oxygen will more readily dissociate. So oxygen is a kind of given, not oxygen. Hemoglobin is a kind of given protein. Hemoglobin is kind of like, hey, I have all this oxygen and them I have none. Let me give them some. So in the case here, oxygen dissociates from hemoglobin and goes to these cells that need oxygen. So it's a very charitable protein, in my opinion. And that there is oxygen dissociation. But what does this look like as a graph or a curve? So this is what this would look like as a curve. So this curve here, we describe this as sigmoidal. Right? So sigmoidal just means S-shaped. It's not exactly like a S, but I'm surely heard the term already. But it's, it starts off like this, goes steep, and then goes to the plateau over here. Now, how do we explain why this graph is shaped like this, right? So the graph's steep beginning, that's over here basically, right? So the graph's steep beginning is due to the rate of oxygen binding rapidly increasing due to conformational changes in the hemoglobin. So basically remember here now, butts come back, boy. Anyway, so basically what this means here now is that at low partial pressures here, right? This would be low partial pressures. It is more difficult for oxygen to bind. So in the case here, this is where you would have hemoglobin molecules in red blood cells. And this at this point here, this would cause oxygen to dissociate. So oxygen would actually leave at this point here. Now where there is higher partial pressures, this would be a high partial pressure here. You would more get the loading of oxygen into the molecules. So like over here, this would be like where the lungs is. Right, so over here would be like the lungs. And down here might be like muscle tissue. So this is where this is where oxygen will start to get dissociated and the oxygen saturation of hemoglobin would be lower. So this is where, oxygen is being dissociated from the hemoglobin. And over here, this is where oxygen is being loaded right into the hemoglobin. So where there's a high partial pressure, this is more where you'll get binding occurring. And at lower partial pressures, this is more where you get dissociation occurring. So lower partial pressure, remember I say think of hemoglobin as a very charitable molecule. Where it have low partial pressures and the hemoglobin saying, these cells that have no oxygen, these poor cells that have no oxygen, I'm gonna give them some. So at this point here, oxygen saturation is low. This is because oxygen is being dissociated. Again, these would be in like muscle tissues and things like that. This would be like around this area here. In lungs, you have plenty for the taking. So the hemoglobin is getting a little greedy now. So it is end up a charitable bit, a little greedy too. So it's saying plenty oxygen, it will start loading up and then rolling away with 4 oxygen tires. So this is what that graph means. So at down here, right, this is where oxygen will more quickly dissociate from the hemoglobin. Up here is where it would more quickly bind. That's what that graph means. Now the last thing we have to look at here is the Bohr effect. This is named after Niels Bohr's father. Or you might know Niels Bohr from like the atom and thing. So his father come up with this. And I think both of them win Nobel Prizes. I think so. I don't know bacchanal with he. When he would like Calvin and them. But Niels Bohr, I think he sound like a normal fella. So the Bohr effect is this. So this refers to how hemoglobin's affinity for oxygen is affected in certain conditions. So this is where this curve, so in the solid line here, this would represent your typical sigmoidal oxygen dissociation curve. This curve, you could think of it as being dynamic, right? So not like, the CXC curve do exist, right? So when they say this grade along, that do exist. But this curve here is dynamic. So where it could shift to the left, or it could shift to the right. And typically, when it shifts to the left, it means that there's an increased affinity to, there's an increased affinity to oxygen. So if the curve shifts to the left, if CO2 levels are low, this increases affinity and allows more oxygen to be taken up. Wait, I feel like I'm mixing up myself, yes? I'm gonna double check there. Yeah, no, sorry. I chain up myself a little bit. No, correct. So oxygen leaves less readily. Nah, it's no curve, boy. Them thing do exist. People like it. Them curves, CXC curve do exist. I don't know who tell all of them thing, but that do exist. Oxygen dissociation curve does exist. Radioactive decay curve does exist. Not CXC curve, that do exist. CXC bell curve, that's a myth. Anyway, so at this point here, oxygen, it kind of, you can think of it as it holds on. It leaves less readily. And I don't know if we could come up with a little acronym for this, but I used to think of it like, well, when I was learning it, I was thinking it like this. When or they ever went to a prize, or they ever went to like a prize giving or something like that. Typically, you would hand somebody something with your right hand, like you would give somebody something or a prize or something with your, is your right hand, boy? Or you shake with your right, whatever. Imagine you're giving somebody, that's not a good example, nah? I know if I giving somebody something, I'll give them with my right hand. I don't know. But in this case here, it's kind of like oxygen is being given to things when it shifts to the right. And when it shifts to the left, I'm trying to come up with a little shortcut here for you. When it shifts to the left, oxygen is kind of, okay, when it shifts to the left, it leaves the muscle tissues behind. That's not gonna. Or it could come up with something. I know it. I don't need no shortcut, but or it could come up with something to remember it. But basically, is this. So just to explain. So the curve shifts to the left if CO2 levels are low. So for example, in the lungs, this increases affinity, allows more oxygen to be taken up. So this would be like if you have decreased temperature, decreased CO2 levels, or increased pH. So I explained the pH thing just now. So the curve shifts to the right if there's a high CO2 level. So this would be like in the tissues. So this would be like in the muscle tissues where you would have less oxygen. So the curve will shift to the right and you have reduced affinity to oxygen. Remember, reduced affinity means that they will more quickly dissociate, which means that they will more quickly leave. And they would leave the hemoglobin molecules and go into those muscle cells, for example. Now, when CO2 dissolves in the blood, I don't think all you get this, but I'll just put it here. When CO2 dissolves in the blood, right, it is formed carbonic acid. And this and lactic acid increases hydrogen ions in the blood and thus it lowers the pH. That would be basically here. So when you have plenty carbon dioxide in the blood, that forms carbonic acid. Of course, if you're exercising, plenty of lactic acid that is built up. So all of this acid, all of this extra carbon dioxide, all these hydrogen ions exactly make your blood a little more acidic. So oxygen more readily binds to tissues and things like that to kind of dissipate these ions and these this acidic pH. Whatever, yes. I know it. I don't know what other way to describe it. But I just know that when the thing shifts to the right, oxygen does leave a little more readily. It's like it giving it away. When it shifts a little more to the left, it's like I holding on to my oxygen. It have enough. All I have enough oxygen already. I holding on to it. When it shifts to the right, it's like all I have enough oxygen. No, take some. That's basically it. But this is in response to carbon dioxide levels and in response to pH. Carbon dioxide levels, of course, is linked to acidity. Read the thing. I don't know what to tell you. When it shifts to the right, it's ready to give away thing. When it shifts to the left, it's ready to keep thing. When it shifts to the right, it's because of CO2 filling up the blood and the cells. This, of course, leads to a decrease in pH because of all the hydrogen ions, carbonic acid, and things like that. Shift to the left, now that this happened, so I keep in my oxygen. I don't need to dissipate no carbonic acid and all kind of thing. Yes, that is true, Anonymous. So I guess you have to know a little bit of chemistry to rationalize this in your head. That's a good one, Simba. The right thing to do is share. The left thing to do is not share. You're moving left with me now. Why you give me some oxygen? You're moving left with me now. Move right now. Move right with me. Give me some. Share some oxygen. Yeah, I don't know. You just have to use them kind of thing to remember. Alright, so that's module 2. And module 3. You know, I now noticed something about Ole group. Ole group here never write a normal CXC exam. Because Ole group in 2020, Ole just write that stupid paper 1 where they cheat all. And paper Cape last year was broad topics. And Cape this year is broad. Ole never write a normal CXC. Ole miss out. But that 2020 thing was real BS. Right, we skipping all them thing. Synapses and thing that I come in. All right. So we have 2 more chapters to do. You're from 2021 first. Oh, you're doing Unit 2 first. That Unit 2 first might be, I feel as a kind of struggle, especially how I only reference in Unit 1 thing. Honestly, I find when you reach university, the exams is be easier. I find, but I didn't write Cape, I write Cambridge, but I find A levels, I think it was real pressure away. And when I reach university, I find it was easier, honestly. Because each syllabus short and each course syllabus, and then coursework and thing is like 40-50%. You just had to go your classes and not drink rum, and you do good. All right. So we have 2 more topics to cover, which is health and disease and immunology, and then we're done. And that would make we here for a good 8 hours, I believe. I really envy all you doing this Unit 2 first. Now, this Unit 2 is when I was going through that, I was real pressure. Unit 1 easier. I find Unit 2 has some kind of things in it. Is it true that UE won't accept science students unless they have CXC and CAPE in sciences 1 and 2? No, that's not true. You could get through. You could I would say it don't get less than a 3. But UE kind of hungry for people. Don't get less than a 3. UE kind of hungry for people. They want money now. So I won't worry too much. All right. So let me start this one here. Health and disease. First, we should know how to define each one. Health is a complete state of physical, right? So we have to touch 3 characteristics here. 3 factors, sorry, physical, mental, and social. A lot of people just forget mental and social, but that's why Trinidad have all these madman and thing, all these mad people running around. When I say mad, I mean like literally insane. I mean unwell, unstable. Plenty unstable people who can't handle themselves. So health is not just you working out and you have good blood flow and you're free of disease or anything like that. No. Health means that you are physically without ailment. You have mental well-being, mentally and emotionally well, which means that you take time for yourself, you take rest days for yourself, you take time to do the things that you want to do. I know that keep on thing is stress, but you have to take the time to do the things you want to do, because eventually by the time you actually get the job you want to get 7 years from now, I don't even know, 4 or 5 years from now, by then you don't even know who you is anymore, and you're just working to do, I don't know, to get money, and then you might be like, what are you using this money to do? So yeah, mental well-being, social well-being, both of those are connected. Do I have any tips for Caribbean studies? Nah, I didn't even do those subjects. Anyway, so diseases can be communicable. And communicable means that they can be spread from one individual to another. COVID-19 or SARS-CoV-1 it's a good example, one that you was exposed to for the past few years. Other examples would be influenza, cholera, dysentery, dengue fever, malaria, tuberculosis, chickenpox, measles, monkeypox, Ebola. All of these here are communicable diseases, so they can be spread from one individual to another. They're infectious and they are caused by pathogens. There are different ways in which diseases can be spread. They could be physical contact. They can be exchange of sexual fluids. Example would be gonorrhea, chlamydia, herpes, hepatitis, HIV, syphilis, and so on. So it could also be droplet infection, which is you sneeze and somebody inhales the droplet, which if they're very light droplets, it could be airborne. Example is tuberculosis. They could be transported by vectors. So for example, the Aedes aegypti mosquito has the ability to transmit the dengue virus, which causes dengue fever. The Anopheles mosquito has the ability to transport the protozoan Plasmodium falciparum, which causes malaria. Now, so there are different ways in which it can be spread. Now, non-communicable diseases cannot be spread from one individual to another. So, an example would be diabetes type 1 and type 2, diabetes mellitus. So, if I have diabetes, I cannot give you diabetes because it's non-communicable. Hypertension would be our next one. Arthritis or osteoarthritis, coronary artery disease, schizophrenia, alcoholism, obesity. All of these are non-communicable diseases. They may be caused by a lifestyle, or they may be inherited like hemophilia, or sickle cell anemia, or cystic fibrosis. So all of these here cannot be spread from one individual to another. It can be transferred. The risk can be transferred by genes. It could be transferred by autosomal recessive alleles, but cannot directly transfer to somebody else. So there are different categories of disease. So this is what cancer is typically considered non-communicable, but there are certain types of cancer. For example, cervical cancer, which is caused by the human papilloma virus, that is communicable because it's spread through sexual intercourse. Of course, men don't have a cervix, but if they don't wash down there and they're uncircumcised and they don't wash and they have sex with a woman who does have HPV, human papilloma virus, they can become a vector or a spreader for HPV and give women the pathogen that will cause cervical cancer. The man himself won't get it though because he don't have a cervix. So anyway, so there are many other categories of disease. However, it is important to keep in mind that any one disease, this is very important, any one disease cannot be placed in a single category, right? So even though you have multiple categories of disease, you cannot really relegate a disease to a single category. Diseases might occupy 2 or 3 categories. So for example, diabetes type 2, right? Diabetes type 2 is caused by a malfunction. It's a physiological disease because it's caused by a malfunction in an organ or system. Well, let me say diabetes type 1. Diabetes type 1 is caused by a malfunction in the pancreas. And it is usually a chronic disease. So chronic means that the disease is long-term. So these here, so you cannot really group one into individual categories, but there are categories in which diseases can be placed in, but you could think of it as a Venn diagram more than individual categories. It's just a way in which to classify diseases. It's not, a thing that kind of just is one for one. So physiological diseases are caused by a malfunction in organ or system. Diabetes will be a malfunction of the pancreas, or diabetes type 2 would be a malfunction of the GLUT4 receptors on the body cells. The coronary heart disease, of course, malfunction with the coronary arteries in the heart, and asthma, which is malfunction with the bronchi and bronchioles. So mental diseases occurs due to the impairment of the mind or the tissues within the mind. So you have Alzheimer's disease, clinical depression, and schizophrenia. Clinical depression does count as a disease, by the way. A lot of people think it's not, but it is. Chronic diseases are diseases that are long-term. So examples of these would be diabetes, chronic bronchitis. The opposite of chronic, by the way, is acute. Acute means it's more specified to a specific time. So like influenza is acute, but pneumonia is chronic. Typically, they say if something is chronic, you get it for more than 3 months. If you get diarrhea for more than chronic diarrhea, might be 3 weeks, yes. But if you have a full, but diarrhea is not a disease, that's a symptom, that's a sign. But if you have a disease for more than 3 months, you just call it chronic. So like, people are long COVID, that's like a chronic disease. Allergies is not a disease. So allergies are caused by allergens and your body's inflammatory response to allergens. Allergens are kind of like antigens. So infectious diseases are communicable diseases, and they are caused by the introduction of pathogens to the body. Dengue fever, malaria, cholera, the deal, COVID. Social diseases are caused by an individual living or working within an environment prone to contamination by pathogens. So they have no access to a balanced diet. So, for example, if you have people living in an area where you have no potable water, like no drinkable water, and they have to drink from a river that people squatting in, or they have to, that's the only water supply that, or they have a well that becomes contaminated by a landfill or something like that, it can lead to cholera and dysentery. So those are social diseases because those are environment based. Those are caused by social factors, economic factors, and things like that. They can easily be eradicated, or the incidence of those diseases could decrease if it was to fix those social factors causing it. So like scurvy, which is a lack of ascorbic acid or vitamin C, rickets, which is a lack of calcium or vitamin D, and night blindness, which is a lack of vitamin A. All of these here are nutritional deficiency diseases, which means that the person doesn't have access to a balanced diet. This is why they genetically engineered golden rice so that the rice could produce beta carotene to get rid of night blindness in some countries like can't remember if it's Malaysia or Indonesia, one of those. Now, degenerative diseases are caused by the gradual loss of function of tissues and organs. So Alzheimer's disease is the degradation of some tissues in the brain, osteoarthritis, where we have degradation of bone tissue and cartilage. So this is basically, again, these could be counted as chronic as well because they are long-lasting, but they result in a gradual loss of function. Our next one would have been, if all you hear about ALS, is like what Stephen Hawking had, is a motor neuron disease, where he had a gradual loss, I think it was like of myelin in his motor neurons. So, but it was a gradual loss. It didn't happen all at once. So that's degenerative. Deficiencies diseases are due to a lack of a particular nutrient in diet. These are typically also classed under social diseases. We could still consider AIDS a social disease in some countries because some countries don't have access to the educational aspects behind it, don't have access to barrier methods of contraception. So that could count too. Yeah. The PowerPoint is on, well, I'll link it after. And there's a video, I uploaded a video of me going through this PowerPoint as well, but really, really quickly. Examples, kwashiorkor, scurvy, and night blindness, right? Okay. Hereditary diseases are caused by inheriting alleles, typically recessive alleles, that translate to malfunctioning proteins. Those of you who did Unit 1, you all would remember transcription and translation. What would happen there is that if you have like a substitute, like I use cystic fibrosis as an example. If you was to swap out one base for our next base, that could translate to a whole different mRNA molecule and a whole different protein. And as a result, it might form a whole different protein or a malformed version of that protein. And as a result now, for like say cystic fibrosis, for example, the transmembrane proteins don't work so well in the lungs, so they end up building up mucus. Right, and this is hereditary. You can't really give some, like if I have sickle cell, or cystic fibrosis, I can't give that to you. No, immunology is the last thing I'm going through. Self-inflicted, also referred to lifestyle diseases. These occur due to the intake of excess nutritional drugs. This is habitual diseases. This is like liver cirrhosis, if you or what is called fatty liver disease, like from drinking too much rum, basically. Lung cancer from, let me say, intake of too much tar from smoking. And coronary heart disease, which is due to the intake of excess lipids, low density lipolipids, low density lipid cholesterol, or having it could lead to well, I feel like I had a brain fart just now, but just to explain the simple thing, but it could eventually lead to a heart attack. Will they ask to explain a specific disease? No, they won't ask you to explain a specific. They might ask you to explain a specific category, and then they will ask you to list a specific disease and describe not a specific one. They'll ask you to list a disease from that category and describe that disease. So I would suggest learning all the categories or learning at least 1 or 2 diseases from each one, and just learn simple ones now. Learn sickle cell anemia or learn liver cirrhosis. Leukemia would come under well, I think cancers would fall under degenerative and chronic. But you could use basic examples. Don't try to go too far with it. Use cholera and use dengue and all kind of thing. Use scurvy. Use them basic examples. Use safe ones. All right, I go into these graftings, but basically, if you should know how to analyze disease data. I go into any, but I'll send the PowerPoint after so you can go through it yourself. But you should know how to analyze disease data, whether it's a line graph, a bar graph, a table, and be able to correlate. You should be able to correlate the following things, right? You should be able to correlate incidence rates of disease. That's basically how often the disease occurs. When you get COVID updates every day, and it'd be like, hey, we have 20 new COVID cases today. That's incidence. That's like how much people catch the disease. Then we have, so what they also call cases, right? We have 20 new COVID cases today, for example. And then we have mortality rate. So we could have 20 new cases and 2 deaths. So that's why it's called a mortality rate. Basically the death rate. I think that's all. Let me double check here in the syllabus just to make sure if it have any other thing you're supposed to know, but it's basically incidence rate, mortality rate, and I'm just making sure. All right, no. Incidence, which is the number of new cases of a disease. Prevalence, which is the total number of cases of people with the disease in the current time. And mortality rate, which is the number of people who died from particular disease. This could be per day or this could be per year. It could be per any time. What if you don't know? Well, you have to learn it. Tomorrow 9 a.m. is the exam. You have to learn it. Yeah, so you have to know incidence, prevalence, and mortality rate. I say incidence is new cases, prevalence is like total current cases, and what was the other thing? Mortality rate is basically how much deaths occurred. I will send the PowerPoint when this video done. I will pin it on a comment. I'm not going through past papers. I'm not going through content, but I will link a whole set of past papers after. If it have any particular papers, I will show that. Ashley, no, you don't have to know about those. Those actually fall under social and preventative medicine. So the modes of action and things like that, you don't have to know them. Just the categories. Does paper to repeat? Not really. For CSEC, sometimes it gets simple. I find Cape that is kind of, they already play lazy with Cape. I find a lot of the questions as be, even if they come a little similar, they still have their own spin to it. I know about the other subject, but bio doesn't. So I don't find so. All right. Last topic, which is immunology. This topic a little long, so hopefully we could finish it by 10 o'clock. And if the video, so you know, sometimes the live does take like a half hour and thing to actually show up when it's done. So I will also post it on community posts on my channel. I'll post all the links on community posts. That's basically, you know, where the person just posts all the shippiness and all kind of thing. I will link all the paper ones and you can do it yourself. I don't think we need to do a marathon for that. You know how much marathon I do? You know how much marathon I do this week alone? For free? So I think people will need a marathon. I got link all the thing. Anyhow. Alright, so let me go here. Immune responses. Let me start. So immune responses are defined as the reaction of white blood cells to the presence of a foreign substance not recognized as a constituent of the body. So basically what this means is if something foreign enters your body sounds the alarm and tries to get rid of that foreign substance. So, for example here, we could have bacteria enter the body, we could have viruses enter the body, we can have allergens enter the body, we could have toxins enter the body. And what happens here is that these bacteria, right? So imagine if I draw a bacterial cell here. Cheapest tinder. So these would have like these little attachments on them. So these attachments here on the bacteria, so this here would be a bacteria, E. coli, whatever it is, right? And this here attached to it, this would be called an antigen. So what an antigen is what the body can detect. So if I was to use an analogy, right? The bacteria would be like the burglar, right? That coming into your body. And the antigen is the thing that would trigger the body's burglar alarm. So the antigen, if the bacteria didn't have any antigens, the body wouldn't even know it there. So the antigen is the thing that would say, that alert the body, it has somebody in here though. Alarm, it has somebody in here. Send them leukocytes now. So that's what the antigen would do. The antigen would trigger the body's burglar alarm. And that would then stimulate the production of lymphocytes and phagocytes and all these nice things. So that is what our immune response is. Now, we have, so I say when we're going through the circulatory system, we didn't really go through the cells that much. So now we're going through it now. Yeah, your dog barking could be the antigen. The dog barking would be the signal, right? And then that would then it would signal to everybody else, it's time to come and defend the body. Cells at work. So phagocytes patrol the bloodstream in search of antigens. And these are like guard dogs, if you know what I mean. You can think of it like this, or soldiers, security guards, whatever you want to call them. When they encounter them, they engulf them. So when they encounter them, they could basically, you could think of it like this. They create an evagination. and they could basically engulf any toxin or bacterial cell, take it into their body, break them down, and then release the remnants into the bloodstream for the macrophages and things to clean up. So basically, just kill them and eat them, and then spit them out. That's what phagocytes do. So I guess they're more like a real bad dog. Anyhow, so there's the thing too now. When phagocytes kind of dread now, because phagocytes, they're not only killing this bacteria, they're basically going to cut off the bacteria head. Think of it, the metaphorical head, and they're going to present that piece of the bacteria and be like, this is what he looked like, this is what they look like. The bacteria be like, the phagocyte be like, I cut off his head, this is what he looked like, this is what they look like, find the rest of them. So they could also act as APCs. APCs are antigen presenting cells. So they could actually take a little piece of this bacterium, they could hold it, well, not inside them, sorry. They could hold a little piece onto one of their receptors or something like that. They could hold a little piece. Like imagine like they're walking around with the bacteria head, metaphorical head, on a stick and showing all the other lymphocytes, this is what he looked like. That is what you call an antigen presenting cell. Now, B lymphocytes and T lymphocytes. So B lymphocytes multiply and they secrete antibodies. And these counter antigens. So B lymphocytes are like the chemists. There's like, we're mixing up some grenades and things for all your, we're mixing up some antibody grenades and we're going to pelt it towards all your bacterial cells. T lymphocytes have different types. So T lymphocytes can be helper cells, we call them T helper cells, or they can be killer cells, T killer cells or cytotoxic T cells, same thing. So you have many other types, by the way, but this is the ones that we limiting it to. So T lymphocytes can be helper cells. So helper cells stimulate B cells and phagocytes. So helper cells could kind of act as messengers to get information around. It can supply energy to some of the other white blood cells. Yeah, different types. The whole thing is basically a big army. So they destroy the cells infected by the pathogen. So killer cells is like, if you ever see cells at work or you ever see them cells and then with the knife, you know, and the real dread ones, I think them is T killer cells. Right. So coming here now, this is something that I think might come. Humoral and cell-mediated responses. So this is the difference here. So let me start here with a phagocyte, right? Let me start here with a phagocyte. So a phagocyte would engulf the bacterium or the virus. So it's going to engulf it, and they could have little folds. They call these evaginations. And basically they could give it a big bear hug. They could give it a big hug and absorb it into the body. So this virus now is going to get absorbed into this phagocyte, into a little vesicle through endocytosis. If I remember endocytosis. And what is going to happen now is that it can actually attach. You see these things around the virus here? These here are basically like little keys, and these keys allow the virus to enter the cell. These also act as antigens. So antigens, remember, is like the body's burglar alarm. So some of these keys here on the virus, some of these spike proteins can get deposited onto the phagocyte. This is the metaphorical head of the virus. So some of it get deposited here, and the phagocyte now, after engulfing it, can say, this is what it looked like. So now this is called an APC, or an antigen presenting cell. This is what you're seeing here. So the phagocyte acts as an antigen presenting cell, shows the antigen to a T helper cell or T killer cell, and the T helper cell is going to recognize the antigen and is going to rally others into action. The T helper cell is like the scout, is like the messenger boy. Now, so there's two different responses here. The humoral response and the cell-mediated response. The humoral response involves B lymphocytes. So remember I say B lymphocytes, think of them as bomber cells. They just make little antibody grenades or little antibody bombs and release those to kill the pathogens, whether it's virus or bacterium or whatever, protozoan, whatever. So these produce antibodies. So the humoral response involves B. So think of it as bomber cells, right? Bomb, antibody bombs. So these produce these antibody bombs, antibody grenades, and sends them in order to destroy whatever foreign enter the body. So, of course, we need the help with T helper cell now. With T helper cell is going to signal to B cells to be like, make this bomb now, boy. Make this bomb, send out this antibody bomb and kill them thing. T helper cells could also stimulate T killer cells. So these are also called cytotoxic T cells, and these will straight up just kill the cell. They have chemicals, they have they can sap energy from it. It had all kind of different ways they could do it. It have dendritic cells that could literally sap the energy from others, kind of like vampires. And they could destroy the cells like that. I say if you ever watch Cells at Work, though, I mean, if you never watch it, don't watch it now, obviously, like before the exam. But if you ever watch that, you would actually get a good little analogy, allegory to it. So, the humoral. So, humoral responses, cell-mediated responses. So let's look at the differences now. Between B lymphocytes and T lymphocytes, this is something that could potentially come here. So both of them originate in the bone marrow. So you might be asking why they call it B lymphocytes and T lymphocytes. Well, this is because the B and B lymphocytes means that they mature in the bone marrow, and the T lymphocytes mature in the thymus gland. The thymus is a little below the thyroid, and that's why it's called B and T. So, which response? The B lymphocytes, humoral responses. The T lymphocytes, cell-mediated responses. Humoral responses are more rapid because those antibodies can be released in very large amounts, and they can quickly get rid of pathogens. They use a lot of energy though. Now, cell-mediated responses, which are T killer cells, and T helper cells help, but T killer cells, they actually have to go and seek out the pathogens, kind of like assassins. And so it takes a longer time, but they're more effective at doing it, and they don't expend as much energy. The secretory products of B lymphocytes are antibodies, and the T lymphocytes, the secretory products are cytokines. So let me say that you was to get, let me use an example, right? You get COVID, right? You get COVID, and what's gonna happen now? So your body, your phagocytes are going to try to engulf that COVID virus. Some of the spike proteins are gonna latch on to that phagocyte, and the phagocyte is going to become an antigen presenting cell. The antigen presenting cell is going to present the COVID antigen to the T helper cells. The T helper cells going to stimulate B cells, B lymphocytes, and T killer cells, and that is going to initiate, these two types of responses. Now, when this usually happens, and when you get viruses multiplying and proliferating, basically viruses multiply very, very, very quickly. They reassemble DNA, they turn cells into photocopying machines for themselves, and they multiply by a bigger amount, right? And as a result now, T killer cells, these are going to secrete a whole set of chemicals called cytokines. And these cytokines, now when this gets released, it's called a cytokine storm. So you basically get a whole set of it in your body. And this is actually why you start to feel sick. You don't actually feel sick so much from the COVID. You actually feel sick from your body's response to the COVID. Because there's so many, they are proliferated and multiplied so many times. Your body is basically expending loads and loads of energy to release these antibodies, to release these cytokines, to make the cytokine storm happen. You're going to get multiple inflammatory responses through all your organs and tissues, and that there is going to make you very, very, very weak, because your body is basically doing all it could to try to get rid of this pathogen. And if it can't get rid of it, well, hard luck. But that's basically what happens here. So B lymphocytes can differentiate into two types of cells here. So B lymphocytes can differentiate into plasma cells. Plasma cells are those that secrete antibodies. And they can also differentiate into memory cells. We'll get into memory cells a little later. So T lymphocytes can differentiate into T killer cells, T helper cells, memory cells, dendritic cells, much, much more that we don't need to get into here. If you'll ever hear about the YouTube channel called Kyrgyzstan in a nutshell, they have a very good video on all the different types of leukocytes or white blood cells and how they interact. Very complex video, but very good. A lot of work went into that. Because, as I said, this is an entire army that is within your body that's trying to get rid of these pathogens. So now we come into the humoral response. What exactly happens during this response here? Remember, these involve B lymphocytes, right? So B cells experience what we call a specific binding response. So if you look at one here, so we have 1, 2, 3, 4. The antigen receptor in the B cell binds to the specific complementary antigen. So basically what this means, sorry, my bad. The B cells, right? They are specific to the antigen. So they will know who the enemy is. And they could recognize that antigen and they would be like, that is the enemy. That is there. The antigen is binding to these receptors here. So the antigen must be in contact with the receptor on the B cell surface membrane. That's these things that you're seeing sticking out here. So you have different types of B cells. You have, let me say for example, you have some that would detect measles. You have some that would detect COVID. You have some that would detect the flu, different strains of the flu, and so on. So now, it's similar to, if you think about enzymes, the enzymes have like a lock and key mechanism, but you have two complementary compounds kind of binding together. Think of it like that. So that's the B cell response. So when they're going to bind now, this is what's going to happen now. When they bind, this is where this is going to trigger something called, well, is this one that bind? Is this one, not the other one. So when these antigens bind to the receptor sites here, this is going to trigger what they call clonal proliferation. Think of clonal proliferation as like mitosis occurring real plenty times, right? So it's basically going to divide, and they're going to form a whole set of B cells. So it's kind of like you have 1, then you have 2, then you have 4, then you have 8, and so on. Right? So you're gonna get a whole set. It's basically man calling his squad, but a man kind of making his own squad. Imagine a man come to beat you up, and you can make your own squad. That's kind of like what clonal proliferation is. So now you have your squad, right? You literally make your own squad. I make 10 of me, and now I want to beat up this man. I want to beat up this pathogen. I want to give him licks. So what I'm doing, I form my own squad, literally form a squad of myself 10 times, 20 times, 100 times. Shadow Ninja. And so you're going to have large amounts of antibodies are produced from clone B cells. Yeah? So we're gonna have a whole set because it have a whole set of me. I could throw I have it at 10 of me. I could throw 20 fists. So, right, so I could have large amounts of antibodies being produced from these clone B cells. So as a result, now you could have these plasma cells. These plasma cells is the bad man on them. These plasma cells is the ones that are going to secrete all these antibodies. Memory cells will also form. Memory cells is kind of like, I go remember you, know. I go remember you. Next time you come, I remember you, know. I remember you, and I come in for you. If you come back here, I come in for you. That's the memory cells. I remember you, know. I take your information. I remember your face. I know your voice. I will hunt you down. And I will kill you. Take on. So that's memory cells. So memory cells now, they don't really do nothing right now. But memory cells are going to remember the pathogen. So they are going to produce antibodies if a future infection occurs. So this squad here, these plasma cells, these could go on. These is the soldiers. Them going to go out and them going to take care of business. Some of them might dead, but memory cells, them not really going out there. Them just going to remember. Them to be like, I see you, know. I see what you do. I'm coming for you next time. That's memory cells. So once again, remember there's a humoral response. We have the antigen binds to the receptors on the B lymphocyte. It undergoes clonal proliferation, and these could then differentiate into either plasma cells or memory cells. Plasma cells is the squad that are going to beat up the pathogen. Memory cells is the I remember here. Right. Memory cells will produce antibodies if a future infection occurs. So memory cells are more associated with the secondary immune response. Now, what is the cell-mediated response? What is the T cell response now? Now remember, I say the humoral response more involved the antigen binding to the receptors. It caused clonal proliferation. And then you had a bunch of plasma cells that are going to fight down the thing, and a bunch of memory cells are going to remember the thing. Right now, these butts is like pathogens, and the muds is like T killer cells. That's what you're doing now. So what T cells do, remember these mature thymus, they utilize an APC, which is an antigen presenting cell, and remember these antigen presenting cells, this phagocyte, this is going to literally hold up the bacteria, the metaphorical bacterial head or the viral head of it, right? Now, these would have the antigen attached to it. So these could be dendritic white blood cells like phagocytes. Dendritic just means they have little branches. The word dendrite literally means branch. Or they could be engulfed within it. So sometimes they could actually be carrying the pathogen within them, not broken down. Phagocytes would do that. So yeah, I said there are different types, but in either way, it has the antigen within it or on it. So the APC now is going to present it to the T cells, typically the T helper cells. And the T cells are going to experience clonal proliferation. And these are going to produce T helper cells and T killer cells, or cytotoxic T cells. And now the T killer cell is going to attempt to destroy the infected cell with the virus in it. And these, remember I say what they do, they secrete cytokines in order to do this. There are many different modes of action, right? But they can attach to it, they can sap its energy, they can secrete cytokines. Cytokine is like a poison. They would like poison it, kill it, things like that. The dread. So the T killer cell would attempt to destroy the infected cell with the virus in it. Basically attached to a little receptor here. Kill the whole infected cell. Not just kill the whole thing. Kind of like if the infected cell become a zombie. Shoot it in the head. So for When it gets the cytokines within it, the cell will eventually undergo lysis. Lysis means it will be destroyed, it will split. And 5 memory cells could also be produced and will proliferate upon future infection. I'll get into secondary responses just now. So that is the cell-mediated response. So that's the T cell response. Remember, T cells have T helper cells and T killer cells. T killer cells use cytokines, like a poison. T helper cells send signals to rally together T killer cells towards the infected cell, whether it's a red blood cell or what. So if you had to imagine all of these cells, say all of them play different roles. Think of pathogens as enemy soldiers. T killer cells are like melee combatants. They have like knives and poison. There's like assassins basically. They have poisons, which is cytokines. They have all kind of different chemicals that could destroy infected cells. B cells is like bombers or snipers, and the antibodies are like bullets or bombs. You could think of it like that. So B cells, be like plasma cells, they're going to produce a lot of antibodies and be like, ploop, 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 pelt that out. T helper cells are like messengers. They will rally together other T cells to where the action is, to where the pathogen is, or where the antigen presenting cell is. Antigen presenting cells are like spies or reconnaissance. They basically would gather information, bring it back, and be like, this is the enemy, this is what they look like. Memory cells are like veterans who have already seen the enemy and they could form strategies. So they too are kind of like spies too. So they would be like just people they're hanging around and be like, I see you now, I see you, I'm coming for you, I'm telling everybody about you. Right? So that's memory cells. If you think of it like a bunch of as like an army, it's easy to remember the different roles. Give it a read back, before tomorrow if all you have time. Alright, so the next part of this is phagocytosis. Phagocytosis is something that when it has come big. It has come for a good 6-7 marks when it comes. Whether it's to draw or label a diagram, or whether it's to write a detailed description of what happens. Fortunately, it is not hard at all. So what's happening here, I said phagocytes patrol the bloodstream and surrounding tissues using plasma membrane extensions. These are called pseudopodia. Pseudopodia is Latin for false foot. Pseudo means false or somewhat like. Podia or podium or pedal means foot. So this is a pseudopodium. So they can actually just kind of stretch around and move around, right? So what happens here? So the bacterium becomes attached to membrane evaginations called pseudopodia. That's what you're seeing here. So they ingest the pathogens via endocytosis. Remember, endo means in. What that means is that they're going to wrap around. They're going to wrap around this pathogen here, this bacterium, and it's going to form a vesicle. This vesicle here, this is called a phagosome. So the bacterium is ingested, and it forms a don't say eaten, right? Because just say ingested. So the bacterium is ingested, and this forms a phagosome. That's what you're seeing here. It's basically a vesicle containing a bacterium. It's kind of like a portmanteau of phagocyte and lysosome. So lysosomes now, which are these here, lysosomes I like to think of lysosomes as like garbage men of the cell. What they would do is that they would secrete enzymes and be like, take out this trash from an man. So what they're going to do is that the phagosome is going to fuse with the lysosome. There's the lysosome here. Phagosome is going to fuse with it. That's what you're seeing here. And the lysosome is going to secrete a bunch of enzymes, lysosomal enzymes. These are catabolic enzymes that are going to break down the pathogen, whether it's a bacteria or virus. They're basically going to disassemble it. So lysosomal enzymes are going to digest the captured material, break it up, basically kill it, cut up your body, and then going to throw him away in a river, right? Or back into the blood plasma. So the digested products are then released from the cell. Throw that out. We have that in here. But remember, it could actually keep some of this material, and the phagocyte could be used as an antigen presenting cell. So you don't need to throw out all. You could keep the head and be like, I'm keeping this as a souvenir. That is phagocytosis. Not hard at all. Very easy to visualize. Keywords here is, of course, evaginations, which is the folds. It folds around. Phagosome, lysosome, and of course the enzymes within the lysosome to break down the material. All they going so far? All they good? Are they keeping up? You don't that much again. We're nearly done. We should be done in a half hour. I just taking a little stretch here. Like my voice box, my larynx working constantly for hours. Right, so I think what we have left to do is the inflammatory response, the secondary and primary immune responses, monoclonal antibodies, and types of immunity, and I think that's it. Head hot. Oh boy. Alright, let me start back. All gas, no brakes. All gas, no brakes. We're going straight to the end, Ollie. We're going straight to the end. Alright. The inflammatory response. Right. So, not that much you have to know about this. Not that much you have to know about this. We have cytokines. And cytokines are chemicals that sometimes stimulate the immune system. And as I said, this is what is released by T cells, mainly T killer cells. So T killer cells are going to release these cytokines. They call it a cytokine storm when you have a lot of them. And these are going to stimulate the immune system. They act like a poison. Some of them act as signals. Some of them act as like poisons to kill infected cells. But these are going to get released, right? And they are going to go to infected cells or infected tissues. So they interact with blood proteins called complements. And these activate enzymes that lead to inflammation. So this is what you're seeing here. So imagine like you have an injury and you have a cut, and that cut there, some bacteria come into that cut. Like you didn't, you treat it, you apply a bandage and it gets a little infected. Well, what's going to happen is that some pathogens going to enter where the skin is broken. And this is going to, as I said, this is going to cause inflammation. So you have these cells here that are called mast cells, right? So mast cells release a chemical called histamine. So histamine dilates capillaries and ease blood flow to inflamed areas. Yeah, sorry. So basically what they do is that they increase the permeability of the capillaries. So this allows, white blood cells and so on to come to that area. And as a result now, it produces a lot of pus because you have a lot of dead white blood cells. Like if you have a pimple, for example, it gets inflamed. And that's because of that histamine here. Or if you have an allergy. So typically when somebody has an allergic reaction, they would take an antihistamine to kind of reduce the swelling, reduce the inflammation. It stops this cascading process from happening. So what happens here now is that because of this histamine being released from the mast cells, the capillaries become a little more permeable. And these white blood cells are able to actually exit the capillaries and they could flow towards the cells here to kind of take care of the pathogens. Now, that is basically an inflammatory response. So this could happen in different ways too. I say it doesn't have to be due to injury. It could be, if, for example, if you're allergic to pollen, pollen can also stimulate the mast cells to release histamine, and that could cause the same response. Or if it's like flu or something like that, or if you're allergic to a medication like ibuprofen. So anyway, so that's what's called the inflammatory response. It don't really come. You usually see it pop up in paper 1. So I'm not gonna stick too long on it, but still read the thing. All right, this is something that comes from time to time. The antibody structure and function. So they like to bring this particular diagram, ask you to label parts of it or describe it. So what are antibodies? You should be able to define an antibody. You should be able to label different regions on the antibody and say what those regions are. So antibodies are blood glycoproteins that are secreted by B lymphocytes, plasma cells, during the humoral response. Each antibody has a specific region to attach its complementary antigen. So this is called the variable region. Or remember how I say you could kind of link this like an enzyme, and the enzyme has like the active site is like a lock and key mechanism. The variable region of this, the part that is different, that is specific to it, that is kind of like the active site of the antibody. Of course, it's not the same as active site, but because it is this specific sequence of amino acids, it is a glycoprotein. Because of this specific sequence here, this is the part here, and I'll just kind of put them in red like this. This is the variable region here. So these would be able to bind specific antigens here. So these would be different shapes that would be complementary to the antigen. So the constant region, that should be the constant region, is common for all antibodies. So that is this part here. So that's the rest of it. So this part here would be the same for any type of antibody, right? It's only the variable region that would be different and that would be specific to the antigen. So these are consisted of light and heavy polypeptide chains, and they link together by disulfide bonds. So this is, it kind of has a actually not too sure. I think it has a tertiary, I don't know if it's tertiary or quaternary, but it has like kind of like a tertiary protein structure because it has, all these different types of bonds and things like that. Kind of like an enzyme, as I said. The same way an enzyme has an active site, this thing has something called a variable region. Okay, this is something that they love to bring, primary and secondary responses. But it came last year. So, did it come last year? Oh no, it did not come last year. It came in 2019, so it might come for all the year, who knows? It's something that let me tell you all the years this come. And this is going back to 2014. It come in 2016. They come in 2015, it come in 2016, it come in 2018, it come in 2019. It didn't, 2020 didn't have a paper 2. It didn't come in 2021. So they love to bring it, they love this graph. It's very relevant today because of vaccination and COVID and all kind of things. So yeah, you never know. It's one of the most basic things that you have to learn if you're going into immunology or anything with medicine. So the primary, you have 2 responses, and you can see them here. The primary response, the secondary response. So let me explain here what is the primary response, right? And I'll put it here in like Caribbean speak. So the primary response is like this. You never get sick before from this virus, but you get sick now. So the body does take a little while, it's take a while to actually figure out what this virus is and how to kill it, how to kill the thing. So it have a little delay now between detection and eradication of the virus or whatever pathogen. So what you're seeing here, this is your primary response. So if it was COVID, you know, sometimes if you talk about COVID, they like to strike a video. They feel like you're giving misinformation. I had to bleep myself talking about it. But anyways, basically, if you get alright, so I'm gonna put it here. Imagine like you was to first get infected with COVID. It could be anything, any infectious pathogenic disease. But the first time you get infected, this is what, so you have an initial exposure to, let me say, a certain pathogen called A, right? What this is going to do? So this is going to yield a moderate concentration of short-term plasma cells until the infection is cured. So basically, it's kind of like the body is like, we're trying to figure out what this thing is. We don't want to spend all we energy on this yet. We're still figuring it out, brother. So we have only a small amount of short-lived plasma cells. Yeah? So only have a small amount of short-lived plasma cells. So this could be due to actual infection, like you actually go out there without your mask or with your mask, who knows, and you somehow, you're breathing somebody COVID pathogens, and you get, that's the initial exposure. So the primary response allows the formation of long-term B memory cells. Now, as I said, remember, these B lymphocytes can differentiate into either plasma cells or memory B cells, right? Memory cells. These memory cells remember what the virus looks like. So some of them going to become plasma cells, that's these here, and some of them going to become memory cells. Memory cells don't really do nothing the first time, they just remember. So this may occur after first vaccine dose. Say whether you get your Pfizer or your Sinopharm or your AstraZeneca or the Moderna, not the Johnson & Johnson because that's a one dose, but after you get your first vaccine dose, or it could be due to natural exposure to a pathogen. This is your initial exposure. This would initiate a primary immune response, and this typically might take a few days, might take a few weeks. It really depends on the illness and the severity of it. So the secondary, so remember, this is gonna produce B cells that could differentiate into plasma cells that secrete antibodies or memory cells that is going to remember the antigen. Now, what's happening here the second time? So you don't get exposed to it the first time. Well, you know what they say, whatever don't kill you does make you stronger. So upon a secondary exposure here, you not only have to know how to explain the graph, you have to know how to draw the graph. Like you have to know the shape. Big topic, big topic. So this is your secondary immune response right here. So basically, this is what's happening here. Right? I get infected a second time, or I get my second shot. But now my body know what this virus thing is, and it could get rid of it. Now for now. So I get the virus there, but I feel this time it not staying too long in my body. So remember all the memory B cells that we would have formed from the first infection? Well, they're still there because they're long term. Plasma cells, short term. Plasma cells just come and go. Memory cells, they stick around. They're there for a long time. So upon second exposure to antigen, B memory cells can be differentiated into a greater concentration. So these B memory cells here, these B memory cells here, these here could be like, I remember here now, a comment for you now. So they could differentiate into a bunch of B plasma cells. They will stick around. They stick around. They was hiding in the shadows the whole time. They was waiting. So they differentiate into. So this basically where you get clonal proliferation and all kind of thing. Again, you get a whole set of antibodies being produced. So now you had this should be rapid production, not proliferation. So now you're gonna have rapid production of antibodies. Let's open this type here. What madness I type here? Right, you get rapid. My finger slip. Right, sorry about that. You get a rapid production of antibodies due to a rapid, as you say, proliferation of B plasma cells. So this takes a much shorter time, and you're gonna get a lot more antibody concentration now. Right? So remember, for the first one, your first dose, you're only making a small amount of antibodies. This is the first exposure. The second exposure, you're making much more antibodies. So I don't know about all here, but some people say they feel more sick. Well, me and I all have vaxxed, but some people feel more sick after the second shot. That's typically normal because the second exposure is all these memory cells that was left from the first exposure. They are going to form plasma cells, and of course some of them are going to be long-lived plasma cells, more long-term. That's going to end up in the bone marrow, and some of them going to remain as memory cells. And as a result, now your body it has a lot of white blood cells that kind of remember the disease. Yeah, immunology is a tough topic, I won't lie, but this part here is like an easy part. I really find this part so hard. Okay. Monoclonal antibodies. So what exactly is a monoclonal antibody? So a monoclonal antibody is an antibody made by cloning a unique B lymphocyte. So there's the thing about a B lymphocyte. B lymphocytes, if you was to clone them, like if you was to artificially clone them, they actually cannot secrete antibodies, so they're useless. They're shooting blanks, or they're shooting at all. Antibody secreting B plasma cells actually do not usually divide. So when it's kind of like this, when they divide and they proliferate and they already make a whole set of clones of themselves, they usually do not divide past that. So you cannot just take them and put them in somebody else because they don't naturally secrete antibody. Clone ones don't naturally secrete antibodies, basically. But long time ago, few decades ago, scientists discovered that you could fuse a B plasma cell with a cancer cell. And what you know cancer is known for doing? Cancer is known for multiplying. So a cancer cell, there's just it don't stop multiplying. So this is perfect because what we want to have plenty B plasma cells. So in this case here, if we fuse it with a cancer cell, it will keep multiplying. It's like Dragon Ball fusion, you know what I mean? You give it this power, right? You fuse it by, hey, I couldn't multiply before, but now I could. So this is going to form something called a hybridoma. So this is what you're seeing here, hybridoma. Now, I wouldn't really worry too much about this part because monoclonal antibodies come real big last year, so it probably wouldn't come this year. But basically, they fuse a plasma cell with a cancer cell. It formed a hybrid cell that basically is a plasma cell that could multiply and produce antibodies. And it could form an endless supply of antibodies, and it is called monoclonal antibodies. So these hybridomas can naturally proliferate and produce a large amount of antibodies. So people who this was a treatment for COVID. They formed monoclonal antibodies against the COVID-19 virus. But then it had different variants pop up like Delta and all kind of thing that it probably wasn't so effective against. But it was a treatment because if somebody has a disease, you can actually inject this person with monoclonal antibodies, and they can, that could act as an army reserve to come in and destroy all the pathogens. Okay, I'm gonna skip pregnancy tests because pregnancy tests come last year. Or you could just look at the past paper and you could see, and you could look at the PowerPoint when I send it to all you, when I link it in the comments after, and all you'll be able to read about it then. But I highly doubt that it come in because it come real big last year, like 12 marks and all kind of thing. If it come, it could be for 2 marks. So still read it, but I go waste no time going through it here. Alright, Olia. We are at the end. Types of immunity. We nearly reach. Hey, Olia, we nearly reach. We nearly reach. We nearly reach. No sleep, gang. No sleep. Olia go sleep 11:30 a.m. tomorrow. Nah, don't listen to me. Get all your rest, eh? Get all your rest, eh? No sleep. We gotta wake up. We nearly there. We nearly done. We nearly done. Taking this man one more time. Taking this man one more time.
As the adage says, you give a poor man a fish and you feed him for a day. You teach him to fish. You give him no.
Alright, that's the last time. Let me go. Let me go. Let me finish. We nearly there. We nearly reach. 8 hours only. 8 hours. 8 hours. You teach a man to fish. No. You teach a... No. Let me finish all it. Alright. So... Only we have 4 types of immunity, right? So we say immune responses happen due to exposure of foreign substances or antigens that are attached to pathogens or toxins. So these antigens, remember, these are foreign substances. They trigger the quote-unquote burglar alarm in the body, signals different responses, humoral or cell-mediated, T cells, B cells, all the plasma cells, memory cells, all these nice things. So this immunity occurs due to the production of antibodies from B cells, right? So types of immunity, we can classify them as either passive or active. So what is the difference between the two? Passive is where you don't need to do any work to get the immunity. The thing was given to you basically from an external source. That's the key point here. It was given to you from an external source. So this could be it was injected into you. or it was given to you via breast milk as a baby. So this is stuff you should be familiar with this from CSEC. So passive immunity would be like maternal antibodies from breast milk, from the placenta, from right? So those was passed on to you during childbirth and after childbirth by drinking breast milk. So passive immunity, no energy is actually expended. The antibodies are introduced from an external source. Yeah? And so this is called natural passive immunity. Artificial passive immunity are like where, again, it was introduced from an external source. You didn't do anything to actually form the antibodies. So they could inject a serum into you. Let me say, for example, I get tetanus. I was playing around a rusty nail. I was playing a rusty galvanized old scrapyard thing, and I step on a nail. I'd be like, oh God, boy, I have tetanus. I'll explain what tetanus is to you. Yeah? I never get tetanus, but contract your muscles, contract them, and imagine you can't contract them. It's pain. It's pain all here. It's pain like Cape Unit 2 is pain. But what they could do is that they could take you to a facility, probably not in Trinidad, but probably not for free, but they could take you to a facility and they could inject antibodies into you, a serum or anti-serum. And that there is artificial passive immunity because it is a cure, it is a treatment, it's not a vaccine. It's like again, you get the infection, you inject some antibodies into it to get rid of it. Now the thing about these two, these here, these are not long term, right? So passive immunity, I'm gonna type this here. Passive immunity is not long term or long lasting because these antibodies were not formed by your bodily processes. It was given to you. Active immunity, on the other hand, is where the body is allowed to produce its own antibodies. So there's no introduction from an external source. So this would be like a vaccine, or this would be like if you get naturally infected. So active immunity is long-term and long-lasting, of course, because the body has yielded immune responses to produce antibodies, its own antibodies, I should say. And this could be due to a vaccine, or this could be due to I get infected. A good example is chickenpox. Really acquired active immunity, and this is obtained from the primary response to pathogen exposure. B cells develop antibodies and memory cells. So basically this is, if I was to explain it like this in terms of COVID, this would be like I get COVID, you know, but so I form antibodies against it and I probably won't get it again. Right? So that's naturally acquired active. You would actually probably still get it again because it have plenty variants, but just say it. Natural passive immunity is where you obtain it from your mother, breast milk or the mother's placenta. This is kind of like this. I is a baby, and I just drink breast milk. So I have immunity from that, right? So that's natural passive. Artificially acquired active is the long-term immunization obtained from the primary response due to the intentional exposure to the antigen. This is a vaccine, basically. So this is like I get my COVID shot. I get my COVID shot already. So my body currently forming some antibodies there, in case I actually get COVID I know part I get blocked off, they will be honest. So if I was to actually get COVID, now I could still get it, but my body would have a faster response to it. It would be able to detect the virus in a much shorter time. I would be able to eradicate it in a much shorter time. The symptoms would be highly reduced. It don't mean that I wouldn't get it again. I still could get it, but it wouldn't be as powerful as if I didn't get it. Artificial passive is the short-term immunization obtained from injection of antibodies, such as monoclonal antibodies. So this is like, I get the vaccine now, but I have COVID. So they're going to inject a serum of monoclonal antibodies in me. That is artificial passive. This is not a preventative measure. So vaccines is not a preventive. You know that people be like, I get COVID and I go in for the vaccine. That is not how it is work. The vaccine is a mild form of COVID. It is a antigen. It is an inactivated version of it. It is the RNA from the COVID virus. So it's not a cure. It is a preventative measure. It ought to be COVID. It could be measles. It could be mumps. It could be rubella. It could be yellow fever. It could be anything. Anybody here who I believe in vax. But it could be any of them other viruses. I'm not talking about COVID, but it could be anything else. So this is basically the vaccine or the artificial active is a preventative measure, and artificial passive is a cure. Is that not, well, not cure, is a treatment. Chickenpox is a good example of something you can't really get twice. All right. Vaccines provide artificially acquired active immunity, right? So it stimulates the production of antibodies due to the intentional introduction of an antigen. They are usually multi-dose because usually you have to have a primary and a secondary immune response. They are used for communicable diseases like measles, HPV, which causes cervical cancer, and rubella. Vaccines have numerous modes of action. You should be able to describe at least 3 of these. So you have live attenuated vaccines, and these basically contain weakened forms of pathogens. This is, if you talk about COVID, this is Sinopharm. So what they would do is that they would have a very weakened version of the virus, and they would introduce that into your body, and your body would be able to have an immune response to it, form memory cells to it, and so on. The difference between antibodies are chemicals, are glycoproteins produced by plasma cells. Memory cells are B cells, are differentiated B cells or B lymphocytes that can retain information about the antigen. Memory cells can also eventually differentiate to form plasma cells to produce antibodies. Gabriella. Inactivated versions of vaccines means that they contain the pathogen, but those pathogens cannot multiply. Subunit vaccines contain a detectable component or protein. So basically they'll contain a little spike protein of the pathogen. I think this is AstraZeneca. And mRNA vaccines, which are fairly new, this would be like Pfizer vaccines. They contain an mRNA blueprint of the pathogen. Basically what it does is that it gives the body the instructions to build part of the, not the whole virus, but like a little spike protein of the virus. And then what would happen is that the body would be able to detect that little part of the virus. It's kind of like the body, it's like the body get a blueprint or get a photograph of the virus, and then it could assemble that part of the virus and show people, hey, this is what it looked like. But I don't really think you need to know about the various modes of action so much. You should be able to just describe at least 2. And last slide on it. Last one. we nearly done. We just supposed to talk about vaccine advantages and disadvantages. You should be able to give at least two of each. Of course, the advantages is that vaccines are often successful at stimulating memory cell production, and so they often reduce the severity of an infection, basically make you less likely to die. But vaccines also reduce incidence rate, which means that less people will get potentially infected. So, me I get COVID yet? It's 3 years, me I get, well 2 and something years, me I get COVID yet? And I never get yellow fever. I never get measles or mumps or rubella. I have the vaccine for all that. I never get on them thing. I never get polio or nothing like that. So it reduces the incidence rate. So less people will get potentially infected and spread the pathogen to the uninfected. So this is why it's called herd immunity. The disadvantages of vaccines are this, and we have seen the disadvantages, we'll be fair. We have seen how flawed that they can be as well. Viruses tend to mutate often and they tend to produce variants. We had plenty of variants. We had Gamma, we had Delta, we had Omicron for COVID. And these variants could be very different from the versions that are found in vaccines, which means that they probably have to make new vaccines. Vaccines cause severe allergic reactions. This is called anaphylactic reactions or anaphylaxis. This is rare, however, but it is a real thing, and it could result in blood clots for some people. Again, this is rare, but it could happen. Lastly, vaccines cannot usually be given to the immunocompromised people who are very old, people who are very, very ill. If you give them the vaccine, they may be at risk of even getting more ill from the vaccine. So again, typically, doctors should be consulted before getting any kind of vaccine. And what I say earlier, that's a wrap. Good luck tomorrow. I will link all of these things in the top comment of this video. And if all you don't see it there, I'm going to post on community posts on my channel. I'm going to post the past papers. I'm going to post the PowerPoint there. And well, maybe some notes too. So anyway, all you take care. Good luck tomorrow. All you could post tomorrow and tell me what come. All right, all you. Laters. Take care. Get some sleep. Yeah, man.